and tell about speakers and then instructions.
Yeah.
I'm going live. You can start recording.
Yeah. Okay. A very good morning to all. Today is the third day of the International E-Conference, in the last day. Today we have a list of 5 speakers, and I am introducing on behalf of VIGNAN'S Foundation of Science, Technology and Research, as well as LLB-SCHOOL. So again, I'm introducing myself. I'm Dr. Vishi Nageshwaran, part of LLB-SCHOOL. Today we have a list of 5 speakers. Again, uh, like uh, like today we'll start with Dr. Perugu Shyam, Assistant Professor from the Department of Biotechnology, NIT Warangal. So he's going to talk about small molecules driven drug discovery. So participants, you may stay in mute mode, and while the speaker is talking over the slides, and especially for the Cisco WebEx participants, please post your questions in the chat box. Kindly be in mute mode, and especially please stop your video. Um, except for the speaker, this is the request to all the participants to kindly stop your video and keep in mute mode, because it will definitely disturb the speaker throughout the presentation. So for YouTube viewers, kindly post your comments in the comment box. Thank you very much, and we will move on to the first speaker. So before that, Dr. I think Ms. Rohini will introduce the speaker in depth. Thank you.
All, good morning. I'm here to introduce Dr. Perugu Shyam, Assistant Professor, Department of Biotechnology, NIT. Dr. Perugu Shyam currently working as Assistant Professor in Department of Biotechnology, NIT Warangal. Dr. Perugu Shyam did his doctoral research and secured PhD from Osmania University in the year 2014. Previously, he did his MTech in Bioinformatics and MSc in Biochemistry from University of Hyderabad, Telangana State. He also worked as a scientist from 2013 to 2018 in the Department of Bioinformatics Center, National Institute of Nutrition, Hyderabad. Dr. Shyam's research interests are in the areas of theoretical and computational biology, machine learning approaches, computer-aided molecular design, and chemoinformatics. His group is interested in applying the data science approaches and also developing indigenous software tools for various medical and health applications. On a final note, he is also an amazing and scientifically driven personality, and we are grateful for his presence among us today. Without further ado, please join me in welcoming Dr. Perugu Shyam. Sir, please.
Thank you for the nice introduction.
Sir, can you share the content again? Because I've assigned the role to the presenter, so request you to share the content once again.
Yeah, I'm sharing now.
Yes.
Is it visible now?
Yes, sir, it's visible. Yes.
Yeah. Shall I go ahead?
Yes, sir.
Yeah. So, thank you for the giving the opportunity for the Vignan University, especially say Krupanidhi and Venkateshwarlu and other faculty members from the Vignan University. So, thank you for the nice introduction. So today I'm going to talk on the small molecules driven drug discovery. So why these small molecules are plays an important role in drug discovery approach. So we have the two molecules like the small molecule and macro molecule. So what is the difference between the macro and micro molecules? So in terms of the macromolecules, we have the biopolymers. We have a protein, we have a amino acids, we have a carbohydrates, we have a lipids. Whether these carbohydrates is it considered as a macromolecule? Whether protein is considered as a macromolecule, whether it is a lipid is considered as a macromolecule. So when you talk about the macromolecules, obviously your proteins is a made up of the polypeptides. Okay. So then the protein is existed that we can consider as a macromolecule. When you have a carbohydrates like small molecules, which are the inorganic and organic molecules, they all seems to be macromolecules. Then these macromolecules and micromolecules, some of the molecules are naturally originated and some of the molecules need to be synthesized. Okay. So today I'm going to give a brief on how we can extract the small molecules and where the sources of the small molecules and how these small molecules will help us in the molecular modeling as well as the drug discovery approaches. Okay. So where we can approach in this context, I'm going to approach my small molecules in terms of the present pandemic disease that is called coronavirus. Okay, there is a coronavirus disease. We are calling as the COVID-19 or SARS-CoV-2 because SARS-CoV-2 is a one SARS-CoV is already it's a second time. Then we are calling as a SARS-CoV-2. Then COVID stands for the coronavirus disease. So this is a virus structure. This virus structure is similar to the virus families like herpes virus and SARS virus and MERS virus as well as the HIV virus. This structurally similar, but functionally it's different. Okay. Then whenever you compare these particular SARS virus structure with the HIV virus, the spike protein that is called glycoprotein. There we can call it as a glycoprotein 120 and glycoprotein 40. Then single stranded RNA, an envelope, and esterase proteins. So these are the common functionalities that is there in the HIV. Then see here, this genome size is almost similar to human genome. So 29,751 base pairs in RNA. So this RNA is a linear RNA structure that is there in the SARS-CoV. So when you talk about this, the SARS-CoV-2 have the large genomic structure, and this SARS genome, when we are trying to identify what are the number of the genes, number of the nucleotides that are there, what is the functionality, and which is the expression pattern, and this particular kind of the genomic sequence we already compared with to check the homology between the existed viruses, and we also tried with the bat COVID and pangolin COVID and SARS-CoV-1 and MERS and common cold COVID and influenza A and B strains. Now here you see this is called SARS genome, which we have identified in the nucleotide databases like NCBI and the UCS genome browser. And this is a spike protein, so these act as a protectors, and this is also attached to the acetyl angiotensin converting enzyme 2, that is we can call it as ACE protein 2. So these are the protectants. Okay, then so you can see this is a single stranded RNA structure. This single stranded RNA structure, so wherever the nucleotides are there, these nucleotides will be converted into a protein. And because we need to convert into a protein form, so when we wanted to convert into a protein form, we need to transcribe and translate the genomic sequence. So that time we can consider the open reading frames. So this is an open reading frame and open reading frame 2, and we identified what are the dimers and what are the isoforms that are there in the particular gene. And we also compare, while comparing with the other organisms, so there is a 96% identity to the bat COVID and a 91% is identical to the pangolin and percentage is the SARS-CoV-1 and it is a 55% is identical to the MERS-CoV. Okay, so this is the evolution rate is a 10 to the power of minus 3 nucleotide per year and mutation rate is a 10 to the power of minus 6 nucleotides. Then, so how the virion entry into the host cell, the rate of is as an average is 10 minutes. Okay, so eclipse period is the 10 hours. Then it has to be brushed out when it enters to the nucleated, when it enters to the host organism. So it is a 10 to the power of 3 virions will be brushed out. So how we can identify whether the particular genome sequence is there, the 30,000 base pairs are there. In the 30,000 base pairs, so how to identify where the mutations is occurred and how do you identify a particular disease in a SARS-CoV? So there will be mutations. So that mutations need to be identified in our genomic sequence when you do the molecular biology analysis like RT-PCR as well as the random diagnostic methods. So these point mutations need to be identified. So before that you need to have a sequence. So that is the reason today that the all diagnostic approaches are doing the RT-PCR. So first we are taking the blood sample or we are taking the swab. From that swab we are extracting the RNA and from that RNA we are doing the RT-PCR. So why we need to identify we need to compare the human sequence with the virus sequence. If any mutations that are observed in the host sequence with the virus sequence, then we can able to identify this is the. So see, for example, the TSC and CCA and TGG and GAA sequence, when it is converted to the RNA sequence, it will be like AUG, GG, CGU and ACC and UUU. Then where the mutation is taken place. So this arginine mutation has taken place. So whenever the mutation is taken place, then you need to replace that mutation. Whether you need to substitute, whether you need to insert the completely different mutation, or you want to completely delete that mutation. That is called insertion, deletion, and substitutions. Okay, whether you wanted to substitute A by B or that whenever you are going to replace the amino acid, that amino acid functionality should be similar. So whenever here I wanted to arginine and substituted histidine because arginine, histidine, lysine are basic acids. So without losing the functionality, I am substituting the particular amino acid. So then how to extract the RNA samples, everybody knows, although I don't want to talk more about on the PCR instrumentation, and normally the swab will be collected and it will be transferred into a FNR tube and the NTPCs will be added and buffers will be added and it will be DNA polymerase will be added, appropriate primers and forward primer and reverse primer will be added and it will do the sequencing. So when you do the PCR, so there is a denaturation stage, annealing stage, and extension stage. So their complementary copies will be generated and it will be identified, and this is a virus, SARS virus structure, and we are adding the primer and primer and primer, whether it will be a spike, whether it is a nucleotide sequence. So where we are going to analyze the sequence, that sequence will be identified with PCR machine, and that PCR machine will give us whether it is the patient is a positive or negative. So this case is common. So whether it is a positive or negative, it's okay. So there are the different type of the approaches that are there in the SARS-CoV-2. This is a virus when the infection will be. So this infection also will take place the three approaches. One is the airborne, one is the waterborne, and it is the foodborne. So now, instead of the waterborne, instead of the foodborne, instead of the airborne, this is a physical borne. Whenever you are contacted physically to the one person, to the another person, or whatever the items you have accessed, then later you may access that items. So that physical contact, the virus may be transmitted from the one person to the another person. So then that is the reason the patients will be affected. So these are the communicable diseases will be affected from the one person to the another person. Then infection rate will be increased. So then this infection rate is increases, and we need to analyze the epidemiological surveillance by the RT-PCR anti-COVID SARS or two antibodies, raising the antibodies, and we need to do the experimental studies, and we need to check the over disease stage, staging and prognostication and therapeutic monitoring, and it may recover or it may lead to death because of the disease stage and disease stage. Okay. So the transmission will be initially taking place from the animal to the human being and human being to human being, and so it is started in one place and it is transmitted to across the globe, and there are the symptomatics. So risk factors are there. So these risk factors and symptoms everybody knows. I don't want to talk more about on that. So then disease is there. So this disease, how do you cure? This is a question. So whether the disease is originated and disease is today existed, so how do you bring the drug for a particular drug? Moreover, this disease is there, so communicable diseases. So first we need to identify the target. The target we have seen earlier. The target, naturally, the target identification, it will take, say, 1 to 3 years time. So once you identify the target and we need to prepare the appropriate assay, then that assay may takes 2 to 3 years of the time to execute your experimentations like in vivo and in vitro methods. And we need to identify, once you picks up the literature survey and identification of the target and then assay preparation, then we need to prepare the lead molecules. Then lead molecules will not be prepared by the biologist, it has to be prepared by the chemist. So the chemist will not prepare one molecule or two molecules. So that one or two molecules may not fulfill or that may not show the better activity. So you need to create a library of a molecules. That library of a molecules may be 100, maybe 1000, maybe 10,000. It depends upon the structure activity relationship. So it has to synthesize the compounds in the laboratory. That is one case. Whether the compounds that may be available in the plant sources, then we need to extract the compounds from the plant material. That is the explant, any part of the plant. It depends upon the literature survey we need to decide. Then, some seaweeds, some other sources also available to act as a lead molecule. So then we need to identify which molecule will be a lead from your selected group. Then you need to do the optimization by checking the potency, selectivity, PK and formulation and in vivo screening. Then if the particular molecule will show the activity, then we need to go for a preclinical studies and preclinical evaluation. Then it will goes to the preclinical and animal studies will give a good results, and then it will be sent to the human to check on the clinical trials, and human volunteers will be recruited to test the compounds, and if there also it will be success, then that will be dose will be fixed by the pharmacologist, and that will be sent to the FDA and NDA approvals. Once it will be get approval, then it will be sent to the production stage. So naturally, this discovery process will take the minimum 12 years to 15 years time will be taken. Okay. So this is we have started when the corona is observed in the pandemic stage. Then we have taken the molecules and we have tested the so many compounds we using the virtual docking and natural manual docking approaches, and we have submitted the paper and it is accepted in the journal of virology. So then sources of the drug, as I already discussed, the plant sources and animal sources, mineral and earth sources and microbiological sources, and semi-synthetic sources and synthetic sources, and marine and mineral and geographical origins also available to act as our drug. So some of the molecules will act as a recombinant DNA technology. So especially comes to the COVID-19 cases or SARS-CoV-2 cases. So some of the now the government of India and across the WHO is instructing us to first strengthen your immunity, boost your immunity. So on what way we need to boost our immunity by taking the immunity boosters such as the vitamin C and other elements. So these are the different type of the food items which we can eat daily in our regular practices. Then we can increase our immunity power. So whether we are not eaten earlier, we are eating, but monthly once or monthly twice or monthly thrice. Nowadays, it is daily required in anyone. It's not to take daily the cabbage or cauliflower or amla or jama or coriander or leafy vegetables, but alternative vegetables you can take and you can strengthen your immunity power. Now, the spices plays an important role to kill the virus in your esophagus cavity itself and your mouth. Okay, so these are the a lot of the Ayurveda and Ayush department is instructing us to use the cloves and the turmeric and haldi and this different type of the other spices. They are asking us to make the warm the water with using the 1 or 3 spices and have it. So because there are a lot of the chemical compounds that are available in the spices that are able to cure our coughs and our itching in esophagus cavity as well as breathing problems, it will be cured. So that is the reason we have the plant drug source that is available in the particular material. Okay, so this also act as our drug sources. These are the plant materials. So now you see we are heard the calcium, clidofavir and idoxuridine and brividine and remdesivir. So this escalin will be used as an antioxidant and anti-inflammatory drug, and clidofavir will be used as an antiviral and herpes diseases, and idoxuridine will be used for a antiviral as well as the brividine used for a antiviral. So remdesivir, which we are daily seeing in the news channels and printed media, so this drug is coming, this drug is coming. This is used in Ebola. Okay. So what are the sources of this material? This is a plant. Okay. So these are the plant material which we are using, that is called corylagin in pomegranate and Alcorina gladosa. These two plants will be used for skin diseases and diarrhea, inflammation, antibacterial and antifungal properties and antiviral properties that are there. So then we have identified the compounds that are from the plant materials. Then we have retrieved from the public domain databases, and we have do it the computational analysis. So now this is Punica gramentum, the compound is corylagin. Then ginger. So ginger, we have used the shogal, gingerams. Some of the methods will be represented as a 6 shogal and 6 gingeral. So these compounds Along with these compounds, we have taken the more than 500 molecules that we have screened the compounds more than 1 lakh. And from the 1 lakh, we have selected the 500 compounds. That 500 compounds are retrieved from these databases. These are the public databases which holds these small molecules in the synthesis and natural and other compounds that are deposited in these public domains and which we have retrieved and we have done the computational studies. And this is the Cambridge crystallographic structural database, and these are the molecules. They have started the database in the 1972, and gradually the deposits rate is increasing the year by year. Till today, they have holding more than 1 lakh compounds in the database. And these are the small molecules which we can construct or we can design using the various tools that are available, so that we can design the molecule. If you know the structure, you can design the molecule and you can find their properties like ADMET properties, their NMR and their TLC and their boiling point, melting point, and solubility rate, and the rotatable bonds and heavy atoms, hydrogen bond donors and acceptors, whether this molecule will act as a Lipinski and other things, everything will be checked by the domains that is available in the globe. So then, so whether whatever the molecule which we have designed, so we need to study of a protein crystals that give the details as for the lock. Because we know the lock and key approach. We have a lock and we have a key. So here the lock will act as a receptor and our key will be a drug molecules. So how do we achieve the drug and receptor will bind together? So this is the computer algorithms that are created and in the form of a software. That softwares will be used to check the binding interactions and binding modes between the receptor with the ligand. So these algorithms will help us in identification of a structure-based drug discovery and ligand-based drug discovery. And while doing this, we are in a position to identify the variations in the lock and key approaches. Which structure of the lock should be targeted? Whether your protein is a target, and the protein, which area is your target? Whether your cell wall is a target, whether your enzyme is a target, or some X is a target. So we need to fix up the target. And is the binding pocket is a good target for you, for your studies? And is the structure-based drug design is relevant for my receptor? Is the 3D structure is reliable? Somebody deposited in the crystallographic structure database, that is called RCSB database. Whether that structure is reliable, then if there is no structure, then we need to model the structure using the computational algorithms. That is a homology modeling, comparative modeling, ab initio methods, and threading. Is the binding packet is the static enough? That we need to check. Sometimes more than one binding sites will be there. Then what is your stand on that? So which key is best fits in the receptor that we need to identify? So you need to design the molecules in such a way that the ligand has to go and bind to the receptor. And what are the prerequisite physiochemical properties for the key for a better binding site and better interactions? So everything we need to identify before going to do the computational study. Now, in my library or in my system, I have a group of the compounds. That compounds structurally similar like this, because all my structures are not identical. My compounds, whatever the scaffolds I have, that scaffolds R1 is another group, R2 is a different group, R3 is another group, R4 is a different group, R5 is another group. So different functional groups are I have kept. Similarly, the structures maybe look like this. In these structures, which structure will go and binds to the my active site of a receptor or my binding site of a receptor? Let's see. I have an active site. Active site is like this. This is for easy understanding purpose only I have taken as an example. I have an active site. From this library, which ligand will bind to my receptor? See here, none of the ligands doesn't fit my active site. Then again, I need to do or remodel the structure. So I need to prepare the ligand which can go and bind here, here, and here and blocks the particular active site. So how do we do that? First, we need to identify, this is, let us assume this is the surface. Whatever the surface is here, this surface we are able to identify, and which amino acids is surrounding the active site. In these amino acids, what are the donors, what are the acceptors that are there? Then, how much surface area that is available? That we need to identify that. Basis on this, then we need to design a molecule. So here you have a donor, here you have an acceptor, then we have to O minus and you have to keep it as a H place. Then here you have a H and you have to make it as a O minus. Similarly, tomorrow not only H and O, tomorrow other molecules will be there. Then you need to check which can be fulfilled the hydrogen bonding or which will fulfill the chemical bonding nature, and it will be covalent and non-covalent interactions you need to establish. Then, so you are targeted, the molecule should be there in a corners or which can go and binds to the particular ligands and block the active. But you need to prepare, so which cavity, which the space has to be fulfilled, and which atoms if you keep, that space will be fulfilled. So that we need to do. So that is, whether you may do the pyrimidine ring, you may do the furan ring, you may do the pentane, you may do the hexane, heptane, decane. So like that, we need to prepare the molecule and we need to fix up the scaffold and we need to prepare the molecules. Okay. Then that is the reason we can create more than 1000 or 1 lakh molecules and we can test. That is called pharmacophore hypothesis. We need to define a pharmacophore and which can binds to the receptor cavity and which blocks the active site cavity by the ligands. Such a way we have to design the molecules. I have oxygen here, I have oxygen here, I have OH group is here. Basis on this, between that space which the aromatic rings, furan rings and other rings will, which or who can go and accommodate the space and block the my active site, we need to prepare a molecule and we can design the drugs, and this is called pharmacophore hypothesis, and we can identify the binding regions and evaluate the disproportion in the space and other things, and we can block the. So it is naturally we have done identified, but how we can able to identify in computational, because our so many algorithms are available till today. So and that algorithms will help us in identifying the better molecules and do this. Take the help of the softwares, and there are the free softwares and commercial softwares available to do the molecular docking and molecular modeling. approaches. So from our studies, we have taken the 1000 molecules, and from that we have identified these are the molecules: atazanavir and doranavir and etricidine and etravirine, hydroxychloroquine, dolutegravir and tenofovir, isopropanol and tipranavir, judovidine and azithromycin and remdesivir and clavivir and dexamethasone and tocilizumab and atozanavir. So these are the molecules. Now the other pharmaceutical companies saying that this COVID will be cured by this disease. But whether these molecules are new molecules, these molecules are not new. If you go and check in the drugbank.ca, if you go and check in the PubChem, if you go and check the Cambridge database, these molecules are already available. So there is an approach is called drug repositioning and repurposing. But that approach also we didn't follow to check the whether repositioning and repurposing for this study. So, but whatever the molecules that are using for a previous diseases, some drugs may have a capability to cure this kind of respiratory problems and cough and fever and your other symptoms that are going to be cured. That is the reason the earlier drugs they are using for a repurposing. So this is a drug bank statistics. So many molecules, the number of the small molecules act as a drugs. These all are the FDA approved drugs. When you go to the drugbank.ca, only approved FDA approved drugs will be available. So that are the number of the small molecule drugs are the 11,408. The number of the biotechnology approach drugs that are the 2,162. Number of the other approved drugs, 4,008 drugs are available, and number of the approved small molecules, 2,631. And number of the nutraceuticals. Today, instead of giving the drug, we are giving the nutrition. That nutrition will cure some of the diseases. So that is the reason the nutraceuticals also act as a drugs, and some of the drugs are the experimental drugs. Those are available in the 6,373, and some of the drugs are illicit drugs, and some of the withdrawn drugs, and number of the drugs that are FDA approved drugs is 13,570. But other PDB accessions are 2,12,468 available, and number of the sequences that are available in the 3,371. So if you wanted to check the statistics, you can go and check in the drugbank.ca starts, and it will be given the proper info. So now our approach is started. So while searching the drug bank, then taking some of the compounds from the plant-based approach with the basic idea, basic Ayurvedic approach, and we have retrieved the compounds from which are the drugs having a similar capability to cure and to block the receptor cavity, and those drugs are taken from the FDA drugbank.com and PubChem database, and we have did the computational approach, and these are the targets. Like naturally, the therapeutic targets will be there, or we can call it as a druggable target. The receptors is a 45%, and enzymes will be 28%. Hormone and factors will be act as a 11%, and nucleic acids like DNA and RNA will be act as a 2%. And ion channels like sodium, potassium, and ion channels will be 5%, and receptors is 7%. So these are the druggable targets that will be used for different purposes. Now our approach is started by one way is the identification of a small molecules from the public repositories as well as the traditional Ayurveda approach, and we have scrutinized the compounds, and we approached for the virtual docking. Before that, we also modeled the SARS-CoV virus. That is, one is we have did for the spike protein, and another protein we have did for the PP1A and PP1B processed with mature subunits, and we have searched for the genomic web browser and modeled the NSP1 protein as well as the spike protein. But later our studies, some of the compounds that are deposited in the PDB database. So whether our predicted structure is good or not, we have checked completely with the dope score and with the Ramsden plot, with the Procheck and PROSA, and whatever the structure we have predicted using the theoretical approach, that structure is well prepared, and then we have selected that receptor NSP1 and other spike protein, and along with that we have also taken the other proteins which is already deposited in the public PDB database, and we have completely 8 protein structures we have taken, and we have performed the molecular docking approach with the virtual screen. Then we identified some of the compounds that will bind strongly to the receptor cavity, and that receptor cavity will be blocking the activity, and we have sent to the communication. One paper is accepted for that, and another paper is unaccepted. So these are the results like raltegravir and hydroxychloroquine, azithromycin, and doraviridine. So then these are the small molecular are acting as a drug-like compound, and some of the compounds already drug-like nature is existed and it is published, and they are traditionally or therapeutically using in our studies. So this is with the 6Y8 protein, and this is a main protease with the unliganded, and this is another protein with the sialic receptors with the HCOW OC3, that is the PDB accession number is the 6OHW, and see the interactions they are shown between below the 2.5 angstroms all the molecules, and this is a surface model whether the van der Waal interactions and the Lipinski rule whether it is satisfied or not. So everything we have checked computationally, and this is whether. So this is a network analysis with the ACE2 receptor. So with our known drug molecules, whether our drug may be binding to the properly or not. So if drug is binding to the receptors and which amino acids are subsequently binding, and we are clustered and we have identified the molecules with the expression pattern and localization pattern and other genomic interaction patterns, and everything we have checked in the network analysis. And these are the glide score, and naturally everybody knows the glide score and docking score and other approaches, and we identified the target and we show. Later we have performed the molecular dynamic approaches to identify the how the molecules, the protein and ligand is bound, and where the complex and receptor alone and receptor ligand complex in another. So we have compared the molecules, the receptor alone and receptor ligand complex another way. So then we identified where the structure is binding, and some simulation still going on. So that basic study only I'm showing here, and we need to extract in the GROMAX and other we need to analyze the results. So this is results which we have identified. The molecule still the fluctuating, so it has to come to the stable status. Then we need to be able to identify and what way the hydrophobic and hydrogen bonds and solvent interacting with the receptor. So if you have any queries, please shoot me. So then I can answer them. Thank you.
Thank you, sir. Thank you for the wonderful presentation. So we have few questions. The first question is the Ayurvedic product clinically proved to be beneficial, reduce the infection?
Yeah, the problem is in traditional Ayurveda, the compound is not purified compound. It's a crude. But that crude may have a number of compounds. but that crude may be curing the disease. So our approach is to take that crude, whichever is available in the Ayurveda markets and Ayurvedic physicians that are prescribing, the extract pure compound, and you can test the in silico and in vivo, then you can publish the work. But there is no scientific evidence. There is no scientific evidence on the Ayurvedic compound. That is the lack of
Not be an inhibiting factor. So there's always like not
the crude compound.
compounds within that crude, but will it not be like affecting as such? Will it not affect, inhibit the infection?
The problem is the whatever the plant-based compounds if you take, they don't create most of the side effects like our therapeutic, like whatever the English medicines which we are using, they are not that much side effects. So because the natural source drugs, whether it is a crude or whether it is a bulk drug, they will be a beneficial effects. I don't say that it's not a beneficial effect. Beneficial effects are there, but there is no scientific evidence on that. That is the reason most of the population is taking the allopathic drug, not
Yes, sir. Okay. That we may have side effects. So then what should be the form of administration for those?
No. We have no side effect. Can you repeat again?
Features did you find for these small molecules?
That is a pharmacophore. I have identified 8 compounds which is already using the different purposes like HIV and malaria and dengue. Those drugs only I have prescribed, I have suggested in my paper, and that hydroxychloroquine and azithromycin and doravirine and raclopride is prescribed in the curing the COVID-19 diseases. Because that I have found some of the compounds I have found from the amla and this turmeric powder, as well as this what we call black pepper. These compounds also have a lot of the inhibitory capacity, but that need to be extracted. Another compounds also. I heard some of the doctors say other compounds also have the inhibitory capability for the respiratory problems as well as the gargling problems in our throat. That I need to check further.
Okay, sir. So I think to close with one last question, since viral infection require narrow therapeutic range dosage, uh, what is the methodology or tools to optimize the dosage regimen for small?
For small molecule, have you closed the question? Hello. So that is the reason this Ayurvedic and Ayush department is.
Yes, sir.
Hello.
Yes, sir. I'm able to connect. Yeah. No.
So are you able to hear? Hello. Yeah. See, already the government of India and Ayush department has come up with some of the spices with making this using the spices with making the kashayam like the semi-solid water form. So they are saying that to use the ginger and turmeric powder and pepper as well as the is what we can call it as the haldi, no, that kadai and other spices they are asking to use. But what compound they're going to cure your disease that they are not doing saying properly. So that is the major problem, and that also small molecule. Whatever the problem, is there, that viral problem, that is a nasal cavity and esophagus cavity and the pharyngitis which is affecting initially, that spices will cure, having the capability to cure the esophagus and pharyngeal cavity.
Yes, sir. And thank you very much. And now we will have Dr. Kripanati, sir, to thank you.
So good morning to you all. So many thanks from the organizer side and also from LLB that Dr. Shyam Perugu has participated and gave us the guest lecture in our conference. And since the time Shyam Perugu is known to me, when I attended a conference that this workshop that he organized on biostatistics in National Institute of Nutrition, I've been in contact with him. And he is the board of studies member for our university, VIGNAN'S University. And we keep inviting him for modular courses and guest lectures on biostatistics and other programs. And Dr. Shyam Perugu has given a detailed account of basic concepts of point mutations and real-time PCR. And further, he also elaborated on the sequence of events in leading to the clinical manifestation of the drugs. And also he made a good mention of the phytocompounds and also from the spices, from the vegetables to boost immunity. In addition to that, he's focusing much on the small molecules and also the phytomolecules. So for the participants, I wish to let you know that the phytomolecules are having good demand in the market now. So he has given a lucid presentation on the small structure driven drug discovery. Thank you very much, Dr. Shyam Garu. So thank you. Thank you one and all.
Thank you, sir. Thank you. Thank you, sir.
Thank you, sir. So before we move on to the next speaker, who is Amit Ajit, so we have David Matrioski from USA, who's meant to give a presentation in the afternoon under the student short talks. And since he lives in Chicago, I request him to, and it's time for him to go to bed, and I request him to give his presentation before we move on to the next keynote speaker. So, David, are you able to connect yourself?
Yes, I'm here. Thank you.
Yeah, changing the role to presenter, and you can present now.
Thank you. So I will
And
be presenting. Yes.
Are you going to present a poster or a talk?
Yeah, we have a little talk. We have a little PowerPoint with a colleague named Samir. So he's also on this as well.
Okay, then. Yes, please go on. And we request you to limit the presentation to 10 to 12 minutes to the maximum.
Okay.
And then we move on to the Q&A.
Perfect.
Yeah, now you may share your content.
All right.
Yes.
All right. Is everyone able to see this?
Yes, we are able to hear you.
Perfect. All right. So this is, of course, David Macharovsky and my colleague Samir. Samir, can you hear?
Samir Al-Idrisi, yes.
Okay, so yes, we're coming from Loyola University Chicago on the drug discovery team. And today we're actually going to be talking about a roundup for the clinical efficacies of remdesivir, hydroxychloroquine, and lopinavir, ritonavir treatments against COVID-19. So I don't have to go through much of this, but in terms of current problems with SARS-CoV-2, COVID-19, we know that the pathogen is spreading from person to person, familial, clinical, and community settings. The virus has been detected in blood, oral, and anal specimens, suggesting various shedding through bodily fluid. And now in terms of what we are mostly interested in is with drug development, drug discovery. We know that the discovery and development of new compounds will require an extended period of preclinical testing before they can enter clinical trials. And our lab is interested at Loyola UNM with repurposable candidates. And that's why we had our sites focused on remdesivir, hydroxychloroquine, and lopinavir, ritonavir before we can talk about our compounds that are being tested right now in BSL-4 labs. So, of course, as of now, there are no approved vaccines and specific treatments. We get daily information updates on several clinical data. And we have several day-to-day news on these clinical data. And our goal at Loyola was to consolidate the most important information in order to help guide clinicians, not only at our hospital, but various hospitals in the United States, just because the federal government, we were having a rough time with getting guidance from our federal government. So that was the purpose of this. I don't know.
Hello, do you have any difficulty?
Yes, one, I think.
Yeah, you may scroll, you may actually scroll on, you may actually move on to the next slide as well. It's just like a normal
Yeah.
click.
Trying to, let me see. Oh, perfect, went through. Thank you so much.
Yep.
So again, our three main compounds that we are analyzing is remdesivir, of course, with its original use as a broad spectrum antiviral, several preclinical tests with Ebola and MERS. With MERS, it did not get to the clinical setting, but with Ebola, it went to the field setting, but stopped in its process because other treatments were more efficacious. The mechanism of action for COVID-19 SARS-CoV-2 is it acts as an inhibitor for RNA-dependent RNA polymerase. We have hydroxychloroquine, as everyone knows, original use in malaria arthritis. In the United States, it was approved for emergency authorization, but about 2 weeks ago, the FDA revoked its approval. But again, its mechanism of action in COVID-19 is with its pathology, as it seems like. It does not seem to have any specific target in SARS-CoV-2 proteome. It seems to have an indirect impact on the COVID-19 pathology itself, and this comes from the endosomal lysosomal alkalization as well as the glycosylation interference. And lastly, we have lopinavir ritonavir with its original use in HIV, mechanism of actions, inhibiting papain-like protease and main protease 3CL-Pro. This is from one of our communicated papers for a similar review. This is the illustration of the SARS-CoV-2 life cycle, along with remdesivir mechanism of action. So we have remdesivir, that is a prodrug. It is metabolized to its active form GS441524, and this is actually the compound that inhibits RDRP. We have the two major polyproteins from the two ORFs that are processed by the two proteases right here, 3CL-Pro and PL-Pro, that leponavir, ritonavir are suspected of inhibiting. And then we have hydroxychloroquine, which interferes with SARS-CoV-2 endocytosis by increasing its pH within the endosome, as well as the hydroxychloroquine interfering with the glycosylation of the virus, resulting in immature virions. So Samir, if you want to talk about the clinical
Can
trial.
you guys hear me?
Mm-hmm.
Okay. Currently, we have a current clinical review paper under consideration, reviewing an array of preclinical and clinical studies on RDV, hydroxychloroquine, and LPVR treatment against SARS-CoV-2. So here we have three key clinical studies that were analyzed in that paper, analyzing RDV treatment for COVID-19. An observational clinical study, as well as two controlled clinical trials. So in the observational clinical study, compassionate use of RDV was granted to 53 patients. And it was quite promising because 68% of the patients witnessed an improvement in oxygen support, as well as a 13% mortality rate was also observed. However, in a randomized double-blinded placebo-controlled clinical trial, RDV treatment was compared to a placebo control group, and no significant difference was found between the two groups. Further, an ongoing randomized controlled clinical trial is currently being done by the NAIAD in the United States. This is a fairly large study with over 1,000 patients enrolled, and they are comparing RDV treatment to a placebo control group at varying days of treatment. Currently, preliminary clinical data shows an 8% mortality rate for RDV treatment compared to 11.6% for the control group. Now, RDV's prodrug requires chemical metabolic processes for full activation. And because these metabolic processes could potentially be in specific cells, and we don't exactly know what tissue types and types of cells SARS-CoV-2 could potentially infect, there could be reservoirs of SARS-CoV-2 in specific tissues that we don't know about when we treat patients with RDV. So because of that and other lack of clinical data, RDV is still in the infancy of understanding for antivirals against SARS-CoV-2. You can go ahead to the next slide. So for hydroxychloroquine, here we have 4 clinical studies analyzing its treatment against COVID-19. We have 2 controlled observational clinical studies, a retrospective clinical observation, and a randomized controlled clinical trial. So for the first controlled clinical observational study, as well as the second controlled clinical observational study, no significant difference was found between HCQ and patients that were not administered HCQ. Further, in an open-labeled, multi-centered, randomized controlled clinical trial, no significant difference was found between patients granted HCQ treatment with standard care versus patients only given standard care. Additionally, in the retrospective clinical observational study, 4 different treatment groups were analyzed in a large patient pool. I believe it was done in France. There were various combination therapies looked at with hydroxychloroquine alongside azithromycin versus their retrospective monotherapies, so just HCQ or just azithromycin, and no notable difference in the mortality rate was found between those 4 groups. So next, we have 4 key clinical studies looking at LPVR treatment against COVID-19. And that includes 2 randomized controlled clinical trials, a retrospective clinical study, and a controlled observational clinical study. So with the first randomized controlled clinical trial, patients administered LPVR treatment versus solely standard care over the course of 14 days witnessed no difference in clinical outcomes. Further, in the multi-center randomized controlled clinical trial, patients granted LPVR, INF-beta, ribavirin triple combination therapy versus a monotherapy control group only given LPVR, improved clinical outcomes were witnessed in the triple combination therapy. This shows promise for possibly drug cocktails alongside liponavir and ritonavir that could be more beneficial in combating SARS-CoV-2 versus just the monotherapy. Further, in a repetitive clinical study, LPVR combination with arbitol versus only LPVR monotherapy was analyzed, and it was found that LPVR alongside arbitol had a significant improvement in chest CT scans of patients versus solely LPVR treatment. And then finally, in a controlled clinical observational study, patients treated with LPVR versus only arbitol were found to have more favorable outcomes if they were treated with arbitol versus only LPVR. Now, what we can see here is LPVR seems to be a bit more promising than the other two antivirals, especially if it's used in combination alongside a drug cocktail, especially in that trial with INF-beta and ribavirin. So I would be interested in more clinical studies looking into those combinations.
So from this, with our clinical guidances under Dr. Dervasul and Kampaya at Loyola, first of all, clearly further robust double-blind large sample clinical trials are needed to evaluate suitability of these potential treatments. The mechanism of action for these compounds, specifically to see hydroxychloroquine if it has a specific target in SARS-CoV-2, is critical for anti-COVID-19 therapies. A recommendation is the inclusion of more world-approved as well as experimental drugs to assess the possibility of repurposing. Through this, clinicians can identify best combinations of compounds that may be of greater efficacy against SARS-CoV-2 compared to monotherapies, where we see that these single-based compounds tend to suffer. There is a possibility that these previously mentioned compounds may earn their place in the clinical realm as proper treatments for COVID-19 and may prove to be components of combination therapies rather than the manner that they are currently being used. Until a SARS-CoV-2 specified compound is developed, then the most direct way to find treatment will be through a multifaceted drug repurposed approach, like remdesivir, like hydroxychloroquine, like lopinavir, ritonavir, and any other compounds that we'll be having soon. And then just a little bit, thank you. So our background, again, is drug discovery, drug development. We have several compounds that we're lucky to be testing at the United States Army facilities. So thank you to Dr. Dine, Dr. Bradfute, and then the rest of the team. And then thank you all for letting us present just a little review of some of the compounds that we're getting ready for the United States. Thank you.
Thank you.
Thank you. Thank you very much. And questions? Anybody? Pranav, do you have a question?
Hello, David.
Hello.
Yeah, so you are suggesting a polytherapy, right? So what will happen?
Yes, go ahead.
Yeah, so what would happen if we give both? Do they interfere with each other in the mechanism anyway? Because due to polytherapy and their administration, though these two drugs are used for different purposes currently, and we are testing it for COVID now. So how do you see the effects for the polytherapy would be?
Yes, so specifically, we are interested in potential hydroxychloroquine with a specified antiviral combination. We've seen more data that leads to hydroxychloroquine acting as a prophylactic because it does not have a direct mechanism of action against the virus. So we are testing right now in various animal samples of various treatments of hydroxychloroquine as prophylactics and remdesivir as an actual antiviral that is able to directly affect SARS-CoV-2. We believe that adding an antiviral to an indirect hydroxychloroquine can be a reputable therapy. And so that's what we are planning as of right now. That's our studies.
Thank you. Thank you for that.
Thank you.
So
Thank you both, and that was a nice presentation. So now we move on to the second keynote speaker of the day, Dr. Amita Ajit. So I give a little introduction about her. So Dr. Amita Ajit is a doctorate in stem cells and regenerative medicine from Sri Chitra Tirunal Institute for Medical Sciences and Technology, Trivandrum. The core area of interest has been stem cells, chronic wound healing, and regenerative product development. She has been associated with various science and technology research programs in reputed national institutions of the country to scientifically or productively contribute towards the betterment of human health through state-of-the-art R&D and science and technology administration. She received the Young Scientist Award for Biotechnology from the Government of Kerala during the 24th Kerala Science Congress in 2012 for the study on the development of a TB vaccine strategy using major immunodominant proteins from Mycobacterium tuberculosis. She's a life member of the Kerala Academy of Sciences and a reviewer for numerous international journals in the field. The current focus of work is on development and commercialization of biological therapeutic products for human use at national and international sector. She currently serves as a consultant International Private Limited, Chennai. So here we welcome you, ma'am. You may proceed with your talk.
Divya, am I audible?
Yes, you are. Yeah.
Okay, very good morning to one and all of you, and thank you so much for having me here.
Ma'am, can you be a bit louder?
Oh, yes, please.
Yeah.
Am I louder now?
Yes. Yes. Yeah.
Yes. Okay, I think I have given my slides to Dr. Abraham so that you could present it for me because I'm having some internet problem. Have you
Okay.
received my slides?
Ma'am, do you have a headphone?
Yes.
Yeah, you can keep it a bit more closer, I think. Some people say it's still not audible.
Oh, well, it is placed pretty close enough. I think there is some network problem.
Okay, okay. Okay, so I'll share the content at the moment.
Yes, please.
Yeah. Yes, can you able to see, ma'am?
Yes, please. Yes.
Yeah.
All right. I'll go slide by slide and so you can help me in changing the slides.
Yes, sure.
Is that fine? Yeah.
Yeah.
Well, so thank you so much for the beautiful introduction, and it's great pleasure being here. So being a stem cell scientist, I envisaging adipose stem cell secretome for potential cell-free COVID-19 therapy. Can we go to the next slide?
Yeah.
Well, as it is already well known, and I believe most of you are very familiar with what COVID-19 is, but in brief, it is a severe acute respiratory illness that is caused by the coronavirus 2. And we know that it delivers a very angry inflammatory cytokine storm, which is more or less a host-dependent action. So it is an immunologic complication which leads to the requirement of an intensive care unit admission if it goes beyond control. Coming to the next slide. As you can see here, the major cytokines as well as the chemokines that are released once the COVID-19 is infected. There are a large number of cytokines as well as chemokines that are released by the body by the human host macrophages or the T lymphocytes and the dendritic cells. And the release is in fact beyond control, and you have in a normal condition, you see that the virus infected cells are either destroyed by the natural killer cells of the innate immunity and the CD8 positive cytolytic T cells of the adaptive immunity, leading to the apoptosis of the antigen presenting cells as well as the relevant cytotoxic cells. However, in a defective scenario in the lymphocytic cytolytic activity due to either or an acquired condition like that seen in the COVID, there is an inability of the natural killer as well as the cytolytic CD8 T cells to lyse the infected and activated antigen presenting cells. So because of this scenario, inflammation is sustained, prolonged, and there is no subsequent overlapping of the proliferative phase. And this scenario leads to the detection of high levels of the interleukins as well as the interferons in the patient. And in such kind of scenarios, where there is no progress at all, it leads to respiratory insufficiency. Can we? Yes, it goes to respiratory insufficiency. There is an immune attack of the body, which leads to multiple organ failure as well as death. Currently, there are no therapies against the SARS virus, but we have only measures to reduce the infection as of now. And these measures include, next. Very common measures that you all might be very familiar with in travel restrictions, containment, mitigation strategies, as well as the total lockdown. Now, coming to this particular scenario where we have absolutely no specific therapy, there demands an urgent need for an efficient therapeutic strategy. Next. Next. Well, now what are the, as of now, therapeutic attempts that are present? You might have heard for the last 2-3 days, I mean, large number of talks on the various therapeutic attempts that are being done, pure antiviral agents, immunosuppressive, as well as immunomodulatory drugs that are essential for treating the particular disease. We have corticosteroids, chloricones, hydrochloricones, and even antagonists against the inflammatory receptors, as well as TNF inhibitors and the Janus kinase inhibitors, and a lot more to say, which are in fact not a 100% solution at all. And unfortunately, they do not act against restoring the inflicted lung damage. In such a scenario where you have a disease that is going on in prognosis, and at the same time, you have a disease that is damaging the entire organ, what kind of a therapy could you go for? So here comes mesenchymal stem cells. Mesenchymal stem cells are in fact cells that are based in the human body in various parts of the body, in various tissues, as you can see here. It can go to the bone marrow, adipose, dental tissue, the umbilical cord tissue, as well as the placenta or the amniotic cells. And these cells reside in secret niches within these tissues, and they respond to injury. Now, looking into, can you press the next? You can see that adipose tissue, as you can see, is an easily accessible, patient-friendly, and easily to isolate cell source. And you have abundant quantity of the cell when you isolate them from the adipose tissue. Next. Here in this slide, I've shown a picture, a pictorial representation on how adipose tissue can be processed. You have adipose tissue.
Oh.
These stem cells can be confirmed as per the international standards for stem cell identification, where a panel of positive markers and negative markers can be checked in, as well as its ability to differentiate into multiple cell lineages. Next. Now coming into the different aspects on why mesenchymal stem cells are really interesting. You can see from this picture that mesenchymal stem cells, they have the capability to release a large number of secretome or cytokines or growth factors or molecules that can target in reducing inflammation, modulating the immune cells, fighting the apoptosis as well as fibrosis, improving the angiogenesis, as well as regenerating the tissue causing an injury, as well as it has the ability to differentiate into multiple cell types, like the bone, the cartilage, or the fat. And in the meanwhile, it can self-renew and maintain its pole. So considering this particular aspect, we can see there is regeneration happening and there is repair happening. So a perspective where regeneration and repair can be brought along together is a very promising solution for treating COVID scenario. Next. Well, as I have asked the question, why mesenchymal stem cells for acute respiratory syndrome? Mesenchymal stem cells have been extensively used in the previous years in both preclinical as well as clinical studies, suggesting their safety, efficacy, and they are free of ethical as well as social issues. Moreover, the paracrine factors, as I have mentioned before, they are very important and they have a very major role to play in the immunomodulation as well as the anti-inflammatory effect mediated by the growth factors like transforming growth factor, hepatocyte growth factor, and the interferon. Again, they do modulate proliferation, activation, and effective functions of all the immune cells. This gives the potential to also differentiate into the alveolar epithelial. In a COVID scenario, we do see that wherever there is an AC2 receptor, which is very prominent in the alveolar epithelium, as well as in the capillary endothelial cells, there is immense destruction of the cell. However, because of the action or the paracrine effects of the mesenchymal stem cells, there are evidences that these particular cells can be regenerated and repaired as well as replaced. Moreover, mesenchymal stem cells are also known as an antiviral therapy, and they have been cited by the Kyoto Encyclopedia of Genes and Genomes. So coming to all these factors put together, they seem to be a very promising candidate. Next. Well, here in this slide, I have demonstrated how this is an experiment that was done by a group somewhere in the UK, where you can see the mesenchymal stem cells were injected in a model, in a mouse model, having an acute respiratory syndrome. The stem cells are seen to activate the PGE2, that is the prostaglandin 2, thereby increasing IL-10, which is an immunosuppressant, and reducing or downregulating the inflammatory markers like the TNF-alpha and the IL-6. As well, it releases a lot of growth factors like VEGF, the keratinocyte growth factor, and the hepatocyte growth factor, which play a major role in epithelial cell proliferation, protection of the pulmonary vascular permeability, prevention of the endothelial cell apoptosis, as well as in inhibiting the neutrophils. The neutrophils play a major role in inflammation. So once they inhibit the neutrophils in the bronchoalveolar lavage fluid, we can immensely reduce the inflammatory action. Next. Looking into the potential of mesenchymal stem cell induced lung repair, as I explained earlier, an additional point is its ability to transdifferentiate into the alveolar cells. So this is done by a pathway of Wnt5a induced activation of non-canonical Wnt signaling. And here there is successful differentiation of the mesenchymal stem cells into the alveolar-like cells based on the paracrine effect again. So you can see repeatedly I've been using the word paracrine. So majority of the impact of the stem cells that are transplanted are more or less because of its paraclinical action. Coming to the next slide. Well, here, in summary, we can see that mesenchymal stem cells play a major role in antifibrosis, angiogenesis, antibacterial effect, chemoattraction, immunomodulation, proliferation, neuroprotection, anti-apoptosis, all by a huge number of growth factors that are released by the mesenchymal stem cell. So the major picture we get here is that these cells in a COVID scenario can alter the behavior of both the adaptive as well as the innate immune cells. They can enhance the restoration of the capillary barrier for the oxygen and the nutrient supply because, as I mentioned, angiogenesis, VEGF-A is released immensely by the mesenchymal stem cells, which later on help in the restoration of the endothelial cells which are present in the capillary. Therefore, there is a restoration. Again, the mesenchymal stem cells can differentiate into the native cells of the epithelium, and again, they are free from infection by the SARS virus. There is a report that says that mesenchymal stem cells are completely free because of the ACE2 receptor. Again, mesenchymal stem cells produce antibacterial properties and are able to directly suppress the bacterial. So altogether, we can say that the paracrine role is much better than the autocrine role of the stem cells. Next, please. Next, please. Well, this is a case study on the achievements of mesenchymal stem cell treatment in COVID-19. I have taken major examples of cases and studies that were done in China. You can see here that in this particular experiment, 7 positive patients, among which 4 who had severe symptoms, 2 with common symptoms, and 1 who was critically ill, were treated with clinical grade human mesenchymal stem cells. And this was administered intravenously in a quantity of 1 × 10⁶ mesenchymal stem cells per kilogram of the body weight, while their condition was worsening severely. And in a period of 14 days, they were put into observation. And surprisingly, the symptoms subsided under 2 to 4 days after infusion. There was a complete regulatory phenotype of the both dendritic cells as well as the CDT cells. Again, a CT scan of the chest showed relief, improvement in the infiltration or the lung inflammation. So later on, you can say patients were reported negative for the SARS nucleic acid test within 7 to 14 days after the infusion. So this is one promising study. Coming to the next. Next slide, please. This is the second case. This was also a study that is done in China. Here, a very critically ill ventilator-ridden COVID-19 patient was infused with 3 times of 5 × 10⁷ stem cells isolated from the human umbilical cord at an interval of 3 days. And the patient was able to walk within 4 days of her second cell infusion. And this patient was a pretty old patient. The essential parameters like the T cell counts were completely restored back to levels. So this is again another promising fact or promising indication of stem cells. Going to the next. Next slide, please. Yeah. So till date, there are about 17 complicated clinical studies along with 70 trials registered on the clinical trial government portal, which report positive results in treating the respiratory disorders using mesenchymal stem cells. In particular, there are 2 on the use of dental pulp-derived stem cells, 5 on the umbilical cord-derived stem cells, and 1 on mesenchymal stromal cells, as well as there is one interesting study on the use of mesenchymal cell-derived exosomes via inhalation route. So despite of the delivery or the route of delivery or the source of the mesenchymal stem cells, an overall basic pre-assessment study says that mesenchymal stem cells are definitely compatible and promising for COVID-19. However, it is also seen that despite these promises, in previous studies, apart from lung-based treatments, in an overall basis of cell-based mesenchymal stem cell therapy, it is seen that stem cells upon administration, they have poor survival, poor engraftment, there is a lack of efficient delivery mechanism, and there is an insufficient biodistribution of the transplanted cells after an intravenous administration, and the low survivability of the transplanted cells, and the lack of homing to the injured site area, as well as immunological responses causing its rejection, and a lack of a tracking system on the fate of the transplanted cell. We really do not know where these cells go about. What happens to the transplanted cell? Do they really differentiate? We do not have a proper tracking scenario for that. And again, it is a very expensive treatment strategy. The manufacturing process, as well as the delivery and follow-up costs are too expensive. So in this particular context, how do we really take the benefits of the cell without using the cell? That is where cell-free therapy overcomes these limitations. Coming to the next slide. Yes, cell-free therapy is nothing but using mesenchymal stem cell secretome. A secretome is nothing but all the growth factors that are released, all the growth factors, cytokines, and molecules that are released by the stem cells into the culture medium when they are cultured. So using that particular secretome instead of the cell alone is a great advantage in overcoming all the basic limitations of cell-based therapy. So our current understanding, as I had explained before, it has been more or less on the use of paracrine factors of the cell that brings about a therapeutic impact. So our current understanding on the mechanism of action relies purely or more or less on the paracrine interactions with numerous different cells in the host body. So administering secretome alone is hence a very promising approach. Moreover, this reduces the risk associated with a direct cell injection while maintaining the efficacy under wide manufacturing scalability and modification potential like fractionation, concentration, combinations with various carriers that are required when a whole cell is to be transplanted. And more or less, this permits a very large scale cost-effective production and easy storage. So coming to the next slide. Well, the question comes is, how do we actually propose or think about a cell therapy? This is one small example or a pictorial representation where it is aspirates are taken out from a hospital. They are processed. Is there an
1 second, ma'am. Participants, please be in mute mode. Participants, please be in mute mode. Okay. Ma'am? Yes, ma'am. You may. Yeah. Can you hear me?
Yes, I can hear you.
Yes, ma'am.
Well,
Yeah.
coming to the approach for adipose mesenchymal stem cell cell-free therapy, I stress on the word adipose mesenchymal stem cell only because of the factor that it can be easily accessible for isolation from patients, unlike bone marrow, which is very complicated. And again, the number of abundant cells that you can get from the adipose tissue is much higher than from the bone marrow or any other source. So considering these two major advantages, I have been emphasizing on the terminology of adipose-derived mesenchymal stem cells. More or less, mesenchymal stem cells from all different sources do have the same impact of paracrine activity in case of healing as well as therapy. But based on convenience, adipose mesenchymal stem cells is the best proposed source of stem cell isolation. So in this context, on deriving an adipose-derived mesenchymal stem cell, you can precondition or culture these stem cells that are isolated to multiply them into larger numbers. And this can be done in a bioreactor, which is more cheap and efficient, and this is a better good manufacturing practice. And the secretome alone can be collected. Secretome should be analyzed by molecular profiling methods like mass spectrometry or protein estimation, as well as Maldito, like after which they are to be ultra filtered, all kind of contaminants, and then freeze dried and then stored into lyophilization, suitable control storage method. And this file can then be transferred for a patient. Coming to the conclusion, I can say adipose-derived mesenchymal stem cell secretome could attenuate inflammation and regenerate the lung damage caused by the COVID-19. Again, traditional immunomodulatory capacity of common immunotherapies are factors which may not induce a strong enough response. When you look into the traditional or the on-the-counter medicines, we have anti-inflammatory. Most of the anti-inflammatory drugs are targeted against one or two factors, or immunotherapy also targeted into factors. But when you use mesenchymal stem cell secretome, it is a cocktail of n number of factors and growth factors that can, in fact,
Yes, ma'am. Are you okay? Hello, ma'am.
this is a representation of using.
Are you able to hear me, ma'am?
Hello.
Hello, ma'am. Are you able to hear me? Hello, ma'am.
Yes, I'm able to hear. Yes, is there an interruption?
Yeah, okay.
Yes, hello.
Yes, ma'am. There was an interruption. I think we couldn't hear you in between.
So, oh, all right. Should I repeat?
Yes, ma'am. Yes. Yeah.
All right. I was saying that in the conclusion that the adipose stem cell secretome is a cocktail of number of growth factors that target not just one or two of the issues or critical injury issues that you can see, but rather n number of them together at a time, unlike the conventional OTP medicines. So it is beneficial on treatment alone or in combination with other therapeutic agents in yes,
Technical. Yes, ma'am. Yeah, there was. Hello, ma'am.
Okay.
Your voice was breaking. Yeah,
Is it
yeah.
clear
Now it's fine, ma'am. Now it's fine. Yes.
All right. Okay. I'm sorry for the interruption. I think there is some
Okay.
network issue over here.
Oh, I see. I can hear you now, ma'am. I think, yeah. I think it's just the concluding part. Yeah.
Yeah. So stem cell signaling, in short, whether you use the stem cell or more or less the secretome, it can 100% help in reduction of the inflammation as well as the oxidative stress. It will stabilize the endothelial fluid lineage as leakage as well as the improvement of the alveolar capillary barrier, and it can promote the tissue repair. So more or less, it is beneficial for use alone or in combination with any other therapeutic agents in people with COVID-19. Coming to the next slide on future perspectives. The next slide. Next. Yes. So coming to the future perspectives, I've tried to make this talk as short as possible because of the technical problems as well as the internet problem. So you will be free to ask me any kind of questions. I'll be happy to answer. But as of now, coming to the future perspectives, we do know and we are very much aware of the potential of mesenchymal stem cell secretome as of now. But what is lacking at the moment is a specific human tissue fat biobank, whereby people can actually take in the human tissue for processing and then derivation of the human secretome. Again, some MSC-based treatments in acute respiratory syndrome resulted in an excessive suppression of the immune response in the injured lungs. Hence, there is a requirement for an optimal dose optimization. And it should be clearly defined with the aim of finding the right balance between their beneficial as well as their undesired effects, which would happen due to immunosuppression. Again, it is very critical that the production of adipose-derived mesenchymal stem cell secretome is in compliance with good manufacturing practices and would follow strict regulations prior to being approved for use in humans. Finally, there is a 100% requirement of a standard reference to eliminate batch-to-batch variations of the secretome. These cells being derived from humans, and there will be 100% donor-to-donor variations. So it is important that the final product is screened for uniformity at the end. So coming to the next slide. So I would like to end my talk with great hope on adult stem cells revolutionizing medicine. Thank you all.
Thank you, ma'am. Thank you. Despite all the technical difficulties, it was a wonderful presentation, and more research on standardizing and optimizing the MSC dosage is very important. And thank you very much, ma'am. And we have some questions to ask.
Please ask me.
So the first question is, what is the expected action plan in India on availing MSC towards COVID-19 therapy?
Well, in India, there are a few companies like LifeCell itself who has a very good stem cell banking facility, as well as we have Stemputics, that is another company located in Bangalore. They are also into stem cell banking, and they are actually looking into the same aspect on how they could bring about stem cells for COVID-19 therapy. So these are all initial investments that they have done, but there is a long way to go for this. We have to do a lot of clinical trials on dose optimization, as I said, which is very, very critical in before administration. And again, there are questions still on whether intravenous is much effective or it can be inhaled, which would be the better option, whether it would be the exosomes alone from the secretome or could it be the secretome as a whole. So these are all few questions that are still unanswered throughout globally, in fact, and India is definitely in the forefront and giving its best try to get things done at the best in the most cost-effective manner.
Thank you, ma'am. And why can't we make this mandatory to preserve the stem cells from umbilical cord due to its widespread therapeutic application?
Well, this is a very ongoing strategy right now. As I said, LifeCell is India's #1 in stem cell banking, and they have tie-ups with almost all the hospitals throughout India as well as in parts of the Middle East, where they have tried to emphasize the requirement for storing umbilical-based stem cells. So umbilical cords are actually stored and collected from different donors, and this is an ongoing process. It is happening in India.
And what kind of, so you talked about the human fat bank and what kind of fat tissue bank, right, ma'am? So what kind of tissues that you normally store? Like, is it like the adipose tissues that you mentioned?
Correct. In fact, you know, the procedure called liposuction, it is right now done in
Yes.
various parts of India as a beauty therapy. So people
Yes.
who are
Yeah.
very fat, who are very obese, they go for
Yeah.
all these kind of liposuctions, and you get litters of adipose tissue. So this is one, it's a waste that is thrown off or discarded by the hospital. But this is an attempt where, you know, companies can actually think of, you know, collecting the adipose tissue or the lipoaspirates and storing it in banks, just like any other organ bank that you have right now. This is something that is lacking right now. So a human tissue bank is very, very important. And there is another option. You need not go for an allogenic transplantation. You can even go for an autologous self-tissue banking, where you just need 5 grams of the tissue, which can be done by a very minor liposuction in any region, maybe from the tummy or from the thighs, wherever you have fat in your body. You can
But
easily
how do you deal with the incompatibility issues or something like that, ma'am? Or autoimmune?
Yeah, there is no, you know, the good thing about mesenchymal stem cells is they do not have any immunorejection problems. So they are MHC compatible, and that is why you can use any donor cell. So it is allogenic donors are also very much compatible. You need not go for an autologous transplant. And especially in patients who are old, like aged patients, it would be more stressful to put them into an autologous transplant. So it would be much practical to go for an allogenous donor.
Okay. And the question, oh, I see. There's some
Sorry,
record
Divya?
going on. Participants. Am I able to see some the record of myself? No, and the YouTube is already in mute mode, I guess. No, the YouTubes are in mute mode, and now I think it's fine. Yeah.
Yes.
Ma'am, there's another question, like how fast is the restoration by MSCs in comparison to the growth rate of the virus?
Well, that is what based on the current clinical trials that they have reported so far, as I had mentioned, shown you a slide on the work that was done in China. It is seen that in 4 days, some in 2 to 4 days, there is immense change and recovery from the virus. So they have even shown that COVID patients are later on seen negative by the 7th day. This is promising.
Another question to finish, maybe if I had listened to it properly during scrolling of the slides or not, but why is it not feasible to actually supply these MSCs? You know, because you said I think it's very expensive, but why can't it be brought down to make it available at the current situation?
Well, one thing is, the most important factor is you have to produce these stem cells in a very good manufacturing practice compliant facility. We do not have numerous facilities in India as such that have stem cell compliance. So that is one problem. And the second thing is right now the culture media, the procedure for growing these cells and making it scalable is pretty expensive. The reagents that are used are pretty expensive, but there are numerous studies and attempts that are being done to bring it into a cost-effective manner. So if not right now, but maybe in the few years to come, they will tackle a new methodology on how to get this cost effectively. We are hoping for that, in fact.
Okay, but there's another one that again I felt like asking, and does the world think that stem cell therapy is still very cosmetic rather the need of the hour?
Well, I don't think so. In fact, I think there are numerous groups studying throughout the world. If you look into the last 3 to 4 years of publications that are put in, as well as clinical trials that are put in, a huge number of people are thinking the other way around, and they are focusing mesenchymal stem cells more on therapy rather than cosmetics. There is 100% interest in cosmetics, but more or less efforts are being put for therapeutic benefits. That is what the current statistics show. So, and it is promising also. But as I said, transplanting the cell as such to the human body requires a lot of optimization, especially on the dose. How many cells do we give over? Do these cells differentiate? These are cells that have capacity to differentiate into different forms of cells that are seen in the body. So there are questions that are still unanswered. Do these cells go into the body, differentiate into the required or the desired cell types, or do they go for undesired cell type differentiation based on the cues that are present in the site of injury or where they're located?
But what if you had pumped in more cells and then would the body not undergo a natural programmed cell death?
Well, these are questions that we have not yet addressed in this particular scenario. We do not know. We do not know. But as of now, there is no issue on safety or any risk that is being reported by using mesenchymal stem cells from adipose tissue. But still, there are questions. Like I have just now a few in my last slide, I have explained how stem cells, when you were given in a higher dose to COVID-19 patients, the inflammation went too down. You know, it was down regulated to an extent that was beyond requirement. So dose optimization is a very important factor in stem cell transplantation. And it is lacking. We have to do a lot of studies, a lot of trials in human subjects. Only then we will be able to give a 100% conclusive answer on what exactly is the number or what exactly is the dose that can be given to a patient.
Thank you, ma'am. And thank you for patiently answering to all our questions. And it was indeed a wonderful presentation. And I personally felt very interesting. Thank you very much, ma'am.
Thank you so much for this session and the opportunity. I feel very bad that I couldn't actually present this properly because of the technical issues that we were having. You know, one person sitting on the other side and changing the slide. It's a little bit difficult. I understand I have troubled you also.
Now the thing is, like, we had to look into the technical stuffs as well at the same time, but indeed it was not very difficult to handle, and no problem, ma'am, and thank you very much for the presentation.
Thank you very much, Divya, and thank you very much to the organizers also for having me here. It's been a great pleasure.
Thank you. And now we move on to the next speaker, Dr. N. Sandeep Kumar. He's the Director of Clinical Development, Genesis Diagnostics, USA. And he's going to talk about COVID-19 and molecular diagnostics. So Rohini is here to introduce him. Rohini, can you please?
Yes, ma'am. Good morning, all. I'm Rohini, CSR SRF from VIGNAN'S University, and I'm here to introduce Dr. N. Sandeep Kumar, Director, Clinical Development, Genesis Healthcare Private Limited, USA. Dr. Sandeep completed his bachelor's degree from Bapatla Engineering College in the year 2008. He completed his MS in Middlesex University in the year 2007, and he completed his PhD in University of London in the year 2012. He did his MBA from the University of Grenoble, France in the year 2019 and postdoctoral fellowship in the year 2018 from French Atomic Energy, Grenoble. He is more into the area of research, clinical research and development. On a final note, he is a fascinating person and we are grateful for his presence among us today. Without further ado, please join me in welcoming Dr. N. Sandeep Kumar. Sir, please.
Sir, you may share your content. Uh, Sir, are you able to share your content? Sir, are you available? Sir, can you hear me? Participants, kindly wait for a few minutes. I think he's going to rejoin, I suppose. Yeah, I think he may have some technical issue in connecting to WebEx, but I haven't locked the meeting, so he can always click the link to get in. So the organizers, please connect with him and sort it out. We couldn't hear you, sir, and let us know. So you can rejoin the meeting, sir, if you're already here and you're experiencing the technical issue. Oh, thank you, Pranav, for sharing the slide, the introductory slide. And I have a few instructions to the participants and viewers, and especially for WebEx participants. Kindly keep your microphones and video in mute or turn off mode. It definitely disturbs the speaker and also the clarity of the presentation. Post your questions in the chat box. For you viewers, post your questions in the comment section. We will collect questions from the YouTube and will ask them for sure. We encourage you to ask more questions. Please type it in the chat box. I don't see a lot of questions in the chat box. Kindly participate. Kindly engage yourself in the session. So if not, I think I think Dr. N. Sandeep Kumar has some technical issue. Maybe we will come back to him a little later. So if everything is okay. Oh, I think he is here. Yes, he's here. So, sir, are you able to connect? Do you still have issues? Can you hear us? No problem, sir. It's okay. I think he's. So participants, please stay for a few more minutes to listen to the talk. in minutes. And in the meantime, we want to make announcements to all the participants of YouTube and WebEx. So we will be sending you chat boxes. So you have to fill them. You can fill them from 4 p.m. onwards today. And until then, we will not be accepting any responses. And I do hear from people like I do see people like repeated and kindly we can definitely track on the time, and then we can definitely mark your presence and issue, and that will help us issuing the e-cert. So kindly subscribe to our YouTube channel. So we will again be pasting the feedback form link and also our YouTube channel link. So this will help us tracking your presence on all 3 days, and those who attended on all 3 days will receive the e-certificate of participation. Uh, we do monitor people from WebEx as well, but the thing is that once we close the meeting, we cannot have track on people who attended, because not all of them like actually message us in the chat box, so we cannot track a lot of people in WebEx. You can definitely click the login link and go to our YouTube channel, and you can basically go to our YouTube channel and click subscribe. That would be better. I think we don't want to like confuse so much. Like we just want you to subscribe to our YouTube channel to mark your presence, and that will help us in issuing the e-certificate. And also please fill in the feedback form links as well. With reference to feedback form link, we will accept responses only from 4 p.m. onwards. So the feedback form link is here. It's quite difficult to track people from who are in WebEx. We do see a lot of participants in WebEx, but again, like we have a lot of speakers and the organizing team compared to the participants as such. Hence, we want you to go to YouTube once again and subscribe to our channel. So we were wondering for the last 2 days how we are going to track people, because people sign in at different time and on different sessions. So we felt this would be the right way and method to mark the participants' presence. So kindly cooperate with us, and this will help us in issuing the certificate of participation. Yeah, the speaker has joined. So, dear Dr. Sandeep, are you here? Are you able to hear us? Yes. Sir, are you able to hear us? Dr. Sandeep, are you able to hear us? So in the meantime, and some people asked me if there are any feedback form for each day. It's not. We have feedback. We have only one feedback form link for all 3 days, and that will be shared today, and that has already been shared in the chat box. And we will accept the responses only at 4 p.m. IST. In addition to that, to mark your presence and to double confirm your presence of participation, kindly subscribe to our YouTube channel. And if you want more updates on the upcoming webinars in the field of biotechnology and bioinformatics, kindly subscribe to our Telegram groups and channel. So that has also been pasted in the chat and comment box. Hope it's clear. I think we've almost answered all your queries with regard to the subscription and also in filling the feedback form link. Kindly check our messages. It's quite difficult to monitor people from WebEx, and the WebEx participants can very well go to the YouTube channel and click in the subscribe button. And definitely all the presenters, be it keynote speakers or the short talks and poster presenters, will definitely be getting a presentation certificate. So, and more details will be announced close to the valedictory function. So kindly stay tuned and kindly be present. So we have two more speakers after Dr. Sandeep Kumar, and please stay in. So, Dr. Sandeep, can you hear us? Dr. Sandeep, can you hear us? I think we were able to see you like a minute ago. So I think there is some issues with his computer. We could see you like like in a fraction of a second, like are you able to hear us? So if you have some technical issues, maybe we can accommodate the next speaker and then we can return back to your talk, if that is okay. So I think there are two Sandeep Kumar as login here, I guess. So if you hear me, can you please unmute yourself and say yes?
Yes, I can hear you.
Okay. And then, sir, what is your... Can you share your content? So can you please share your content?
Are you talking to me?
Yeah.
What content, please?
I mean, am I talking to the right person?
No, you're not. I'm not a presenter.
Oh, okay. So, I'm talking to the presenter. So kindly excuse. Yeah. Kindly mute yourself. So, Dr. Sandeep Kumar, yes, can you hear us?
Yeah, can you hear me, madam?
Yes, yes, I can. I can. Yes.
Okay.
So kindly
Yeah,
share your content.
finally, yes. I think it's a bit of a security issue on my laptop. I was logging in my company laptop, so I do apologize for that. So I had to use my own laptop.
Okay, no problem. So please share your content. The icon is next to the video mode. So are you able to do it or do you have any
Yes.
difficulties? Yeah.
Now it's a bit slow actually on my side.
Okay, I can understand. Yeah.
It's been a long time this laptop, so it's fine.
Okay. So participants, please bear with us for, I think, thank you for your patience again. So, Rohini, can you introduce again? Can you introduce Dr. Sandeep Kumar again to the audience?
Yes, ma'am. Sure. I will. Yes. Good morning, all. I'm Rohini, CSR SRF from Vignan University. I'm here to introduce Dr. N. Sandeep Kumar. Dr. N. Sandeep Kumar, Director, Clinical Development, Genesis Healthcare Private Limited, USA. Dr. Sandeep finished his BTech degree from Baparla Engineering College in the year 2008 and MS from Middlesex University in the year 2007 and PhD from the University of London in the year 2012 and MBA from the University of Grenoble, France in the year 2019 and postdoctoral engineer French Atomic Energy Grenoble in the year 2018. He is more into the clinical research and development. On a final note, he is a fascinating person and we are grateful for his presence among us today. Without further ado, please join me in welcoming Dr. N. Sandeep Kumar. Sir, please.
Oh, yes. And oh my God, there are two. I think, so you can share your content. Yeah. So you can share your content. can share your content now. Can you hear us? Can you hear us? Yes, now you can share your content.
Yes. Able to see my screen?
Yes, yes, sir, yes.
One sec. Can you hear me, ma'am?
Yes, I can. Yes.
Okay. So can I start, ma'am? Are you able to see
Yes,
everything?
yes.
And am I clearly
Yes,
audible?
sir. Yes, yes, you're audible and we can see you.
Okay.
Yes, sir.
Okay. Okay. So thank you very much for your patience and perseverance.
Please share your screen, sir. We didn't see the screen yet.
You haven't seen the screen.
I mean, there is an option called share content next to the video mode.
Yeah, yes. I did click on that, ma'am, actually.
And then you can go to the option called screen, share screen.
Yes.
Yes.
Can you see my screen, ma'am? Now?
It's booting, sir. We will see in a minute.
Yep. I think it's on.
Yes, I think. But we can't see the screen yet. I was able to see while it was starting to share.
Yep. Can you see, ma'am?
Is it a blank screen or do you can open your presentation?
Yes, I did. Indeed.
Yes, now it is seen. Yeah.
You see my. Okay.
Yes.
Finally, we got there.
Yeah.
Okay.
Yeah. Okay. Yes, sir. You may start. Yeah.
Yeah, I can start, right? Okay. Thank you very much. Yeah, I do apologize for this inconvenience, all of you. It's been a long time. I opened my personal laptop, so thank you for the wonderful introduction, by the way. And so for the past 4 months, we've been working on COVID diagnostics, right from the PCR level to the NGS level. So in this presentation, I'm not going to go deep into the subject as I have lost a lot of time because of my laptop switching on and swapping. So the first thing I would like to say on a lighter note is that COVID-19 yes. Yep. So those were the days where we used to go socialize with people, interact, visit so many places. And as soon as this pandemic has hit, the whole world has gone so empty that we are not even able to speak to others with wholehearted and then we had to suspect everybody as if they got coronavirus or something. So this is what we are in now and what this pandemic has done. And literally, in a short sense, the whole COVID-19 has shattered the world. So, ma'am, am I audible, ma'am? Hello?
Yeah, yes, sir. Yes,
Okay,
yes.
Thank you very much. Yeah. So, from a free world, we have come to a position where we have to wear masks all the time as we go out. It's really a punishment for us, and this is what you see all the time. And here I am to present to you what we don't see in the lab to keep everybody safe and sound. So these are the things that you don't see normally in a day-to-day life, and which is playing a greater part of a role in terms of disease in terms of containing the disease as well as preventing it from further progression. So this is a PCR CT graph from one of my runs. So in this presentation, what I would like to present is one part of my work that I'm doing at Genesis Diagnostics since February the 14th. So in this presentation, I would like to present you the molecular diagnostics or molecular diagnosis of COVID-19 and why testing is the key. I know you have so many people talking about we need to test, but why is it so key? Because I don't think nobody has touched on this part. And COVID-19 genome assembly, and this is a major part in terms of designing the diagnostic kit and even the NGS, and what kind of a role this plays, and what kind of discrepancies that we're going to get will be discussed. The factors in designing the COVID-19 diagnosis test, and it's not only pertaining to COVID-19, but it applies to all the diagnostic tests. And we also did, we are not only a diagnostic lab, we are also a hospital as well. So we have a 120-bed hospital, and we have the finest doctors in the world, mainly pulmonologists. So I would like to share a small study that we have conducted out of curiosity to see how this COVID-19 diagnostic kit that we designed is working. So coming to the first thing, which is COVID-19 diagnostics. So detection is the key to contain COVID-19, because the first thing that anybody having a little bit of symptom, it doesn't matter whether it is related to COVID or some other flu, is to go for a diagnostic test. So why is diagnostic test key? Because this gives us an exact position of how the patient is, and in terms of his to design his treatment strategy on anything. So with respect to COVID-19, as of now, there is treatment. It's only passive treatment that's going on. So detection is the key for COVID-19, and detection after getting the symptoms and before getting the symptoms, which makes a lot of difference, because it's as everybody knows that the COVID-19 is a very rapidly progressing and with a high reproducibility rate of 4.5. So early detecting of or early detection of COVID-19 is a very important or a late limiting step in terms of cutting down the further transmission of the virus. So I would like to present this with a small graph actually. So this is what happened to us in India. So if you see this small curve there at the very bottom, which is in a dotted dashed line. So this is where we are at in the month of May or end of April, like that, after the introduction of the lockdown. So, and if you look at the very high bell-shaped curve, which is a spike, these are the number of cases that you will be seeing. This is a classical example in the US, basically. And I'm talking about New York, Pennsylvania, as well as Texas states. So if ever there is no lockdown, this is the number of cases that you're going to see with respect to the spike. And India, being very cautious about it, has introduced the lockdown, but unfortunately due to some, or we have to move our economy going, so we had to take off the lockdown. So we will be reaching this bell curve in very shortly, and this is inevitable until we will find a concrete view. So at this juncture, why is detection important? So detection is very important to make this whole bell-shaped curve a very flat curve like the ones you see on at the bottom. So as soon as you see a person positive for COVID-19, he has to quarantine, because there are a lot of factors that influence it. So which I'll be sharing in my next few slides, which we have done a small study. So quarantining a patient who is positive, despite showing no symptoms, is a key step in preventing or flattening this bell-shaped curve. And this is the aim in terms of and also slowing down the community transmission of COVID-19. So I think this is a slide that you might be seeing a lot of times. I don't want to go deep into this. So COVID-19 is one of the SARS family of virus, which shares all about 85 to 86% of genomic similarity. Despite that, but it has a lot of reproductive capability compared to SARS-CoV-1. It's a 30 kb genome, which is compared to other genomes, it's pretty much small. And the key for designing any diagnostic test lies in selecting which gene or which gene you're targeting. So this is one thing that we need to take home from this whole genomic assembly of COVID-19. So coming to the next part, which is the slight symbolic way of representing the amino acids of spike protein and non-structural proteins on COVID-19 genome. So if you look at the initial kits that were designed by major institutes like CDC, Center for Disease Prevention Control in the US, as well as World Health Organization, they have come up with a very strange genes. Like CDC is only concentrating on nuclear protein genes, as well. If you look at the WHO, they look at RBRP and E gene. So these are the two recommendations by the world famous institutes for containing all the pandemics, and who are doing it very closely in terms of its transmission. So we haven't, as of our company goes, we did not take any of this into consideration. We have designed our own kit and validated it in our own lab and submitted to the FDA for emergency use authorization. And we got the authorization in the month of May 25th. So, and I would like to share some of the details of our validation as well in terms of designing it. So if ever you want to use any kind of a COVID-19 kit to design, or look at the primer designs from CDC, you're free to go to the website of CDC or WHO to find the primer sequences. There are numerous primer sequences, and the numerous sequences from the day one. So what are the main factors in terms of designing the COVID-19 diagnostic test? So the main factors are nothing but the primer design itself. So we took RNA-dependent RNA polymerase as our target gene for designing the primers. So, and what we observed during our experience with COVID-19 diagnostic test is the quality of the swab is a crucial factor in determining or limiting the false positives to false negatives. And this is a key step. And most of the people think that I have given a swab, and if ever the healthcare worker or a nurse, whoever is responsible for taking the swab, is not swabbing well, really impacts the way the results come out from the COVID-19 diagnostic. So it mainly applies to molecular diagnostic tests, especially RT-PCR and NGS, or LAMP test. So coming to the next point, which is the swab collection. So the spot of swab collection is a very important step, because normally people think that, okay, it's the same, it's a systemic infection to the upper and lower respiratory tracts. But it does really make a lot of difference, which I'll be sharing you some of the data that we have done in our lab. So the spot of sample collection makes a lot of difference in terms of getting a positive and a concrete positive and a false positive results. And finally, this is the stuff that we don't get normally used to in our everyday life, which is specificity and sensitivity. So these are the two crucial factors, or two sides of a coin for any diagnostic test. So COVID-19 test has to be designed in such a way that we need to limit the number of false negatives as opposed to false positives. Even though if a person is false positive, it doesn't really make any big changes to his life because he doesn't get any symptoms of that. But if it's a false negative, he would infect so many people down the line. So specificity and sensitivity are a biggest biggest bottlenecks in terms of designing a COVID-19 or any diagnostic test. So we need to kind of go to the rationale where we need to optimize these two parameters such that they're in imbalance. So it's like moving the center of mass of a car when you're turning around a steep curve. And coming to my first so this is the graph that we got from one of my technicians, where we have collected almost 150 swabs from different people, and out of that we selected 41 patients who are very positive. I would say we gave them triple positive. We had an internal rating. So as you could see that the throat swabs and nasal swabs have a completely different way of giving us the results in the early days of onset of the symptoms, which is a very crucial factor. As you could see that there is the black line, which is the throat swab, has in the crucial period, which is called the window period of infection, the viral load seems to be very low, as you can see from the CT values on the y-axis. And the CT values from the nasal swabs correlate with the exact positive disease state of the patient. So it is this reason that we insist on having a nasal swab rather to a throat swab or a oral swab. So this is one of the findings that we kind of did internally to make sure that whatever we are swabbing is the right spot, and which is a key designing factor in terms of the COVID-19 test. So, and one of the factors that I would like to discuss out of all our COVID-19 testing results is that there were high viral loads that were detected as soon as the symptoms were onset. And molecular diagnostic, or maybe the RT-PCR, played a very crucial role in terms of determining positiveness at the very early stage of infection before the onset of symptoms, as you can clearly see here for the 41 patient data that we have. And one interesting thing that we kind of noticed is that some of the patients that we found positive do not have any symptoms, and while some of them had, which correlates with the age or a high risk group, or more than 55 age group. It also depends on the BMI. And what we found was even the asymptomatic patients had a viral load similar to the symptomatic patients, but they do not have any symptoms, but they've been transmitting this virus to so many people since the last 4 or 5 days since their infection. So we are very intrigued by this discovery, actually, in terms of being asymptomatic and transmitting to so many people. And this is the reason why it is so crucial to test the patient or a person who is in contact, maybe a primary contact or a secondary or a tertiary contact. So it's a very crucial factor, because he becomes a super spreader, or he or she becomes a super spreader. So this is the reason why it's very important. So coming to the next factor, which is the specificity and the sensitivity. One of the very important thing when designing a diagnostic test is that we need to understand the cross reaction or analytical specificity of the test. So as coronavirus being a very close relative for coronavirus family, so it's always important to determine whether we are testing the right COVID-19 or not, or SARS-CoV-2. So we need to test a list of compatible, closely related viruses, and which is a key step in terms of designing a diagnostic test. So this has to be borne in mind, and so whenever whatever test that the people design, they have to look at the close relatives, and there are very good machine learning programs to do this online. Some random forest models that you can go for. There's a lot of coding that can be done on this respect to kind of find the right parameter set for COVID-19 test. So if you have any kind of interest on this part of the area, I would definitely help anybody who's interested in terms of designing it. And we have an in-house program, parameter sets. And coming to the last part, which is specificity and sensitivity, while we are designing our kit, is that what we have tested is we have tested some patients where but a very good percentage of positives and with a negligible number of false positives and false negatives basically. But in reality, whatever samples that we did were not on real samples. So the samples that we tested for emergency use authorization were all contrived synthetic samples. So I don't think this really implicates the real-time scenario. So whenever designing a diagnostic test, it's always good to go with the real samples or real-time samples. This is one of the major factors that really influence. And looking at this data, anybody can say that this diagnostic test is the best one. In reality, it's not like that. It's a completely different story, which I'll be sharing with you in my next preceding slides. So, as I told you that I'm not convinced with the way the contrived sample study went through and validation went through. So, what we have done is we have at our disposal Jefferson Hospital, which is our main partner. So, we have done a small study with people of 41. And this is the data that we had from them. So this is one of the that we have taken from the patients through nostril swabs, and we limited ourselves to a cutoff of 25, an average CQ value, and selected 41 subjects for the study. And these out of this 41 patients, we had 55 of them. 8 of them greater than 55 years old, and the rest of them are below 40 years old. Just to give you like a small demographic. The reason why we took 25 is because whatever study that we did were consistent with this value of 25 being more positive or a triple positive on our scale. So this is the determining factor, and also we did some ROC curve analysis to find out this average CQ value. And if you're interested, I can share the data with you afterwards. And this will be published in next coming months. So this is a small diagram that one of my colleagues put forward actually with the 41 patients. So this gives a clear indication of how the whole diagnostic test of ours correlated with the exact matching of the clinical symptoms. So on the 7th day of these 41 patients, these people, they came to our facility at different dates. Hence, we did not give the exact date of admission, and we took this as a random 4-day. And then these people were admitted into our hospital on the 7th day, which is 1 week after the test is done. On the 8th day, we found that 51% of the patient had a shortness in breath. And so some of them had very severe symptoms, respiratory distress syndrome. And 16 of them, out of them, they succumbed to death to COVID-19. And it's 6 of them, they succumbed to death. Actually. So all these 6, they were all above 55, and this doesn't reflect the whole population that all the 55 age people will be affected, but these people are more prone to the disease, and the disease prognosis would be very high in these people. And what we saw in consistent is that fever is the common symptom out of all, and cough, 1 by 4 didn't have any symptom of cough or anything, and most fatigue or myalgia were not achieved by almost half of the population that we studied. So this is a sample study that correlates with whatever diagnostic test that we designed, and this is a very powerful implication where all the diagnostic test has to be correlated clinically, and this is the one piece of puzzle that is missing from most of the laboratories, and we did this.
Hello, sir.
Yes.
Sorry to interrupt. We have another speaker from the US, and it's already 1 a.m. for him. So we have some.
It's my last slide. It's my last slide.
Yeah, yeah. We have few questions as well. Yes, thank you, sir.
Okay. To conclude my talk, actually, I thought of presenting my NGS data. We found a very good, interesting NGS data, but unfortunately, I could not present it right now because I'm out of station. So the first thing that we found was that the testing is a main key because everybody knew it, and choosing the right primers and the chemistry, along with the quality of swabs and the spot of the swab to eliminate false negatives is the key for any diagnostic test. And I would highly recommend to follow up clinically whatever diagnostic test that's been designed or validated in order to have a concrete or a full-scale positive result, because it's going to have a lot of implications in a patient or a human being's life. So I would suggest all the diagnostic tests or labs kind of go for this kind of, and also do make some amendments according to whatever chemistry that you have designed to make sure that this applies, this correlates or in synchrony with the clinical samples. And one more thing that we found is that there are some sequences that are submitted to public databases where we find some kind of inconsistency, but despite that, most of the sequences are defined, and we do see a lot of mutations in the spike protein area because this is the area that we are concentrating on. And I think our data would be very useful for people who are designing vaccines in the future, consistent vaccines or sustainable vaccines as of today. So thank you very much, and I would urge everybody to be safe and stay healthy. Thank you.
Thank you very much, sir. We have a few questions. So the first question is, do you do RNA library prep for COVID-19 genome sequencing?
Genome sequencing, yes. RNA library prep, yes. Yes, we do library preparation. Yes. That's the first step that we do after and also we have an in-house. So normally people in the US, they are using a lot of kingfishers nowadays to make the sample costs spiking up. So we have designed our own methodology to find a right extraction method as well as to find the right way of making these RNA.
And what are all the potential, like the genes that are like upregulated from the genome sequencing that is like predominantly upregulated, like in your RNA-seq, for example?
Okay, so upregulated. I'm sorry, we don't look at the upregulation here, ma'am, per se. What we look at is the sequence change or a mutation, mainly with respect to spike protein. So if ever there are any changes to the submitted sequences, for example, when you go to NCBI, you see the Wuhan submitted sequence. And if this is our reference sequence that we did, and we don't look at the content of RNA actually, because I don't think it would be applying for the cells that are being infected by the virus. So as you can see that the reproducibility of index for COVID-19 is 4.5 almost. Means 4.5 crore viruses will be coming out of one infection, one spot out of epithelial permanent cells. So what we look at is the changes in the sequence, mutations or the dynamic mutations and the strain capacity.
And how long would it take to like submit those sequences and do the cross checking of any sequence changes or mutational changes that you normally see?
Basically, we have to publish this data.
Yeah.
And we want to make sure that whatever we saw is in line with the in silico modeling. So we saw two mutations in spike protein, which we think that makes a lot of difference in terms of affinity to the ACE2 inhibitors. Sorry, ACE2 receptors. So this is what we are modeling right now, ma'am. But this is a very academic project actually. Myself being an academic, I'm more interested in this, but we are also into clinical trials. So that's the reason why we have to conduct this test.
Yes.
NGS is not part of our diagnostic ambits. So this is out of our pure interest, as well as to make sure that whatever we are designing is in line with the clinical setup. And submission wise, as soon as we publish, you'll find our sequence in the public database. I'll send you the link if you want. I'll share it to Dr. Venkatesh.
Yes, sir, would be nice. Yes, thank you.
Yes,
And
and it makes a lot of. Go ahead. Sorry.
Yeah, this is another question. Like, what is a biomarker in the diagnosis of a COVID-19, or the immunochromatography of the COVID-19 contains which biomarker? Which biomarker do you use for your immunochromatography?
Okay, that's a good question, but I'm not into immunochromatography. I'm purely molecular clinical stuff. I think I'm not the right person to answer this question, but yeah, I think it's mostly I did not get what biomarker you're looking for. You mean on the virus side or on the host side?
I think it's no. I'm not very sure about it because this is the question that came from the audience. The question.
The biomarker. No. I'm thinking like biomarker is like the one that is expressed in the blood as soon as the virus is infected, or what kind of like? I did not get what. Are you looking for what kind of protein you detect while you're doing a immunochromatography?
Yeah.
This is what I am stuck at. So.
Okay. So I think that's not something which covers into this session, I guess. And
yeah,
probably
we are not into biomarkers, ma'am. We are into diagnostics and how this whole diagnostic setup is going towards vaccine development, and what kind of strains that are being discovered in real patients. And whatever data that I showed you right now is real data from the patients. And this is out of our own pockets and whatever money that we have invested. And
yeah, purely using genome sequencing for diagnostics.
Yeah, it's a PCR. We have also developed LAMP-based technique as well, madam, which works very well in India basically, because it doesn't need a lot of equipment. Basically, it needs two heat blocks and a nice program to design LAMP primers. And this works very well, but it cannot be multiplexed. So it's called the loop amplified primers. It's a LAMP-based technique, and there's one company in San Francisco. They have already got the EUA approval for marketing it. I think this is the best thing that any academic entity could design.
And that's much better than the RT-PCR.
it's almost equally sensitive, but it's comparatively far less expensive compared to
RT-PCR.
probe-based. Yeah, because ICMR, I was reading through ICMR guidelines, and I was in touch with Bangalore. So I was surprised that they were looking for a probe-based test with 2 gene compulsory on the RT-PCR, RT-qPCR, for example. So that means you have to use 3 probes, because RNase-P would be your internal extraction control. RNase-P being 1 copy for each cell makes a very good internal extraction control. So that is the golden curves that you can see on my PCR curves.
I think it's less expensive and even quicker, because you can see it on your eye basically. Because for RT-PCR, definitely we have to wait for the results, and it takes some time for the preparation as well as to run the PCR as such. So I can understand that. And before closing this session, one last question: does mutation in virus genome greatly affect the reliability of COVID-19 detection using RT-PCR?
Yeah, yes. If your primers fall in that area, and then the mutation is not point, and it's a big one, definitely affect the overall melting temperature and so on and so forth, as you can see.
Definitely, and
it's like a lottery, I would say.
I think it's more important to design primers that's in the less variable region.
Yeah, I really appreciate the move from ICMR basically, because they have made a mandate that 2 genes have to be introduced, and right now we are using 2 genes in our lab, which is RDRP as well as N1 gene, which is both a combination of WHO and CDC. And I would suggest everybody who is into diagnostics would definitely go for a 2 gene diagnostic rather than 1 gene, because it would be giving more false positives, as for my experience is concerned.
And how do you deal with the false negatives, if at all there is?
That's a very good question. That's a very good question. False negatives, yes. Let me show you one slide actually, but as I'm limiting on time, the main problem comes when a patient is falling at a very later cycle, that is 33 to 40 cycle period. This is where I call this as a dangerous window, because if ever you see a patient with an amplification of RFU around 1600s, and still this guy doesn't have any symptoms, which means I would say definitely, I would make him false. I would make him a false positive. I would call him a positive basically, but he's false positive basically. So it's better to have a false positive than to have a false negative, because false negatives are super spreaders. So there is no way that we could contain.
Do asymptomatic patients likely fall under the category?
Asymptomatic, no, ma'am, because the asymptomatic patients do have viral load.
Oh yes, yeah, right. Okay, I've been stupid. Yeah, definitely. When they actually
you're fine. I do understand your question.
Definitely look into the swabs, and when they are doing when they are analyzing at genome level, definitely they will be detected. So there's nothing to do. I can understand there's nothing to do with the asymptomatic stuff, asymptomatic behavior of the patients. I can understand at genome level, you can clearly differentiate, the positive from the negatives.
Yes, absolutely. And
yes, I can understand that.
The asymptomatic patients are so because they see the result as positive, but still they don't take any curative measures. That is one of the biggest hurdles that we are seeing a lot of positives in India. So they think, okay, I'm asymptomatic. This test might be wrong actually, but it's not the case. End of the day, maybe his window period is a bit extended. Maybe on the 10th or 15th day. So this is what we saw from our studies. It's a very interesting study this as COVID has given us, and it's a real boon for molecular diagnostics in terms of
Yes, sir.
NGS as well as introducing the different kinds of PCR into to the market.
Yes, sir.
And
Yes, sir.
if
Thank you.
somebody is interested from your institute towards any diagnostics or anything, and it's the best time. And this is a very good entrepreneur. So I thank you very much for giving me this opportunity, and I
Thank,
congratulate you for really approaching me for this. And I do apologize for the initial hoax that happened.
thank you, sir. Thank you. No problem. And it was a very interesting talk, and it's like diagnostics is something a lot talked about at this point of time. And thank you very much, and thank you for the wonderful session despite the technical delays.
Yeah, I
Thank
do
you, sir.
apologize for not presenting the whole data. Actually, it takes another 1 hour or something. So I made myself very crisp and clear. If somebody has any doubt or clarifications for the information, I'm ready to accept any.
Definitely, sir. You can post your mail ID to the chat box, and people can get back to you later. Thank you very much. And now we move on to the next speaker, Dr. Andrew from the USA. And Dr. Andrew from the Animal Health. I'm sorry, there is some background noise. So our next speaker is Dr. Andrew from the Animal Health and Food Safety Research. He's a scientist from there, and I think it's the company name is Diamond V Company USA, and he's here to talk about the impact of nutrition on balanced immunity, a tool of mitigating against viral infections. So he's an animal health and food safety research scientist from the Diamond V Company. And to be more clear, so I'm repeating it again for this. So now we have Rohini to introduce Dr. Andrew.
I'm Rohini, CSR SRF from VIGNAN'S University. I'm here to introduce Dr. Andrew. Dr. Andrew, Animal Health and Food Safety Research Scientist, Diamond V Company, USA. He has over 6 years of professional experience in animal immunology, health, and nutrition. Most of his span is from research and industry practice. A good portion of his experience has been in the area focused on the interrelationship between animal nutrition and immunology across the globe. His travels across the globe have exposed him to diverse practices of animal health in regions like India, Middle East, and Africa, and the USA. He has leveraged on being a global citizen to understand cultural and ideological diversity. He can learn and unlearn things for the benefit of development. He intends to use the knowledge and skills acquired through his education and career to improve the livelihoods of many people across the globe. He is well skilled in formulating diets using the modern softwares and applications, the use of current technology in animal health. Example, artificial intelligence is part of his DNA. This has enabled him to revolutionize and increase efficiency in the sector. He is able to carry out proper diagnosis and treatment of diseases, hence improving animal health, which results to increased productivity. He has received several awards, notably Conference Awards, Lead Speaker at International Conference on Animal Nutrition India 2019, Research Fellowship ILRI, and University Postgraduate Scholarship, among others. On a final note, he is a positive, energetic, and inspirational person, and we are grateful for his presence among us today. Without further ado, please join me in welcoming Dr. Andrew. Sir, please.
Hello, Dr. Andrew. I think, okay. Thank you. I hope you're able to share your content. Yes, it's perfect. So you can turn on the full screen mode. Yes, sir, can you hear us?
Yeah, I can hear you. Thank you very much.
Thank you, and we sincerely apologize for the delay.
No problem.
Thank you very much.
Yeah, thank you.
Yes, sir.
Yeah, thank you very much from your end, and it's a good opportunity you have given me, and it's also good again you kind of uniting with some of my colleagues whom I might know, and I've had some previous travels in India at Calcutta, West Bengal, towards that state, and New Delhi, and some of the northern states, yeah, including Punjab and others, and also Pune. I've been to some of the areas there. So that is my area of jurisdiction, because you'll understand that I serve India, Middle East, and Africa. So it's a very big geography, and we are based in US. That's where we have our headquarters. So it is a great opportunity and a privilege to present to you. I'll make it very short presentation, and a good conference which has really have been followed from the beginning, and I like what the previous speakers have taken me through. So in my presentation, it will be a bit different. Kindly, somebody can mute the speaker. I'm hearing
Mute
a lot of
yourself.
noise. Kindly mute
Okay.
the speaker.
Yes, yes.
Yeah.
Yeah.
Thank you. So what I was saying is that through my presentation, I'll just be able to get you through some of the things which were not really captured by our previous speakers. And it is good that I've been following the conference, the speakers, and the presentation all the 3 days, including the third and the last day. So I was able to filter out some of the information which I didn't want to be repetitive. I didn't want to repeat what my colleague speakers in the previous sessions have really gone through. But very quickly, I'll take you through a brief introduction. I just give you a glimpse of what we do, what are some of our facilities, including our Research and Innovation Center. Then I'll take you through some of the basics and a bit deeper science on the nutritional immunology. Then I just summarize and give a space for question and answer. And that's, I think it will be much more interactive for us. So if I look at the Research and Innovation Center for Diamond V, as you clearly stated it, that is the company I work for. That is the corporate campus we have. We are situated in the state of Iowa. It is a Midwest state. It's one of the agricultural states in the Midwest, US. And the city which you find our company is called Cedar Rapids in the state of Iowa. So we have so many facilities across the world. And our headquarters, despite being in the US, we have some offices in Latin America, in Brazil. We also have an office in Mexico, and we still serve the Europe with the office in Netherlands. And for India, which is the region which I do serve, we have an office which is located in Kenya, serving India, Middle East, and the whole of the Africa. Then in the Asia Pacific, talking of the Asia and most of the Oceania, including the Australia and New Zealand, we also serve all those regions. And then we have another one in China up there. So we have the global presence worldwide. What do we do with our services, which we collaborate with different institutions, including universities, just to do a lot of research on our products, and we also give services towards that endeavor. So this is our Research and Innovation Center. It is based still in the headquarters. These are two divisions. We have two divisions in our organization. We have the human-based division for human health, and then we have animal health division. So the animal health division is pretty order. I've for now, we have been in existence for the last 77 years also, yeah, being established in the year 1943. Then that gave rise to the human health division, which we call Embryo Sciences. I'll be able to share the link after towards the end of this presentation. So what have we learned through the tag of all those years, and what have we been doing in terms of the science and research? We have been earned as the trusted experts in nutrition and health, because as you'll see in my preceding slides in terms of the nutritional immunology, which is an aspect I want just to talk here and there, is that we leverage on our expertise, having more than 150 PhDs in our organization, and DVMs and MDs and MScs. We leverage on that pool of skills, which I believe most of organization, the profit-making organization, some of them they do not have that kind of expertise. Then we collaborate a lot with various universities, the state universities like the IOM. We go to Kansas and some of those surrounding state universities around. So we do a lot of, not limited only to the US, but in the other parts where we have our global presence. I was in India last year, and actually I got so many professors in one of the conference I was, and it was good. Actually, we started a chit chat on how we can collaborate in some of the areas of the research. So it is a great opportunity that LLB is giving me just to take you through some of what we have discovered, what we have been doing, and maybe explore some of the partnership going into the future, despite now this pandemic. Well, how did we have two divisions? We have research on the animal, and we have research on the human health. So the scientists at our organization, Diamond V, started looking at animal health and immunology. That was the start. That was the beginning. Back in the year 1943, during the World War II. Employees now within the production manufacturing facility, which I've shown in one of in the previous slide, is that the employee benefits were kind of changed to accommodate healthy plant workers. So here it's a story whereby I'll just need to show in terms of some of discovery, which was it was done accidentally, but it came now to give rise to the human health division in terms of the research. So the company experienced unusually low health insurance premium. So what happened is that we had the animal health division, which we do manufacture products which support immune and health balance. Yeah. So it was realized that these workers who were actually based in the manufacturing facility, they were inhaling, these products through through all the manufacturing process. So it came that they tended to claim less sick days in terms of the leave. So that now prompted the scientists in our company to actually look into that and collaborate with the human doctors in order to elucidate what could be this and maybe try to discover what is happening. But that was just based on what we had seen, the benefits of the products we manufacture in terms of the animal health. So it's like we could correlate and maybe give it a benefit of doubt. So we hypothesized that could this be an effect of boosted, balanced immunity, which is happening to these employees. So that was a case which has had to just follow the scientific protocols by taking some of the saliva samples from the employees who are involved in the manufacturing facility. I'll show that. I have that research. It was published, and I'll be able to project it on your screen. So it was hypothesized that the plant workers were healthier due to long-term exposure to the animal products. So what could that be correlated with what these humans were kind of benefiting? So what happened is I've said that some saliva samples were taken from the employees within the facility. So here included the human blood and saliva study, whereby the age, the gender, and the tenor comparison between the workers exposed or not exposed to the fermentation product, just to have that experimental group and the control. So the results from the study actually suggested that the plant workers had healthier immune system. Okay, what does that mean? As you can see on my footer is that the natural killer cell activity, which now I'll provide diagrammatically the mechanism into which this was discovered, and the salivary IgA, which is immunoglobulin A levels were higher in the plant workers than the control group. So here is just the graphical illustration that this is the treatment in two groups which were involved. So we had control and we had the treatment in which our product was used. And what happened is that in terms of the results, as you can see, the percentage of immunoglobulins, and this is immunoglobulin A. And for just the benefit of the participants of this webinar is that, as you all know, there are different types of immunoglobulins, especially this particular immunoglobulin A. It is more associated with the mucosa. We still have the high abundant immunoglobulin G, which now you find around 70 to 75% of the total immunoglobulins within the in humans. Then we have immunoglobulin E, we have D, we have M, maybe as trace immunoglobulins. So of interest here was the salivary immunoglobulin A. So the percentage here, and actually as I show that the results here, they were actually statistically significant, and the difference was very significant between the two treatments. The treatment with the group which used the products of Diamond V versus the control where they did not get any exposure to the product. But again, all that background, I wanted just to lay about this topic, which is really related to my title. So what is immune balance? That is just a hypothetical question which I just wanted us to maneuver, only for me leading the discussion from my hand. But again, you'll find that the immune system's optimal state, as the definition, which I just give for the purposes of really understanding this subject matter, the immune balance is the immune system's optimal state, yeah, where the body best reacts to signals to either boost, yeah, the immune performance when needed, or to suppress. So the keywords here are boost or suppress. And the suppress an inappropriate reaction, so or to suppress inappropriate reaction to substances that could be naturally harmless. So the balance is actually what I wanted. I didn't really want to get into the nitty-gritty of boosting immunity, because we know as clinicians that boosting immunity for either the human or the animal, I'll be talking much of the two species, because we do these comparison studies most often, and working alongside that line, I would be just justified to look at and quote my studies based on both the human and the animal, because I've got that two aspects of comparison. So here I wanted to just stress on, it's either boosting, but you boost when now the performance, if there is a need of doing so, but you also suppress if there is inappropriate and it is maybe unnecessary. Now we need that. So that suppression and boosting now brings into the issue of the optimal state, and that is what I'm calling the immune balance. Okay, going forward with the immune balance and to what research has been done in the humans. So as for now, because the human division was established later on for Diamond V after the animal, because the animal health division has been in existence for the last, I think, more than the 77 years now. But now, out of the animal benefits in terms of the immunology, which now this nutritional supplement, if I could call it, was given out and was showing the benefits, then it came that this study actually the scientists and just to get, and with collaboration with the third-party researchers and also the research institutions, including the universities, they had to get into this, doing the studies, doing publishing. So these are some of the 13 peer-reviewed published scientific articles, and these are just particularly which were done on the human products, which we call it Epicor. That's a trade name, but for the just reference purpose, I'll provide a link for you just to look at some of those studies in details. So this was just to look at what would be the effect of this product on the colds. These are human colds and flus, and also allergies. But for the purposes of this presentation and conference, I would just like to pick some of the viral infections so that now we can really live up to the theme of this, because I would like to relate to what is happening currently in terms of the COVID-19, so that now we see, are there opportunities? Are there some gaps we need to fill in order maybe to catch up with the nutritional immunology as a tool just to mitigate against the viral infection? So those are some of the studies. We have one still in the press, be getting 14 or more studies in that. They are less in human because we established this human health division later on. But on the animal side, we have done a lot of studies. We have published more than 150 peer-reviewed scientific articles in reputable journals. So humans, we have done a bit what we could do just because of the time span in which we established the human health sciences division. So we have done a lot in terms of research. And as we in Diamond V, we believe that the key collaboration being industry actors and academia, there must be that synergy, and that is synergy we really like to bring in, so that now we really kind of solve societal and community problems through our research-based evidence and the services which we give and the products we give to our clients. I would like to note one thing, one of the very good speakers mentioned about, and he was giving this data, I think, from the drug bank. Yeah, he was looking at the numbers in terms of how the drug bank has categorized some of the products here in the market. And I think he was elucidating much more on the drugs in the market, yeah, and the drug discovery process and all that. So he came to mention something about nutraceuticals. So talking of this subject matter, because the way we approach COVID challenge, and not only COVID challenge, but the viral infection, it has to be holistic. Which opportunities? Do you have opportunities, the vaccination? Yes, that's good for the scientists to explore. Do you have the opportunities in the drugs discovery? Yes, that's another opportunity to explore. But also related to that, there is now another solution which can come up in terms of nutraceuticals. This is just the use of nutritional products to just alleviate and mitigate against the viral infections. So that is what now I'm bringing on the ideology of nutritional immunology. So looking forward to that particular study which was done in the humans, and some of the results which were actually seen, is that the group of humans, the subject who were treated were subjected to the treatment with this Diamond V Epical product, which is immune balance product. I'm not saying it is immune booster, because that is not what it does. It is immune balance such that it works, the metabolites within the product, they work naturally with the biology of the animal, boosting when needed, suppressing when needed. Because we know a lot of inflammation, which can be as a result of immune reaction, when it is excess, it is lethal. So it needs to be kept at a good level for that matter. So two of the immunoglobulins here, salivary immunoglobulin A, and we have the serum immunoglobulin E. So this just shows what really happens with the product here in yellow, if you can clearly see on the slide on your screens. Then we looked at the results in terms of the normalized salivary immunoglobulin A, and you can see it's being positive and it is going up in terms of this treatment, which involved Epicor, and that is compared with the control group. The same happens here with immunoglobulin E, and we looked at in terms of the duration in weeks. So this slide is just very summative in terms of the good beneficial benefits we can really leverage on in terms of the nutraceuticals, the nutritional solutions which are geared towards solving and mitigating against the issue of immunology. So again, I would just want to capture this, still related with the animal, with the human research, before I give very few animal studies based on there. So this is 2007. It was a cold and flu clinical trial. And these were two trials, and I'll just explain. And it is for the purposes of documentation and maybe reference. It was done by this group of researchers, one led by Mayad in the year 2008, and that was done, the publication was it is in the Journal of Urological Nursing. You get that publication. And still 2 years later, the same group of researchers led by Mayad, they had now to just back up what they found in 2008 in terms of the nutritional benefits towards immunity and immunological balance for that matter. So that now they look at what could be the effect in terms of alleviating some of the, we know cold in humans and flu, sometimes it can be compounded by other infections, but mostly we talk of the rhinovirus, the influenza, and these are some of the viral kind of examples these researchers wanted to elucidate and know whether we could get beneficial or some benefits towards the human health, and bringing a lot of benefits and mitigation aspects towards that matter. So these are just the nitty-gritty in terms of how the methodology, the design of the study, and the total number of subjects which were involved in that particular study. And I would just go ahead to present. So these results, as you can see in this particular slide on your screen, it is the results of that particular, of these particular two studies. So it is summative again. So the results were very positive. And here, the researchers really wanted to show what is the total number of days that the subjects took when they had these infections, the viral infections. So what was their findings is that 26%, there was 26% reduction in the total days of the symptoms with the cold and flu. So this, it is the control. As you see, it was done as placebo, such that the subject were also exposed to in similar manner, but without now this product. So all that was followed using the proper guidelines and the scientific protocols which are laid down for that matter. So again, very good benefits, leveraging on the nutraceuticals and the immune products, which are again an angle which maybe is a challenge for us, just based on what kind of scenario we are having, just to explore, other than now getting into vaccinology, which is very okay, and also getting the drug, which is very okay. So this is another platform which really we scientists and researchers need also to collaborate and explore whether there are something we can do. So quickly, how does that technology really work? And when I talk of that technology, it is the technology in Diamond V products, both at the human division and also at the animal health division. What we realize and what it is actually it's a group of bioactive compounds, yeah? And these bioactive compounds, which I just really mentioned there, there before in one of the slides, is that they work naturally with the biology of the animal. And the animal here, I'm talking either the human or even the production animals, which we do a lot of research using the models also towards just realizing the general benefits, both for the human and across some other species. So I like this analogy. This analogy, which appears on your screen, immunity, I call it to be kind of taking the security principle, guarding. And the security principle, it can be multifaceted. You find, like for this house, this owner of the house, there is a protective barrier. As you can see, there is also the surveillance. There are some guard dogs, as you can see there, which sometimes the moment now they bark, it's like they cascade the signals, and then we can get activation on non-specific protective mechanism. So what happens next? If now the primary immunity fails, what happens is now that homestead becomes now, it's like we need that backup, which always maybe it is a state police department if anything happens, and then we need also at the same time maybe to keep memory of that. So this backup, which actually comes as the last option after everything has failed in here, it is what is in now the biological term as the acquired immunity, because we have the innate immunity, we have the acquired immunity, and I'll get to the nitty-gritty of that in one of the slides I'll show preceding. And this was just analogy just to credit that by the researcher Badlow in the year 2016. It made it very simple for people even to understand the immunology, the nutritional immunology principle even much more. So what you can see in the slides here, what you can see in that slide there is the barrier function. And the barrier function, one of the speakers, I think in the first day, mentioned something about also the issue of gut health. And before we even go to the antibodies, and I believe now that is where we need to ask ourselves, how do these technologies, the nutritional immunology technology, really kind of synergize and help the vaccines in different scenarios in terms of increasing the titers of the antibodies. So that has been there. So it's like we are talking, there is no one-way approach, certainly, maybe for some of the pandemics, like now what we have is the COVID-19. Sometimes it might be discovered that the vaccine, which is very good, the scientists are doing a very good job every day and night. The drug discovery also process, and also the need of the nutraceuticals, like now we are talking in this particular presentation. Some of these three mechanisms can be deployed just to complement each other. Yeah, like I have seen in some studies we have done in our data, in our research data for Diamond V, is that some of the diseases, and when we used, we applied this technology, nutritional immunology, that it was able, even after vaccination for particular set group of animals, it was discovered when we took the samples, it was discovered that the use of the nutraceuticals and this nutritional technology, it boosted the antibody titer. So it means that there is actually a way in which we can explore collaborative kind of mitigation between vaccines, because as we know, we are scientists, that vaccines, they are not 100% effective. Yeah, within population, we know there are those going to get it, there are those who are going to benefit, others will not. So it's a matter of balance in which technologies we do use, and we all require this technology for just that very purposive mitigation against these pandemics, because COVID-19 maybe will not be the first and the last pandemic which is eating the world. Maybe, God forbid, that we might encounter more. So it is for us, we scientists and researchers, that we prepare enough to know how do we collaborate, that we are not working in a silo, in isolation, such that the people working on the vaccines, the people working on the drugs, the people working on alternatives, including the nutraceuticals. And I also understand India does a lot of fight also. It could be good also we bring the minds together. And I'm happy this conference has provided such an avenue. So this is just an illustration of the barrier function, in which it could be the first mechanism just to handle any pathogen which is coming into the body of the animal. Then this is now the classical kind of classification, if I could say, of between the natural immunity and the acquired immunity. And when we talk of the viral infection, remember, just for the purposes of the record, is that it does more become effective when now we talk of this acquired immunity. And that is where the technologies of vaccinology come in. So vaccines, they do very effective in terms of provision of the antibodies, and that could be now effective in mitigating that. But what I'm saying is this: the nutritional technologies are actually coming to support this, because you'll find that, like I've said, that some of our studies, they have been shown to in some viral infection to actually boost the antibody titer, even when the vaccination was done in that particular animal. This is again a very good illustration. I usually like it, just to credit that to this group of researchers in the year 2015, led by Abbas, and that was done in the cellular and molecular immunology. And you can just get it there from 8th edition. So another good analogy and illustration, you get a microbe. Sometimes the innate immunity, including the barriers and all that, will come in. The phagocytes, the dendritic cells, the NK, which I call the natural killer cells, interleukins, the complement, and all that. But sometimes, before we even get to adaptive immunity, if we could have killed this microorganism at this initial stage before proceeding this, it's like we are saving much of that energy which the humans could need for maybe maintenance and all that. When it comes to the animals, I deal with animals a lot. I could save that energy just for maintenance, and even more for production. So that is the story behind this. So how do this kind of, suppose now the microorganism sets in the body, how does this response come in? Like I said, innate immunity is the first line of defense. Yeah. So you can see against the time in hours, within the first few hours. So again, that will come in and usher in adaptive immunity. So this is now when we are stimulating immunity at this stage. And this is a process which spends a lot of energy just to have this adaptive immunity in place. So if we are having any nutritional technology, like I started by saying, that it boosts and it complements the antibody titers, which are provided by either the vaccine, it could be actually very, very good for most of the species in terms of the immunology and the whole system of immunity. So we have this tag as a company, Immune Strength for Life. And what we have done over the years, a lot of studies since in 1943, before any products channeled to the market, before any services got to our clients, they are always research-based, and that has been part of our DNA. And we like it that way because actually that has just gave us a very good feedback from the clients, the customers whom we work with around the world. So the M1V research demonstrates the effects of our technology on the immune system. You'll find there is increased surveillance. I just gave that analogy. It is not very scientific analogy, but just for layman to really understand, you need that first kind of response. Then you can get a more robust protection. And that is when we come to the nitty-gritty of the antibodies, the vaccine science, the vaccinology and all that. And that is when you create what I call a bigger wall. And that is a robust protection barrier. This is not COVID-19. I know most of the speakers, the previous speakers and presenters have delved much in terms of the COVID-19, how the structure is. We know the COVID-19, this is not COVID-19 as I'm projecting there. We could expect some crowns and all that, and the spikes and all that. But again, I wanted just to pick it from another angle. Talking of the respiratory viruses, and I took a lot of research base which we have done in the animals, and this could be also a good model, which sometimes we extrapolate towards the human health sciences, which is still a division within our company. So these publications, I'll really be willing to share with you for the participants maybe to get the nitty-gritty and what were the positive results in terms of how did our Diamond V technology really get in to mitigate in various situations of different respiratory viral infections. And this, to be just clear, these were done mostly in the poultry, and we animal scientists, we classify that group of animals to be monogastric. So these products really get, because they have single stomach, just like the humans. So most of the models, we extrapolate some of the studies, but we have now to validate in real human clinical trials when we want to get to that side of the humans. So this is just to show the titer levels post-infection and the virus shedding. You know, virus shedding, it can, that is actually that is a precursor to the spreading, because the more you are shedding this virus, the more it is now going into the population. So how do? You need that technology, whether vaccine, whether drug, whether the nutritional technology, which is going out to really kind of reduce this into surmountable levels. And this is just a study which was done by a group of researchers in the year 2015 led by Lincoln. And this is one of the Diamond V technology in blue against now the control. So the two group of animals, you can see the virus shedding and what our technology really did in terms of the amount of the virus which were being shed into the environment. And you can see the difference between the two. The means they were statistically different, and that was measured at P005, and that really gave some of those good benefits we really need to elucidate in terms of trials and research before we bring the technology to life, either to be applied in humans or even to be applied in animals. There is an issue of gene expression. Thank that my previous speakers, they had talked much about the gene expression. I don't want to dwell much on this. I would rather maybe interact with the question and the answer segment. So this is more borrowed from, it was a trial done in broilers, more to the animals. But the same principle really applies. Upregulation of some genes, yeah, and that is one of the things which our technology was found to be beneficial and should do.
Dr. Andrew?
Yes.
We have another speaker on board, so can you please finish a little bit soon? We have some questions as well to ask you, so we need a little bit of time. Yeah.
As I conclude, thank you. As I conclude, this just a continuation of the respiratory virus. So what we can get here is that the immune strength for life, which is provided here, it gives all these benefits: increased surveillance, more robust protection, reduced clinical sign, which makes them a bit less severe. Then of importance is faster recovery of the humans and the animals in which our technology is applied. So those are some of the references. I can readily make them available. Then I would like to acknowledge the conference organizers and LLB for the invitation, which I got. It is really a very great opportunity to interact with the fellow speakers and the scientists, and also the presenters in general for this particular conference, and also the audience for being patient enough. Most of you, I believe, from the first day have been following all these proceedings. So thank you very much. It is the time. I welcome any questions and answers, and I'll be able to respond to them. That is my email. Just get in touch in terms of the conversation. We can continue the conversation just because of time. If any questions are needed to be followed up, and I'll be very, very much happy to do it. Thank you very much.
Thank you very much, Andrew. And we've also been seeing you from the very first day. Like, you've been very active in attending all the talks. And there's one question from the participant. And what is that? How is saliva and blood of the human useful in evaluation?
Okay. So, very quick one on that. I gave some examples of where the immunoglobulins are really distributed. We have different types of immunoglobulins which form part of antibodies, and immunoglobulin A, as you could see in that slide I presented there, it is the one which is associated with the mucosa. So the saliva, it is one of the medium in which we get that antibody. So getting saliva, it was very important. Getting the blood, it is where we have a lot of immunoglobulin G, IgG. So IgG, it makes 70 to 75% of the total antibody immunoglobulins types in humans. So it was very beneficial for the samples to be taken from blood and saliva.
Thank you, Andrew. And I think people will definitely. I think they have no questions anymore, and we'll get back to you again at some point of time in any workshop or a conference. I think the university will also be in constant touch with you. So thank you for the wonderful session, wonderful talk. Yeah.
Thank you, ma'am.
Thank you. And now we have the last speaker of the morning session, Dr. Venkateshwarulu from Swansea University. So I'm here to give a few details or brief about his profile. So Professor Venkateshwarulu was born at Chilakaluri Pet in Guntur district, India. So he obtained his bachelor's in biology and chemistry from Acharya Nagarjuna University, and MSc in biochemistry from the University of Hyderabad, MTech in biotechnology from IIT Kharagpur, and PhD from the University of Sheffield, where he worked on antifungal drugs. So Professor Venkateshwaralu conducted his research work initially at the University of Bristol and recently moved to the Swansea University, UK, where he is presently the Professor of Molecular Cell Biology and Pharmacology. So he made several original and novel discoveries, especially in understanding phosphoinositide, that's the PI, and ADP ribosylation factor ARF small GTPase cell signaling in cancer, cardiovascular neuroscience, and reproductive biology as well. So he has been studying individual and interplay between these two cell signaling events. I mean, the PI and ARF small GTPase cell signaling with respect to cancer, and the other signal transduction pathways downstream of cell surface receptors using multidisciplinary approach. He was a recipient of a prestigious BBSRC David Phillips Research Fellowship and a Commonwealth PhD Fellowship, studentship. So based on his outstanding published work, the Professor Venkateshwaralu has been elected as a fellow of RSC, Royal Society of Biology, Chemistry, and a few others from India as well, and has received an ABAP Senior Scientist Award from India, and and a senior doctorate from Swansea University. So here we have here introduced about him, and I welcome Professor Venkateshwaralu sir to deliver his talk. So, sir, are you visible?
Yeah.
Yes. Yeah. Now you may share content.
Okay.
So you have an icon next to the video mode, right?
Yeah.
And and then just click on that, and then there is an option called share screen.
Yes, I'm doing that.
Yes. Yeah. Yes. Yes, sir. You may proceed. Yeah.
Yeah. Okay. Can you hear me?
Yes.
Okay. Thank you very much for the nice introduction and also giving an opportunity to present our work in this conference. What I'm going to talk mainly about the work we have been doing for a last decade or so, and how that work can be used in developing a novel antiviral treatments for COVID-19. So kind of last few years, we've been working mainly on 3 different diseases. One is type 2 diabetes, second one is cancer, and third one is sepsis, which is based on the bacterial sepsis. So first I'll start by discussing with one of the drug target for type 2 diabetes called glucagon-like peptide 1 receptor, and most of the work is done by one of my PhD student a few years ago. Her name is Dr. Aisha Thompson, and in which I'm going to talk mainly about the importance of the structural requirement, like the N-terminus in trafficking of the GLP-1 receptor to the plasma membrane, and GLP-1 receptor internalization, the molecular mechanism involved in the internalization, and the importance of the C-terminus of the GLP-1 receptor activation and trafficking. And finally, small molecule agonist of GLP-1 receptor. The current drugs mainly useful are peptide based, which has to be taken in injection form, and it can be very inconvenient as well as very expensive. Therefore, there is a great need for developing orally active small molecules, and some of them are in clinical trials, which we've been working for a while to understand their mechanism. The second part of my talk would be on lytic peptide targeting of breast cancer. This work is mainly done by Riaz Janu when he was a PhD student in our lab. and also he continued on targeting in pancreatic cancer when he was a postdoc in our lab. And currently, one of my PhD students, Yueshi Gyo, she's working on therapeutic targeting of sepsis mainly for multi-drug resistant bacteria. So I'll talk about the work we've been doing and how it can be related to developing antiviral drugs for the COVID-19. So let's start with type 2 diabetes. One of the target I mentioned is GLP-1, which is a glucagon-like peptide 1 receptor, and the agonist for that is a GLP-1, which is mainly secreted by L cells in the intestine. It has a wide range of physiological functions, but I'm going to concentrate today on its role in glucose-dependent insulin secretion because of its relevance to the type 2 diabetes. Most of the conventional drugs like metformin works glucose independent manner, and therefore we have to measure blood glucose levels before taking medication. Whereas GLP-1, in other hand, because it works in a glucose dependent manner, you don't need to, you can take a fixed dose on a regular basis and able to treat type 2 diabetes. And therefore it became a very interesting treatment for type 2 diabetes, mainly in the West. It's not become that popular in other countries such as India, mainly due to its it's very expensive, because currently I think it costs about $200 to $300 per month, whereas conventional treatments such as metformin and all are very less expensive comparatively. So if you give a brief introduction for glucagon-like peptide therapies for type 2 diabetes, endogenous GLP-1 has an half-life of about 1.5 minutes. As soon as it gets produced in L cells of the intestine, and it gets broken down by a peptidase called dipeptidyl peptidase, which breaks at this point and makes it an inactive form, because glucagon-like peptide exists in two isoforms. Both are shown here, and often broken down, there is a 2 amino acids, and then it becomes an antagonist. So in order to overcome that, many companies are actually developing dipeptidyl peptidase resistant glucagon-like peptide 1 mimetics. For example, liraglutide, which is similar to endogenous one, just that it has been modified in such a way that it becomes resistant, and also and thereby its half-life has been kind of increased to 12 hours. And also when you connect two liraglutides to a Fc region of IgG4, the half-life can be increased to 96 hours. That means you need to take only once in a week the treatment, which is called dulaglutide. And another homolog called exenatide, which is originally isolated from the glia monster lizard saliva, and which is naturally resistant to dipeptidyl peptidase 4, and it has an half-life of about 4 hours. But the half-life can be increased by formulated in such a way that so that it can also be taken once in a week. As you probably most of you are aware, type 2 diabetes it is prevalent most of the world, and currently 500 million people, which is equivalent about 50 crore in Indian number, are required medication one way or the other, and therefore there are many companies working on different forms of in developing either monoagonist glucagon-like peptide or a multi-agonist for treatment purpose. So glucagon-like peptide targets the receptor called glucagon-like peptide 1 receptor, which is a cell surface receptor, and obviously it's an important target for type 2 diabetes. It is a G protein coupled receptor, mainly functions at the cell membrane. Upon agonist binding, such as GLP-1 receptor couples to GS pathway to increase cyclic AMP production. It also couples to GQ pathway to increase intracellular calcium accumulation as well as ACT phosphorylation. It also internalizes to dampen the biological response or to resensitize the receptor through the recycling. Therefore, we have been looking into various structural requirements of the receptor in cell surface expression of GLP-1 receptor. For instance, here I'm going to give an example of the importance of N-terminus requirement for the GLP-1 receptor cell surface expression. So we first looked into the, it has a signal peptide that has to be cleaved off for the receptor target to the membrane. So our objective was to design mutants in such a way that to explain importance of signal peptide in cell surface expression of the receptor. To do that, what we have done is we created three different mutants. So the first one is where we deleted signal peptide. If cleavage required, probably it won't be affected by deletion of the signal peptide. And the second one is where we replaced signal peptide with viral signal peptide so that it won't be cleaved off from the mutated version. Or we also mutated a particular amino acid so that the signal peptide no longer get cleaved off from the receptor. And then we expressed in cells to study whether the signal peptide alteration has any effect on the maturation of the receptor. As you can see, this is the mature form of the receptor. So when we replaced signal peptide with viral signal peptide or mutated, it's no longer form a mature form. Here we have done immunoblotting using two different antibodies, anti-VSVG or GFP, where it's been tagged with two epitope tags. Then we went and looked into the cell surface expression because it's not been formed mature by either replacing signal peptide with viral signal peptide or mutating so that it prevents cleavage. We saw no cell surface expression of the receptor. Because it's not expressed their cell surface, we don't see any activity, which read here based on the cyclic AMP response. We also used immunofluorescence-based assay to measure cell surface expression. The red color indicates the antibody, anti-VSVG antibody binding outside the cell without permeabilizing. So red color indicates cell surface expression. No red color means there's no cell surface expression. As you can see, in wild type and signal peptide deleted construct showed cell surface expression, whereas the other two mutants failed to show cell surface expression, indicating that signal peptide is an important requirement for cell surface expression of the receptor. So we also looked into the importance of other region, which we call hydrophobic region after signal peptide requirement for the cell surface expression of the receptor. In this case, we have made a series of deletion at the N-terminus and looked the effect on maturation of the receptor, cell surface expression, the activity, as well as again receptor visualization by immunofluorescence. When we deleted 35 amino acids or 40 amino acids, we saw loss of the maturation and thereby loss of the cell surface expression and also and also the activity and also the cell surface by immunofluorescence, indicating that the region between 31 to 40 amino acids also play an important role in targeting the receptor to the plasma membrane. Like many other receptors, GLP-1 receptor also undergo N-glycosylation, which is a sort of type of a glycosylation, which plays an important role in targeting many receptors or a secretion of the proteins in eukaryotic cells. So what we have done is we try to study the importance of this glycosylation in GLP-1 receptor targeting to the membrane. So we treated with cells, when we treat with tunicamycin, which inhibits glycosylation, we can see there is a shift in the mobility of the receptor in Western blotting, indicating the non-glycosylated form move faster than the glycosylated form. Whereas when you delete whole N-terminus, that's 140 amino acids, 45 amino acids, then we don't see any effect because it's already, there is no N-glycosylation, and therefore tunicamycin has no effect on mobility of the deleted construct. Or similarly, when we mutated 3 amino acids, which we know are required for glycosylation, so then we don't see any effect of tunicamycin treatment on the mobility of the receptor, indicating that N-glycosylation does play a role in maturation of the receptor. Again, we confirm through enzyme treatments, which is showing that it is a N-glycosylation is sensitive to PNGAF. There are different types of N-glycosidases. So, and also we looked important for cell surface expression when we treat the receptor with tunicamycin to stop the N-glycosylation or delete the regions required for glycosylation or mutate the sites required for glycosylation. We don't see the expression of the receptor at the cell surface. Because it's not expressed, these mutants also fail to induce cyclic AMP response. We have done several other experiments, but due to time constraints, I'm not going to details. But based on the work we have done, we have proposed a model for the trafficking of the GLP-1 receptor to the plasma membrane. When it gets synthesized inside the endoplasmic reticulum, the signal peptide masks N-glycosylation sites of the receptor. When this signal peptide gets cleaved off, that result in exposure of the N-glycosylation sites, which result in glycosylation, and the N-glycosylation along with the hydrophobic region after signal peptide and some other amino acids, conserved amino acids, play an important role in processing of the receptor in the endoplasmic reticulum as well as in the translocation to the Golgi. So we also looked at the importance of internalization of the receptor in biological response or resensitize the receptor to the recycling. In fact, the GLP-1 receptor internalization is an important factor for insulin secretion. Therefore, the study we have done plays a crucial role in understanding how GLP-1 receptor can alter or increase or change the insulin secretion in diabetic patients. So we have done this using a series of reactions experiments. In this particular experiment, what we have done is, as you may remember, GLP-1 receptor couples to GS as well as GQ pathways. Therefore, we used the inhibitors and activators of these two pathways and studied the effect on the receptor internalization. Just to summarize, only the inhibition of the GQ pathway affected the internalization, indicating the GQ pathway is important for the internalization of the receptor. So here we have done a cell surface analysis. As you can see, we saw less loss in presence of the GQ mutant. Similarly, in immunofluorescence, see, internalization means this is cell surface expression. When cells are treated with the GLP-1 agonist, you can see the accumulation of the receptor intracellular in the form of a dotty appearance or punctate formation, and that is affected only by having this GS mutant, indicating that the GS pathway is important for the internalization of the receptor. Because GQ pathway is a very classical one, which we already know how it leads to ACT phosphorylation. So we kind of designed the pathway, targeted that with the different inhibitors, and studied the effect of the inhibitors that target GQ pathway on internalization of the receptor. In this particular case, for example, the binding of GLP-1 to the GLP-1 receptor result in the activation of GQ pathway, which result in activation of the phospholipase C, which then hydrolyze PIP2. It's a phosphoinositides into IP3 inositol trisphosphate 145 and diacylglycerol. IP3 then binds to IP3 receptor on endoplasmic reticulum, which result in release of calcium from ER into the cytoplasm. That result in activation of PKC, and which through RAF cause phosphorylation of ERK, which controls internalization of the GLP-1 receptor. So we hypothesized this and treated with various inhibitors that target this pathway and studied their effect on receptor internalization. As you can see here, the receptor U73122, which is inhibitor for phospholipase C, which inhibited cell surface expression in a dose-dependent manner, which studied here by two different assays. One is ELISA based, and the other one is immunofluorescence. As you can, the inhibition means there is no punctate appearance of the receptor inside here, which shown here by immunofluorescence. So I'm not going to go quickly on the second one is the PIP2 inhibitor. PBP10 is the PIP2 binding protein. When you add in it also inhibited the receptor internalization in a dose-dependent manner. It does so by binding or mopping up the endogenous PIP2 levels and thereby prevent the availability of PIP2 for phospholipase C action. We also used another inhibitor, which is the inhibitor for IP3 receptor, that also inhibited the receptor internalization in a dose-dependent manner. Calcium, which also affected the internalization of the receptor. PKC and phosphorylation, ERK phosphorylation inhibitor also caused inhibited in a dose-dependent manner to the internalization of the receptor. Therefore, to summarize this part of the talk, so what we have done is we used a series of inhibitors that we know to inhibit the GQ pathway and studied their effect on internalization of the receptor. And as you can see, they confirm that the GQ pathway is required for the internalization of the GLP-1 receptor. So we then went and looked at the importance of the C-terminus for the GLP-1 receptor cell surface expression, activity, and internalization. So we kind of, again, we deleted a series of regions within the C-terminus. GLP-1 receptor is about 463 amino acids. So we made a series of deletion at the N-terminus. For example, we deleted here 13 amino acids, 20 amino acids, 23, 33, and 53, and studied the effect on the cell surface expression. As you can see, when we deleted lost 53 amino acids, then we saw loss of the cell surface expression. Then we also made internal deletions, which also affected the cell surface expression. As you can see, the deletion of the between amino acid 411 to 418 also affected the cell surface expression, suggesting this region is important for the targeting of receptor to the plasma membrane. So we also confirmed that by immunofluorescence. So just due to the limitation of the time, I'm not going to details. And then we looked into the activity, which is in this case cyclic AMP production. When we use this series of deletion constructs, as you can see, the region between 419 to 430 is required for inhibiting the receptor activity. So then we looked the importance of the C-terminal domain for the internalization of the receptor. to just summarize, when we looked again the region between 430 31 to 50 is required for the inhibition of the internalization. As you can see here, when we made a deletion of 430 to 450, that affected the internalization of the receptor in liraglutide stimulated cells, which is a GLP-1 analog. So just for summary of this one is we identified the different regions of the receptor that required for cell surface expression, internalization, and also for GS coupling. Therefore, here the amino acids 411 to 418 are required for cell surface expression, whereas 430, 150 is required for the internalization, and 419 to 430 is required for the GS coupling activity. As you may remember, I mentioned there are a lot of GLP-1 analogs currently in use for the type 2 diabetes treatment. Unfortunately, they are not only expensive because they cost around $200 to $300 per month, but also they have to be taken in injection, and therefore it's very inconvenient for chronic conditions such as type 2 diabetes to take on regular basis. So there is a lot of interest in developing orally active and relatively less expensive compounds for the treatment which targets GLP-1 receptor. So we have chosen two compounds, there are many, to study how they control receptor behavior in endogenous system. So what we have done is we looked the small molecules importance in cyclic AMP production or activation of the receptor by looking the downstream cyclic AMP production, calcium accumulation, phosphorylation, and GLP-1 internalization. So when we look for the cyclic AMP, the small molecules has to be taken at 100 times more concentrated than GLP-1 to produce same amount of cyclic AMP production. And similarly, calcium accumulation is unaffected by or it's not produced by small molecules, indicating that the small molecules specifically activate only GS pathway, not the GQ pathway. Again, we looked into the ERK phosphorylation, which is also downstream of GQ pathway. Because this compound doesn't activate GQ pathway, therefore we didn't see any ERK phosphorylation in cells treated with this compound 2 and compound B. Since the GQ pathway is required for internalization, that also unaffected by stimulating cells with compound 2 or compound B. We then looked whether the small molecules and bind at the same site as which we call as allosteric agonist, whether they bind the same site as the orthosteric agonist, that is GLP-1, or they bind at different sites of the receptor. So to address that, we have used make use of antagonist called exenatide 9-39. As you can see here, when you use the GLP-1, activation of cyclic AMP production is inhibited by including antagonist exenatide 9-39, whereas the antagonist is ineffective in inhibiting cyclic AMP production by compound 2 stimulation or compound B stimulation. Similarly, there is no cell surface loss. That's internalization also affected by this antagonist, indicating that compound 2 or compound B binds at a site different from that of orthosteric binding site. We also just confirm that by making a mutations. One mutation we made in V36A, which affects GLP-1 binding. Another one is K334A, which affects the GS coupling. If the compounds 2 and B bind same site as GLP-1, then their binding also should be affected by a V36A mutation. But as you can see here, V36A mutation affect only GLP-1 binding, and because it's affected, we don't see any cyclic AMP production in when this mutated expressing cells are stimulated with GLP-1. However, when you stimulate same mutated expressing cells with compound 2 or compound B, we can see cyclic AMP production, again confirming that the binding site for this allosteric agonist is different from that of the endogenous, it's orthosteric ligand. So when we made a K334A, that mutant kind of decouples a receptor from the GS pathway. And when we mutated this, we can see a reduction in cyclic AMP production when cells are stimulated with GLP-1. Whereas if you look into it not only affected by this mutation affected activation of the receptor by GLP-1, but also by compound 2 and compound B, indicating that these ligands or agonists bind at different sites, but still bring a same conformational change that result in coupling to the GS pathway. So just to summarize this part, so if you take the GLP-1, it binds the receptor, which result in activation of the GS pathway, which result in increasing cyclic AMP production, whereas GQ pathway result in through increasing intracellular calcium ERK phosphorylation, the internalization. Small molecules which are orally active, what they do is they not only activate only GS pathway, but also inhibits the GQ pathway induced by the GLP agonist. So the second part is going to talk about the targeting of cancer using a particular receptor. Here it is interleukin-13 receptor alpha 2. So just for those who are not familiar with this receptor or the cytokine, IL-13 is play an important role in allergy and other atopic diseases. It has two receptors, low affinity IL-13 receptor 1 and high affinity monomeric receptor alpha 2. Alpha 2 is a decoy receptor because it has kind of shown to overexpressed in a variety of cancers and therefore become an important drug target for treating cancer. So what we have done is we developed a novel lytic peptide, which has its hybrid peptide, which has a one portion that binds to the receptor, the second portion which binds the cross membrane and disrupts the membrane and thereby kills cells by necrosis. As I already mentioned, it has a two moieties. One is PEP1, which binds to the receptor. Another 421, it is a cell membrane disrupting peptide. So what we have done is we studied whether this hybrid peptide specifically target breast cancer cells grown in 2D monolayers and 3D spheroids. 3D spheroids kind of a model system for the breast cancer tissue. So we look first in the expression of this receptor in cancer as well as non-cancer. As you can see, non-malignant compared to non-malignant in cancer, we see a 100-fold increase in the receptor expression, more so in triple negative breast cancer, which is confirmed through immunohistochemistry. The staining here, we stained with isotype control or anti-IL-13 receptor alpha 2. As you can see, the stronger the signal means more receptor expressed in triple negative breast cancers. Then we took a model cell lines which kind of corresponding to the non-malignant. For instance, MCF10A, which is corresponding to the non-malignant cancer breast tissue. MCF7, which is a model for non-triple negative breast cancer, whereas 231 and LM2 are two cell lines that represent triple negative breast cancer. When we look the expression at mRNA level, the expression is only seen in TNBC model cell lines. Similarly, total protein expression also seen only in 231 and LM2. And also the expression at cell surface is higher in TNBC model cell lines. So just it's a cartoon shows how the PEP1421 targets specifically cancer cells and thereby kill them. As you can see here, the receptor is overexpressed in cancer cells, and therefore the ligand or part of the PEP1 binds the receptor, thereby brings the lytic peptide into the close proximity of the membrane, which results in interaction through the charges, because normally membrane is negatively charged, whereas this lytic peptide is a positively charged, which result in binding and leads to membrane disruption, ultimately that result in cell death through necrosis. So we choose a cell line called COS7, which doesn't express alpha-2. So we overexpressed the alpha-2 and studied the effect of these two peptides, PEP1 on the proliferation. As you can see, reduced proliferation when we added the hybrid peptide, or when we measure the cytotoxicity, as you can see there is increase in cytotoxicity in particularly COS7 cells that expressing alpha-2, but not the control cells. And also we established the specificity. PEP1421 lytic peptide specifically targets interleukin-13 receptor alpha-2 expressing COS7 cells, but not luteinizing hormone receptor expressing cells. So we went and looked into the sensitivity of the breast cancer cell lines based on their expression. You can see 231 and LM2, they express high levels of alpha-2. Therefore, they are more sensitive to the lytic peptide, which is again confirmed through cytotoxicity assays. So we also looked into the mode of action. As you can see here, the necrosis is the process used by lytic peptide to kill the cells. As you may remember, MCF cells, they are kind of express very low levels of alpha-2 in normal condition, but when you treat these cells with epigenetic drugs such as trichostatin A, which inhibits the histone deacetylation, or 5-azacitidine deoxycytidine, which mainly inhibits DNA methylation, when we treat them with in a dose-dependent manner, you can see increase in alpha-2 expression in MCF cells when by these drugs. So we went and looked whether the treatment of the TSAR 5-aza deoxycytidine also increased the sensitivity of the MCF7 to the lytic peptide. As you can see, there is an increase in sensitivity of the MCF7 to the lytic peptide when we treated cells with either 5-azacitidine or 5-aza deoxycytidine or TSA. We also made a 3D culture. Again, the expression is high in 231 and LM2, and also because of the high expression, 3D spheroids also of 231 and LM2 are more sensitive to the lytic peptide treatment. Here we used a different approach to establish that ethidium homodimer, which is a fluorescence dye which binds to DNA, but it's membrane impermeable. That means it cannot enter into live cells, but in dead cells it enters and binds to DNA and fluor. We can visualize that by fluorescence microscopy. So the ethidium homodimer shown here in red color entered into spheroids only when they are treated with PEP1421, indicating that this lytic peptide can also target cancer in tissue format. Again, we also, when treated with cells with epigenetic drugs, MCM7 has become more sensitive to the lytic peptide. So the third part of the talk, I'm going to look into the importance of ADP ribosylation factor 6 small GTP signaling for sepsis treatment. So for those who are not familiar with sepsis, sepsis also known as blood poisoning. It has high mortality rate, about 5 million deaths per year, and it also causes severe complications such as septic shock, organ dysfunction, and acute respiratory distress syndrome. As you can see, it's been shown how it does severe complications here. The source of infection could be in lungs when there is a change in the permeability, which result in bacteria entering into a bloodstream. That is where we call this as a blood poisoning. And then it can cause leakage of blood vessels by disrupting the endothelial lining of the blood vessel, which result in organ dysfunction and ultimately cell death of the patient. So a few years ago, there's a group studied the molecular mechanism related to vascular permeability induced by the lipopolysaccharide, which is a component of the bacterial component. As you can see, lipopolysaccharide, it binds to toll-like receptor 4, which result in activation of two different pathways. One is NF-kappa B pathway, another one is activation of OP6. NF-kappa B pathway activation result in production of inflammatory cytokines, whereas OP6 activation leads to increase in vascular permeability. Therefore, indicating that we could specifically target OP6, thereby prevent vascular permeability or stop the vascular permeability without affecting inflammatory cytokine production. So we kind of looking on a novel assay to address this issue, to develop a novel drugs for the bacterial sepsis, for which we kind of choose an assay here, shown here. It's a high throughput manner. We can do this. You have a biotinylated gelatin coated as on the bottom of the well, and then you put monolayer of epithelial cell, endothelial cells, and then add on top fluorescently tagged streptavidin. In normal situations, streptavidin cannot pass through the gaps between the endothelial cells to reach a biotin to bind. But when you increase the permeability, then you can see an increase in the gaps, thereby streptavidin can pass through the gaps and bind biotin, which we can visualize by fluorescence microscopy. And here I've shown an example here. As you can see here, when we treated cells, monolayer of endothelial cells with various concentration of lipopolysaccharide, we have seen increasing the treatment resulted in increasing streptavidin binding to gelatin, biotin gelatin, indicating that vascular permeability increase. So one could use this assay to measure vascular permeability in a very quick manner and also quantitative manner. And after establishing this assay, we went and looked at the importance of OP6 inhibitors for vascular permeability. When we treated cells with OP6 inhibitors, so for those who are not familiar, OP6 is a small GTP binding protein. It exists in inactive GTP bound form and active GTP bound form. And it activated by GTP exchange factors, inactivated by GTPase activating proteins. When OP6 is active, it through effectors regulates vascular permeability. So if we inhibit this OP6 activation, then we should see a reduction in vascular permeability induced by, for example, lipopolysaccharides. So here we have done is we treated cells with pre-treated with OP6 inhibitor or OP1 inhibitors, and then studied the vascular permeability induced by a lipopolysaccharide. And the vascular permeability induced by lipopolysaccharide is inhibited by OP6 inhibitors, basically QS11, SECNH3, and NAV, but not by NF-kappa B inhibitor or OP1 inhibitor, indicating or confirming that OP6 play an important role in regulating vascular permeability. So while we are looking to the literature, we identified that coronavirus actually it's become lethal and kind of third stage of the infection, and thereby cause kind of similar to sepsis to organ dysfunction, and which leads to death. In fact, it's been shown trace a ferocious rampage. It's shown to be affecting brain to toes in infected people. So, as kind of last 3 days been listening to the importance of COVID-19, which is a global pandemic disease. It affects about 5% of the patients, which are mainly patients become critically ill, and it is caused by SARS-CoV virus, coronavirus 2. So coronavirus, SARS coronavirus 2 enters into nose lining mainly through using ACE2 receptor. And if our immune system fails to clear the virus in the initial stage, then this virus through windpipe attacks lungs. In the lungs, virus become deadly by causing disrupting the oxygen supply. And in serious cases, it can also not only damage lungs, but also many other organs. One way it does is by leaking the blood vessels. So we wondered, can we use the OPSIC signaling pathway we studied in bacterial sepsis for the COVID patients or COVID infections? So we kind of made an hypothesis. For example, let's assuming that SARS enters into blood vessel from the source of infection. Here in this case is lungs, and then it causes blood leakage, which finally result in organ dysfunction, and that leads to death. So what we have done is we hypothesized that inside the endothelial cells, SARS-CoV binding to ACT receptor result in activation of OPSIX, which result in increasing vascular leakage, and which ultimately result in organ failure. We couldn't do much in confirm this or testing this hypothesis due to lockdown. Once the lockdown is lifted, PhD students eager to study this, testing this pathway, and so that we might able to develop anti-viral treatments for COVID-19. In addition to, we have been studying other aspects which I didn't have time to go through. One of them is we work on mesenchymal stem cells requirement for bone healing, as well as we also work with vascular LVAD, which is artificial heart pumps for the people who are critical stages of heart failure, how blood gets sheared and all those things in collaboration with a local company called Callan Cardio. So just I will end by showing the picture of Swansea University. You can see it's one of the campus called Singleton campus. These are the buildings, and this is the sea front you can see from the office. So it's a lovely view of the university. And I will stop here and take any questions.
Yes, sir. Thank you, sir. Thank you for the wonderful presentation. There are a few questions, and I will start with the first one. Can we study the N-glycosylation of proteins with mass spec?
Yes, you can do it. In fact, that's one way to do initial stages to identify how we can measure the N-glycosylation. Because when you use the mass spec, the mobility of glycosylated peptide fragments would be different from non-glycosylated, and based on that, one could study which sites being glycosylated.
Thank you, sir. Another question. The inhibitors are specific for these proteins in the pathway, or they are already in use for any other diseases?
Yeah, some of them, for example, F phosphorylation inhibitors are already in use for cancer treatment, whereas others are less specific because it's the pathway is very ubiquitous. That means it's required for normal functions as well. Therefore, it's more difficult to target this pathway unless we specifically introduce these inhibitors into cancer cells. One way to do that is by targeting. That means you introduce or put them into like nanoparticles or liposomes, and then decorate them with specific ligands that target those liposomes or nanoparticles to cancer cells, and that way you can specifically target the pathway.
And the next question, and in case of nanoparticle, what would be the base material?
I think it could be used. Yeah, you can use a gold particles, or I think even nowadays people are using. I think liposomes are better than the nanoparticles because there are some side effects with nanoparticles because they tend to accumulate inside the liver. Therefore, I would say
Exactly, and this is something I had in mind. Yeah.
Yeah, you can do is you can alter it by, let's say, by decorating with a specific receptors, or sometimes you can modify by pegylation. If you put a peg coating, then you can prevent aggregation and accumulation. But I think still we are early stages of using nanoparticles. Whereas if you use liposomes, which are similar to the exosomes inside our body, and therefore they won't be rejected, as well as it's more easy to target, and fusing exosome to the target cell is easy, and then the contents can be easily delivered into the target cell.
Yes, sir. And are the C-terminal domain, I'll go back to the question. Are the C-terminal domain mutations are previously known, or they are isoforms of same proteins?
No, there's no. This is a very basics work we have been doing. Only known mutation we know is it's in the N-terminus in Japanese population, which affects ligand binding of the GLP-1 to the receptor, and therefore those patients are less responsive to GLP-1 based drug treatments.
Okay. And are there any side effects for this diabetes drug?
As for, in fact, it has a better usage nowadays. This is the only known drug. Like I'm guessing it is. They're talking about liraglutide, which is not only useful for treating type 2 diabetes, but it can also used for weight management because it's the only known drug or well-established drug to regulate weight because it inhibits lipolysis. I'm sorry, it protects or it will prevent insulin inhibiting lipolysis in fat and therefore useful for reducing the weight.
And I have two more questions to finish. And the
Sure.
first question is, I think it's a continuous. I think the first question with two parts. What's the length of the synthetic peptide for IL-13RA2, and what's the dose you are using for treating cancer cell lines?
Okay, there are two different things. I think for the type 2 diabetes, GLP-1 is about 30 amino acids, which is generally used at 100 nanomolar in vitro studies. In vivo, there is a fixed amount because it cannot cause hyperglycemia or hyperglycemia. Sorry, it's
Yes.
kind of a.
Yes, I muted the speaker. Yeah.
Okay. So it's a well established dosage you can use for GLP-1 analog for the type 2 diabetes. Whereas for the cancer, the lytic peptide we are working, you need it in micromolar concentrations. For example, we have to have a micrograms, like 1 to 2 micrograms in vitro studies. Whereas in vivo, we don't know yet because we haven't started animal work.
And another question is, what happens if we block soluble AC2 protein?
It's very important question and interesting. In fact, I was also looking into that because one way to develop drugs is you can develop a minimal structure of ACE2. Therefore, you can prevent virus binding to membrane-bound ACE2. But I think what I understood is it's not a straightforward because it seems soluble ACE2 actually increase the uptake of virus. Therefore, if you try to use that as a drug, it may not work. So increase in ACE2 levels may be not protective, but it can be kind of make more infection. Is that what you are asking, or is that different?
I think that answered the question. So I think the participants are expecting the paper references to be pasted once more, and you can share it in the chat box as well, sir.
Okay.
And one last question to finish again. I think that's the recent question I got. What is the difference between black poisoning and COVID-19? And I mean, I think probably they referred to sepsis, I guess.
Yeah, it's a blood poisoning, probably they meant. Blood poisoning
Okay.
it's a generic term. Any foreign body enters into the blood is called blood poisoning, because it induces immune response, or it elicits immune responses. It could be COVID-19. It can also cause a blood poisoning or bacteria. It's a generic term. That means it affects the blood. Once foreign body enters into our system, it causes it can lead to organ dysfunction through blood leakage from blood vessels.
But is it predominantly bacteria or like any other organism that enters any other foreign body which enters the human system?
Predominantly bacterial. In fact, it's mostly the streptococcal pneumonial based bacteria, but sometimes you can get local. In fact, mitochondria can cause autoimmunity. For example, if there is a trauma which result in lysis of cells and release of mitochondria, which can cause sepsis. So it is called non-bacterial induced sepsis, which is also as dangerous as bacterial induced sepsis.
Yeah. Like self-replicating organelles anyway, so even if they could be considered as a foreign body.
Yes, yeah.
Yes. And so it's definitely different from COVID-19, I mean the SARS-CoV-2 virus, what we generally term it as blood poisoning, isn't it, sir?
Yeah, I think the compared to what I understood, the SARS-CoV-1 mainly localized in the lungs. It doesn't enter into the bloodstream, whereas this new version, coronavirus 2, enters into the bloodstream and cause havoc in a whole body. It causes from head to toe. I think the post-mortem samples, many people got shocked the kind of destruction this virus can do. And therefore, yes, this blood poisoning can be lethal. Luckily, it is not happen most of the. I think it's about 1% of the infected people affected by seriousness of this infection.
So hence, with respect to this virus, it's termed as a foreign body entering into the system, and can be termed as blood poisoning as well.
Yes, yeah. Blood, as I said before, blood poisoning is a very generic term. It could be virus, it could be bacteria, or it could be mitochondria. As I said, our own mitochondria, when it gets released during trauma or any other situations, that can also cause sepsis.
Does this condition prove fatal?
Yeah, that trauma very often in trauma people gets organ failure due to this particular sepsis, non-bacterial sepsis.
Okay, sir. And I think with that, the questions are over. And then again, as I said, participants are requesting you to share the paper references once more so that they could follow it in the future. And thank you very much, sir. And we sincerely apologize for the time delay.
It's fine. It's fine. I think the references are there within the slides, or they have a copy of it, or we are not going to.
I think participants can definitely go back to these videos in the future.
Yeah.
And then you can always look into the presentations and get these references. I think that will help for sure.
Okay. So you want
so you can type it in the chat box, sir, but if there is a big list, And I think I would recommend the participants to go back to the videos. It will be and it will be recorded automatically in the YouTube, and you can always go back to this presentation, listen to it once more, and you can take the references from there. So that will be good enough, I guess.
Yeah,
And
I think most of the references, yeah.
yes, and so it should be fine. And thank you very much, sir, for the wonderful presentation. And yes, and participants, there is a short break, and we will resume back at 2:30 p.m. for the student presentations and poster. I mean, especially the poster and the oral talks. So we will go for a short break now. And especially I request all the presenters to return back at 2:30, and we want you to present, if it is a talk, it should be within 10 to 12 minutes, and of your presentation, and 2 to 3 minutes of your of Q&A will be conducted. I mean, the questions will be asked for 2 to 3 minutes, and for posters, 5 minutes, and 5 minutes of your explanation, and I think 2 to 3 I mean 1 to 2 minutes maximum of Q&A. So participants, please at 2:30 p.m.
Hello, Divya madam.
Yes, sir.
Oh, madam.
So, and also we request you guys to kindly look into more about Vignan University. So you can, so we are going to play a short video of the university, and you may take a look at it, and you may take a look of what they are actually doing and where are they located, etc. And kindly take a look of these videos in during this break, and we'll see you back again at 2:30 p.m. Hello, Pranav. Hello, Pranav.
Yeah,
Can
yeah.
you? Yeah. Is it possible to switch on the audio as well?
It's playing. It's playing. Audio is playing.
Oh, really? I think I
Yeah,
didn't.
I can hear in YouTube.
Oh, I see. Probably it's not very much visible in the WebEx, I guess. I mean, it's visible in the WebEx. It's not audible in the WebEx, I guess. Or should I have to do something to switch on the audio?
No, no.
Okay. Yeah. I think it's not audible here, as some of them commented.
In YouTube, it is audible.
Okay. In YouTube, it is audible.
Now you can hear?
One sec. No. No. I mean, ideally, there is only a background music for the video, I guess.
Yeah,
And
that's it.
unless there's any, a testimonial or something in there.
No. Just music only.
Just music only, so it should be fine.
Yeah.
Yeah, you guys can take a look. Well, somebody is saying, oh, Mahesh is saying like it's not audible in YouTube as well. But there's not going to be any dialogues in this video or any testimonials given by the university board. It's going to be just an introduction about this university and where it's located and what facilities it's got. And because they are the main organizers, and I have to definitely announce this at this point of time, and I've been doing this since morning. So VIGNAN'S Foundation for Science, Technology and Research is located in Guntur, Andhra Pradesh, India. And they are the main organizers for this e-conference, and they are like their specialization is more into like immunology based research, immunoinformatics in fact, precisely. So kindly take a look at their videos. You can always go back to their videos too later. If you wish, we can post the link and you may go back to the videos later to have a look. Yeah, I think, yeah, once you type in vignan.ac.in, you will be able to see the video. So here is their website. Here is their website and about them, their future of students, departments, and the courses that they are offering, and the kind of research they are doing. Everything is mentioned here in the website. So kindly take a look. So yes, we will come back at 2:30, and in the meantime, you can take a look of their website and video. Thank you. Hello everyone. I hope you are all back to listen to presentations. So I have the schedule for the afternoon, and we will go with the first speaker of the afternoon session. So let me check. So before we move on to the oral and poster presentations that are meant for evaluation as well, so I request Deepakshi Kasat. She's the founder of the BioTalk e-magazine to deliver a short talk about her online e-magazine. Yeah. So Deepakshi, are you available?
Yeah, I'm here.
Yeah, hello. Hello, everyone. So here is Deepakshi Kasat. She's a biotech graduate and she has recently opened an online magazine. So you can look into her. She will also talk about her website and everything. And so here she is. She's the founder of the BioTalk. So can you
Can I begin
start?
now? Yeah, sure.
Yeah.
Can you hear me and can you see the? So thank you everybody for having me here on this platform. So I'm Deepakshi Kasat, the founder of the BioTalk magazine. I started this magazine 2 months ago in May 2020. Basically, I had just completed my undergraduate in biotechnology and I wanted to do something productive, bridge the gap between students and the industry. And I came up with this idea of the BioTalk e-magazine. So before I go further, I would like to show this short promo video that I've made so that you can get a brief idea of what this magazine is about.
Deepakshi, can you make it into a slideshow?
Yeah, sure.
And we couldn't listen to the audio.
Um, that's right.
Yeah, one second. I think we have. I think Pranav can give you an idea. One sec.
So, Deepakshi,
Yes.
when you're sharing the content,
Yeah.
when you click share content, so you will see an option called optimize for text and image on the top,
All right.
share content. So there you need to optimize for motion and video. Okay, one thing you can do, like you can stop sharing first.
Or I can just continue with this because like it's fine if the background music isn't there. If the content is there.
Okay, no problem.
Okay, so but please make it a slideshow.
Yes, ma'am.
Yeah, and you can talk over the video.
Yeah. This is a short promo of a BioTalk magazine, and this is what we do. You can. Section of our magazine, which I'll be watching the promo video, I'll start talking about what the magazine is and how I came up with this idea. So after my undergraduate got over and I was trying to connect with the industry, I found that there was a huge gap between the students, their skills that they acquire, and the industry's requirements. And so that is when I thought of connecting these two, bridging the gap, and making a platform where people can put forth their knowledge, their views boldly, openly, and connect with other people as well. So that is where the BioTalk magazine came into picture. And yeah, we are a biotech magazine platform where, as our tagline says, we are biotech for everyone. We want to make our articles accessible to the biotech community per se, but to everybody who's reading our magazine. So even though we have topics like COVID-19 research and genomic sequencing and all, which normal people aren't aware of, but we edit it in such a way that it is accessible to not just the community but to people as well. So yeah, that's about what we do now. A June issue. Now, what happened was initially when I started this magazine, I thought I'd just reach out to a few friends, my professors in college, and limit it to them. But I also thought that might as well put like a small poster or calling for articles and try to see if anyone responds on Facebook biotech group. So I reached out there and suddenly, and I was surprised by the kind of response I got. I got deans from universities. I got the PhD scholars, industrialists, and students coming to me to send in their articles. So this kind of faith on such a new platform was incredible. So that's how I came up with the June issue. Students sent in their articles, professors sent in their articles. We broke it down. So it was a process where you send in your topic, your synopsis. Our editors review your topic and see if it fits the guidelines of our magazine. Then we send you certain guidelines for formatting and the kind of we send you certain points that you can add use in your article. Then we give you a period of 10 to 15 days to write an article and then send it to us. So sending an article does not necessarily guarantee it being posted, but we try to make sure it is we edit it for grammar, for style, for tone, and then finally post it on our magazine. So when we post something on our magazine, it is viewed to the entire world. It is accessible to the entire world. We do not keep the copyright of what you submit to us, which means you can use your article elsewhere as well, but it will also stay on our magazine. Now, when we publish the July edition, you can download the June edition on our website as well and view the July edition as you go to the website. Now, we have 4 different sections on our website. The first one is recent discoveries or news. Second one is opinions. Third is you inspire us section, and fourth is the editorial part. So in what's new, we cover everything that comes under biotech, which is a recent discovery, recent theory. For example, COVID-19 is a hot topic. 3-4 articles on that as well. And these are all these articles are viewing COVID-19 from a different point of view. Someone thinks it's a conspiracy. Someone thinks someone covers the IP aspect of it. Someone covers the diagnostic part. Someone covers the therapy. So we have a very holistic view of every problem at hand, which we put in the magazine. Second part is in the news section, we also have this research corner where people who fail to publish their researches in a journal can send us their research, and if we think it is legit and if it is well put and well researched, we will put it in our research corner for a month. Next we have is the opinion section. Now what I've seen is at a lot of places we get a lot of filtered content. We do not get what people actually think, what their perspective is on a lot of issues. So this is our opinion section, which is why it said bold basic biotech, where you can put forth your views very boldly.
Deepakshi, your slide hasn't changed.
Yeah, ma'am, because I'm just talking about this.
Oh, okay, that's
yeah. Okay. Yeah. So that is what our bold part, that is what our opinions part is. We had this writer Rohit Singh talking about the COVID-19 as in a very very bold way. I'm sure if you read the article, you would actually connect with it. So this is a platform. Since we're not restricted by any political or educational influence, we can be out there as we want to be. However, we do not we do not intend to hurt anyone's sentiments. We have a screening process to, if the content is too vulgar or good, we cut it down and put it appropriately. Now, next is the every month we put an interview of a scholar or a professor or an industrialist who has successfully contributed something to the biotech community. Last month we had Dr. Anupama Harshal. She is an independent scientist of the Department of Biotechnology in India and a professor in Bombay. What these people talk about is they talk about their experience, why they chose biotech, because I feel like there is a lack of awareness of our field. Like computer science, IT, these fields are so out there, everybody knows about it and everybody wants to do it. But biotech, which is such an important field, is not really given that much importance because of the lack of awareness. So these people will come, they will talk about their experience, why they chose this, and mainly the opportunities there are for students, for freshers, even for their colleagues who can collaborate with them. So we put some opportunities out there as well. And last is the editorial section where editors, me and 2-3 editors of mine, we post our what we think recent topics are, and we post an article over there. So now, what is your role and how can you contribute? Firstly, I thank this platform so much for having me here, because reaching out to such an immense biotechnology audience, that is incredibly useful for my platform. You can send us articles, you can send us your topic, your synopsis on the email ID, and we will review it. We will get back to you. And if your article, mostly or most of the articles we get are worth posting, honestly, after a few edits here and there. But we will get back to you immediately with the kind of work you give us, and within a few days you will see that posted on the website and reaching out to a massive audience. Now we are a non-profit magazine, so we're not making any monetary benefits here. We just want to contribute to this biotech community in any way possible. If you go to our website and see our June edition, and if you like it, if you have any issues, you can send us your feedback on biotalk.m@gmail.com. And if you have any articles, anything related to biotech, you can send it to us on connect@thebiotalkmagazine.com. And also, we have recently collaborated with, or actually we're in the process of collaborating with LLB Scientifically. They are a YouTube channel. They have a YouTube channel of their own. We have organized this LLB-SCHOOL. So they'll soon be posting a promo video there. We are very thankful to them to give us this opportunity to reach out to even more audience. And LLB Scientifically has honestly pushed us very well. In return, they've also, in this collaboration, they'll also be sending us articles from their institute. So you'll get to see major content over there as well. So yeah, send us your articles. I'm free to answer. Thank you.
Thank you, Deepakshi. Thank you very much for the introduction of BioTalk. And yeah, I've just posted the website link in the chat box. You can also do it from your part as well.
Yes, thank you.
Later. And I think participants and audience, you may contribute if you are related to biotech or bioinfo, whatever, you can contribute some nice content to BioTalk magazine. So it's a non-profit magazine, and I think they have some good content. They are maintaining the standards as well. So I think it would be very nice to go back to it once. Thank you very much, and thank you, Deepakshi, for the talk. Thank you very much. So, okay, so now the next presenter. I think the next presenter, it's going to be a poster presentation. So we will have Anil Kumar available. So do we have Anil Kumar here? So before we move on to the presentation, please watch the VIGNAN'S University video. So, Arun Kumar, just a second, like we wanted to put up this video before your talk. Pranav, can you restart the video once again? So that's it. And now please listen to Anil Kumar's poster presentation. So Anil Kumar, please share your content. You may share your content now. So poster presenters, you have 5 minutes to explain your poster, and then you will have like 2 to 3 minutes of Q&A, depends on the number of questions. So please restrict to the time. Anil Kumar, can you listen to me? I couldn't hear you. I couldn't hear you. Yeah, can you hear me now?
Yeah, madam.
Yep. Okay.
Clear my voice, ma'am?
Yes, your voice is clear. You may start. Yeah.
Okay, thank you. So, good afternoon. So, myself, pharmacy, Rajmandri. Conceivable character of traditional medicinal rhizome turmeric for prevention and management of the novel pandemic COVID-19 infection. So, COVID-19. So now everyone facing this pandemic situations. So not only nowadays, almost like human history is observing a very strange time fighting invisible enemies. So now we are facing the novel coronavirus. So this novel coronavirus is episode was announced as a public health emergency of international concern by World Health Organization. So overall, these pandemic situations, so everyone have to boost up immunity. So that is a very important criteria in these pandemic situations. In this lockdown period, I was collecting the review articles and research articles. So what is the importance of the
Anil Kumar, I'm hearing
why?
some background. Anil Kumar,
Hello?
I'm hearing some background noise. Kindly mute your YouTube.
Okay.
Yeah.
I was literature review, the medicinal rhizome of the turmeric. why I am collecting in our India ancient times. So the traditional medicine practice is very important in our India. So in our government is also announced Ayush
Anil
department.
Kumar, still I can hear it actually. Can you please mute it? Just mute your YouTube or close your YouTube at the moment. Okay, thank you. Yeah.
Clear?
Yes, it is fine.
Yeah. So April 21st, the Ayush government, the government is the official notification is announced. At present, there is an treatment for COVID-19 infections. That's why you have to improve our immunity boost up and increases the Indian traditional medicines have a wide potential for usage in such pandemic conditions, owing their long-standing use in the community and huge number of ancient references and large number of publications in the scientific journals. So on there, these phytochemical constituents is made up of action and clinical efficiency is very good. At the same time, it is also essential to have a scientific evidence on use of any way of formulation and prevention management of COVID. So I was collecting these literature review. The medicinal rhizome of turmeric. Turmeric, generally, we are using the turmeric is a aids in making our immunity stronger, and its immunity is our body's natural defense against the disease causing either bacteria or virus. And are you observing in my poster? You see in the first line is a completely arranged in the traditional way, because the turmeric has been used in the terminal from birth to burial. So it is viewed as a hallowed and promising, and the piece of turmeric is also turmeric and colored in thread is used as a sting in humors in Indian people. So it is applied in whole body in the bride and on the earlier day of marriages. So that is the complete pictures, and even government colors and market apart within are viewed as a favorable. So at the same time, are applied on the cheek, forehead like that, and Indian women have been as an attractive in the great Indian writing in their face, bodies are sparkling in yellow. So at the same time, it is also used in the Hindu temples like ceremonies, for example, homa and pujas like that. So what collecting these are, so it is a traditional material, and at the same time, as per the literature review, it acts as a good antiviral and antibacterial, antifungal, anti-inflammatory action. So the broad research over the past 50 years has demonstrated that the curcumin is the main constituent in this turmeric. It is also a part of a golden spicy turmeric, and a broad clinical trials over the past 50 years have tended to pharmacokinetic safety and efficiency in these nutraceuticals against the various infections in the people. Some peoples also promoting and seen in the various pro, including cardiovascular diseases, joint inflammatories, cancer, peptic, gas, like that. So many of diseases, the curcumin or curcuminoids are the one of the best remedial therapeutic infusions throughout the hundreds of years around the world. So the rhizome of the turmeric is very much reported in the treatment of in the different respiratory conditions. Why? Because in this pandemic COVID-19, the signs and symptoms is also like shortness of breathing and respiratory event of diseases. So that's why it is the curcumin is very helpful for this. The cure of, or not a complete cure, at least you have to prevent and management of these symptoms, because the curcumin is also against the variety of viruses, so including virus, vesicular stomatovirus, and felin infectus, and herpes simplex virus. These type of virus is also proved the curcumin is completely reduces the activity virus. I hope the COVID-19, the coronavirus also some better give the curcumin is give the some better activity against this COVID-19. This not only why is very important because of like to have to take the some culinary uses of this turmeric. You know, everyone a spoon of the turmeric powder with a glass of milk twice or thrice daily is very effective, best on the empty stomach. And if in case we have to take the half teaspoon of the fresh turmeric powder mixed with the 30 ml of warm milk, is very effective and throat infections like that. Why? Because in the COVID-19 patients are also suffering from the like that coughing and dryness of the coughing, throat infections like that. So before you have to take the some precaution and management of these symptoms, definitely you have to increases your immunity booster. So that's why you have to face these pandemic situations, especially who are suffering of this is the seasonal time, rainy season. So definitely this type of influenza virus, flu virus is developed. So that's why before you have to take the some awareness, definitely you have to cure these type of signs and symptoms. And the curcumin, everybody knows the curcumin is a golden spice. The curcumin is a kitchen of queen. So that's why the curcumin is used the preparation of. That's why you have to take the more quantity of the curcumin. Why you have to take the more quantity? Because the curcumin and turmeric having the low bioavailability, at the same time high elimination. So when more quantity, there is no problem in our body because it will be ready to facing the elimination. So that's why you have to simply have to take the 3 to 4 sufficient for per day. So overally, what is my future as the curcumin? So one of the significant concern with the developing of the curcumin for clinical accuracy is very important. At the how to take the nanotechnology is broadly. So how to go for the nanotechnology based novel methodologies are by and a large forceful investigated worldwide to improve the curcumin's bioavailability and reduces the perceived toxicity. And it is also helpful who are suffering from these symptoms like cold, cough, high fever, shortness of breathing, and gastric disturbances. So obviously that will be helpful for everyone. So and the curcumin is answered to boost or enhance the immune response during the viral infections and multi antiviral resources. Potential of the curcumin demand, estimating its likely application to control of the COVID-19 along with modern therapeutic practices, but some experimental data is need and that one. So are you observed? I think in my poster, the first line the traditional importance of the curcumin, and second line and like the glass of milk that is a fill up with turmeric, and next one is the importance of the kitchen of queen in turmeric in our kitchen. And see in the last if you are improve your booster or immunity, definitely have to against the viruses like flu viruses. See everyone, that is the my main theme, that is the prevention and preventive and management of the novel pandemic COVID-19 infections. So I hope I cover my review and thoughts. Yes.
Yes, thank you. Thank you, Anil Kumar. And participants, do you have any question? Any questions, participants? So do you think so? Turmeric turmeric is usually like antiseptic and anti-healing and all, no, and all those things. How can it really? How can it really support, or how can it be like very antiviral? So, do you think there are lots of articles supporting antiviral properties?
Works for the antiviral activity in this so against of the influenza viruses, herpes simplex virus, and viruses, especially hepatitis B viruses. These viruses, they are completely the antiviral activity in this curcumin. So that's why I'm choosing the curcumin. Is maybe in future it will be useful for the COVID-19 patients. And what is the my theme is the symptoms, like you have to suffering from any cough, cold, like that. So immediately have to take the gold milk, that is the mixing of the milk with the turmeric. So that will be is very recovery, even within 1 or 2 days. Definitely have to recovery. That is the main thing, and you have to use the more natural, because the pushing the oil department. So how to do the medicines researchers. So that's why and in this situation, the felt necessary to make a serious effects of the development of the drugs based on any oil systems only recognized the Drug and Cosmetic Act 1940. There is no regulatory provisions in this Drug and Cosmetic Act rule 1945. So that's why clinical trials, like Ayurvedic Siddha, is some difficult the facing the clinical trials. So at the same time, you have to generate the clinical data and scientifically has undertaken the consult of the DCGI, CDSC, as well as the other research experiments. So definitely have to increase this, because already so many antiviral, anti-inflammatory wound healing activities is done. The curcumin is the best medicine for the tradition. So obviously the COVID-19 is also the higher severe actions, like moderated symptoms and low symptoms. They are taking the curcumin. So definitely they are relieving and the throat infections.
But so far it has been more of an assumption or prediction based on its effect on previous viruses, right? So is it right to tag it with COVID-19, or it is right to say that it can be very helpful with respect to COVID-19 infection as well? Because it's not really proved, because we do take turmeric like every day into our cooking, but still people get affected by COVID-19, right? So what's your comment on that?
Here, generally, the turmeric is adding the cooking. So what the herbal medicines are these curcumin or curcuminoids. When you have to give the more expose of the heat, what will happen? Natures immediately they are evaporated. So that's why we have to take in the food, but the big quantity of the curcumin is entering our body.
So what is the limit, or yeah, so what is the limit or the maximum limit that you can suggest for having this curcumin?
Yeah, so nearly per day 3 to 4 per day. So you have to take
should it be in uncooked form? Should it be in uncooked form, or it should be into cooking?
Yeah. So
okay.
In our Indian Prime Minister have to take the gold milk, the mixing with the gold, milk plus is very helpful. So
does the excess amount lead to toxicity? But does the excess amount lead to toxicity, and what can we do?
If in case you have to take the more quantity, there is it not shows any toxicity, because the turmeric and curcumin are elimination rate is more low bioavailability, because Bioavailability means the rate and extent amount of the drug is present in the systematic circulation. But it shows the low bioavailability, definitely it will be ready to elimination. So that's why when you have to take quantity, this will be in the elimination rate. So it excreted through urine, whatever it.
Okay.
If in case you have to take in more quantity, it now shows in the loss more toxicity.
Okay. And one last question to wind up. And what do you suggest that how curcumin in our body?
Yeah, ma'am. Once again.
I mean, so the question is, how can we boost absorption of curcumin in our body? So it has to be, I mean, absorption, or it's how can we increase the bioavailability as well? So, I mean, bioavailability in the sense it should be like a purified curcumin or the crude content of turmeric itself.
Extracted the curcumin, then you have to convert it into the tablet. Generally,
Okay.
if in case you have to take conventional dosage form, it is ready to facing the elimination rate readily. But if in case you have to convert it into the sustained release or extended release, any controlled drug delivery, that will be what will happen. These drug delivery systems are maintain the sustainability in our body almost more than 10 to 12 hours. So definitely some quantity of curcumin is lost, but the quantity is gradually releases. So it will be maintain some absorption of the curcumin is body.
So the absorption will not be very significant, or its bioavailability may not be very significant if it is taken as a wholesome food, but in the form of a capsule where curcumin is predominant, where it is purified at its best levels. So that will be more beneficial to cure or to fight against the COVID-19 infection. Is that it? So hence it's clear, and thank you very much for the presentation. Yeah.
Yeah, thank you, ma'am. Thank you so much.
Thank you. So now the next presenter will be Fabina. So Fabina, are you ready? Can you hear me? Okay. Can you hear me?
Yes, ma'am. Can you hear me?
Yes.
Um,
Yeah.
okay, good afternoon, everyone. I'm Dr. Fabina from Saratoga College. The title of my poster is The Molecular Docking Study on the Inhibition of COVID-19 Protease by Exploring the Therapeutic Bactive Compounds from neem. We all know that the coronavirus disease 19 is caused by novel SARS coronavirus 2, and it is causing infecting many people all around the world, even death in some places. As a director indica, neem is a member of Maliaceae family, and it has been widely used in Chinese, Ayurvedic, and Unani medicines worldwide. Even in Indian subcontinent, it is used in various treatment and prevention. This neem has more than 140 bioactive compounds isolated from its leaf, stem, flower, seed, so on. And they are used traditionally for curing many diseases. And these bioactive compounds have been identified to possess activities like antimicrobial, antimutagenic, antioxidant, anticarcinogenic, and even it has an antiviral property. The literature shows that it has a capacity to inhibit influenza virus, herpes simplex virus, polio virus, and even the early stage of dengue virus. So I thought of seeing the energy between this bacter compounds from neem and coronavirus protease. So in my study, I have chosen 5 main protease of COVID.
Is
So
it
these
okay?
yes,
Okay.
ma'am.
Yeah. Yeah.
These proteases are downloaded from protein data bank in .pdb format, and the ligands were chosen from Dr. Duke's phytochemical and ethnobotanical database, and the ligands were downloaded from PubChem in SDF format and later converted to PDB in Open Table format. And all the ligands were checked for Lipinski rule. I have selected 16 back compounds. So in 16, 13 compounds have either a 0 or 1 violation, and only 3 had more than 2 violations. So I left that, and I have taken that 13 to my study, and I have compared it with 2 anti-HIV drugs, Sapinir and Elvinafir. And then the docking was performed in Autodoc 4.2 version, and all the confirmations were analyzed, and the protein ligand complex was saved, and the interaction were visualized in PyMol. So here you can see the results of the protein and the ligands of active compound. Here I have included only the photos of active compounds which have binding energy more than that upper threshold minus 6 kilocalories per mole. So when if you look at this graph, these are the various bioactive compounds and the main proteases. Here the binding energy of the protease and lymph were good in 6YB7 when compared to the other proteases, but some of the compounds like aciduridine, beta-citrosurol, epiacetodiridine, and vetnin, they have equal or more binding energies when compared with antiviral drugs, Secumir and Elfinavir. Similar findings were observed by Kumar, but he used different receptors, 6VSB, spike glycoprotein main protease, and he compared with remdesivir and lopinavir, and even he got good binding energies. And then Shanmuga Subramaniam, he docked with same 6LU7, but with different bioactive compounds from me. He also got excellent binding energies and compared with remdesivir and hydroxychloroquine. So my conclusion is, in my study, that aciduridine, beta-citrosurol, epiacetodiridine, and vetnin appear to have a best potential, and it acts as a coronavirus protease inhibitors. So, however, it is necessary to investigate the potential medicinal use in vitro and both in vivo conditions. Thank you.
Thank you, Fabina. There is a question. How similar is the HIV protease to SARS-CoV-2 protease? So as you have considered protease inhibitor of HIV as reference, how stable is your interactions?
With stability, I didn't study, ma'am. I just checked the binding energy.
Okay. What is the further a plan on this project? Are you going to do any molecular dynamics simulations, something like that?
Maybe I'll do, ma'am, because I'm new to this docking. I just learned from the YouTube. Actually, I'm a microbiologist.
Oh, I see. And what is the software do you use?
Autodoc, ma'am, 4.2 version.
Okay. So, and I think you didn't answer the first question. How similar is the HIV protease to SARS-CoV-2 protease in terms of its structure, or I mean,
Structural similarity, I didn't study, ma'am. Maybe I'll look after it.
did you look at its sequence similarities or something like that?
Maybe I'll do, ma'am. I'll study hereafter.
Okay. So, what you took some structures from PDB and you verified the potential ligands and then docked with the protease, isn't it?
Yes, ma'am. And I checked the
Okay.
binding energy.
Okay. And then, okay. Yeah. So any other questions, participants? Okay. So with that, yeah, thank you for the post, and we will move on to the next. Yeah. So the next speaker will be Safia Sultana.
Hello.
So I think the next poster presenter. Yes, Safia Sultana. Can you hear me?
Yes, ma'am.
Can you share your content?
In the share content, we'll have to share through share screen.
Yes, exactly.
Just a minute, ma'am. Is my screen visible, ma'am?
No, not yet. One second. Probably you are not the presenter yet. I will have to. Yeah. Okay. Please try now to connect to share your content using the share content option. Yeah, can you hear me?
Yes, ma'am. Webex would like to.
Yeah, now please use the share content option, which is next to your video icon, and use the share screen, and then you can actually open your poster document.
No, is it visible, ma'am?
No, not yet. Yes, yes. Are you using your mobile?
Yes, ma'am.
Okay, so yes, I understand it's okay. So you can blow up, for bits and pieces of the poster while you're explaining. So please use the landscape mode. Yeah, please use the landscape. Hello, yeah. Please use the landscape mode. At the moment, it's in portrait.
Yes, ma'am. 1 minute.
I think.
I'll visualize it when I'm explaining, ma'am.
Okay. Yeah. I think you can. Yes. That should be fine. Yeah.
Shall I start, ma'am?
Yes.
A warm good afternoon to one and all. I'm Safia Sultana from Sri Ramachandra Institute of Higher Education and Research. I'm currently here to present my e-poster on the topic de novo virtual analysis in identification of natural phytoconstituents for anti-COVID-19 therapy. So coming towards the introduction part, I would not like to grab much attention in explaining all the basics of coronavirus. Henceforth, giving you a brief outline would include coronavirus, which came into our notice in the month of December in Wuhan, was classified under beta coronavirus and was identified as a severe acute respiratory disorder. Till date, there is no drug treatment available for it. Various pharma industries and health sectors are trying to launch either a vaccine or a drug or some other alternative treatment for COVID-19. When I started my research, it was remdesivir and chloroquine, which was given to the coronavirus patients. So I took those two compounds as my standard drug. So further, when I went into this research, there were researchers who focused only on a single target, like either a main protease or a helicase or a papain-like protease. But I would like to suggest that a virus infection would occur when it binds to the cell and it undergoes replication, and then with the help of some structural proteins, it will further be replicated and release enormous number of virus inside a cell. This is how a virus replication takes place. So instead of focusing on a single target, if a virus escapes that target, it might enter into the cell and also produce various number of viruses inside it. So it's important to target more than one, specifying not only a single target and considering more than one target. So in my present study, I have considered around 7 targets as I have visualized here. It is main protease, papain-like protease, helicase, and NSPs, which is nothing but non-structural proteins, 12, 14, 15, and 16. All these targets were downloaded from PDB. There were various number of targets under a single name itself, main protease. So shortlisting of these targets was done based upon the literature review and also based upon the sequence similarities, resolutions, crystallography, etc. So coming towards the aim and objective of this study, witnessing the history, we can say that nature has always been an open-hearted to provide drugs for every infection and treatment. So in this pandemic situation, I'm currently considering all the natural phytoconstituents from our Indian medicinal plant databases. So around 15 to 20 plants are shortlisted from the IMPACT database, that is Indian medicinal plant database, for either respiratory disease related or coronavirus related. So further shortlisting of the phytoconstituents of each and every plant was done using knapsack database, as is visualized here for Tinospora cordifolia. So shortlisting of all these phytoconstituents from 15 to 20 plants gave me around 550 to 600 phytoconstituents. Further shortlisting of those 600 phytoconstituents was done purely based upon the literature review upon either it is related to respiratory or coronavirus. So from that, I could conclude around 168 phytoconstituents. So those 168 phytoconstituents were from these 15 medicinal plants, such as Zingiber officinalis, Mentha spicata, Terminalia cebula, etc. So these 168 phytoconstituents were further taken for bioactivity score prediction and molecular property prediction. As we all know, a drug once enters into a body should have a particular molecular property to undergo absorption. So bioactivity score is nothing but the drug to which receptor it will bind and how effectively it will bind. Further, moving on to the next step, after shortlisting, after finding out the Lipinski rule and bioactivity score, they were further docking is nothing but a computational tool used for determining binding efficiency, that is how well a ligand will bind to that particular receptor. It can include various interactions like hydrophobic interactions, etc. So docking was not performed with a single tool because a single tool will give you a particular efficiency, but performing docking using various tools can give you a cumulative result that will be very beneficial in concluding. Further, the analysis was performed using for ADME properties, which is adsorption, distribution, metabolism, and excretion of those best selected candidates. For the toxicity parameters was also performed, since it plays a major role when a drug is given to a human being. So the 7 targets, as I told earlier, were as follows. Then coming towards the result part. So this is the result which I got for Lipinski rule. From these results, I selected, this was performed for 168 phytoconstituents, but I was able to incorporate only 11 here with 2 standard drugs, that is remdesivir and chloroquine. The drugs which showed either 0 violation or 1 violation was considered here, and the top 11 were the standard drug. Similarly, the bioactivity score was also calculated. In this way, I used the first docking tool, which is nothing but COVID-19 docking server, which was recently launched as a as a similarity to that of the Mastro of Schrodinger, which is considered one of the standard tool. And then the second docking was performed using Pyrex docking tool. And finally, the third docking was performed using Autodoc 4. And all these results were accumulated to determine which phytoconstituent can be taken for further evaluation. As you can see, amiritoside D and tinocordifolin, they were the two phytoconstituents which provided better or efficient results when compared to other phytoconstituent and also the standard drug remdesivir and chloroquine. So further, the ADME parameters were estimated for these two phytoconstituents, and it showed that it has better absorption through HIA model and CaCO2 model, and it showed better pharmacokinetic property, that is GI absorption also was high, and it also had a good range bioavailability score. So coming towards the blood-brain barrier, which was not a part of this, but I have included that here. So it also has mild permeability to the CNS. So, and the toxicity parameters was performed using test software through oral rat LD50 predicted values, and it gave values range was 500 to 1000 mg per kg. So it can, the drug will not be toxic to around 1000 mg per kg. Ethyl dose 50. So by this way, I could conclude that amiritoside D and tinocordifolin belonging to the family Tinospora cordifolia, one of the Indian medicinal plant, can be considered as an effective candidate for taking further in performing various investigations like in vitro evaluation, in vivo evaluation, and considering it for a treatment for COVID-19. Thank you.
Thank you, Safia. So one question is, either remdesivir or hydroxychloroquine are not targeting SARS-CoV-2 proteins, I think. Is there any reference to support this claim to use as a reference?
When as I stated before, ma'am, when I started this investigation, only those two drugs were used. So this was a very long process for shortlisting each and every phytoconstituent based upon those two standard drugs. So shortlisting was done upon those two drugs, and further now whatever the drug is going to be launched, or as they are saying in August 15, the vaccine will also be launched, and various other Indian medicines are also being used. Based upon that, I would further perform more docking results for correlation and present it as a larger work being done upon coronavirus.
Mm-hmm. But they are not targeting SARS-CoV, right? So they are used as treatment option only. So which COVID docking server or I mean, what is that? Okay, there are
COVID-19 docking
I mixed
server.
up. So there are two questions I mixed up. That's the first question is the follow-up of the previous. But so when you said that you've used these two drugs like based on what you got as an information, I mean the remdesivir and hydroxychloroquine, chloroquine, sorry, but they are not targeting SARS-CoV, right? They are only used as treatment options. So what do you say about it? Any comments?
Till date, they have not specified any antiviral drug being used. There was a drug, that is Fanivir, which was being considered, but later on it is also now not considered for its treatment. That's why pharma industries have been working on for developing any antiviral that will target that SARS-CoV. So initially they considered that this remdesivir and chloroquine can together produce a synergistic effect in targeting that SARS-CoV. So that was the reason I selected this. But as you have, as I have got this question, I would start working on various other drugs that has been given as an article in targeting the SARS-CoV-19 and incorporating that with my results.
Yeah, that is. We need drugs that could directly target to mitigate its action, right? So, other one is which COVID-19 docking server you are talking about. That is what, and this is from Arshi Naqvi. She's asking which COVID-19 docking server you used.
The name is itself COVID-19 docking server. You can check it in the Google Scholar also. You will find various articles in it. And initially it was started in the month of December by a Japan, I forgot the name actually, from someone from Japan who did a research and who launched this. And it is similar to that of Mastro of Schrodinger. They have mentioned it in that article itself.
Okay, okay. Thank you for the nice presentation. Yeah. And now the next presenter is going to be, please hold on a second. It's going to be Solai Murugappan.
Yes, ma'am. Hello? Yeah, ma'am. So ma'am, I'm actually doing a paper presentation and not the poster one.
Yeah. I didn't mention it as anything. So you are giving an oral presentation, I know. Yeah.
So shall I start sharing?
Yeah. Please.
Just
So what's your, so I have to assign you into a presenter. Oh yeah, you're already, yes. Yeah, you may start.
so is it visible?
Now it is, and please switch it on the full slide mode, and now you can start. Yes.
Yes. So good afternoon, everyone. I'm Murugappan, a final year B Pharmacy student from Madras Medical College. Today I'm going to present on the combination of 3 potent herbs that can inhibit the pathogenicity of SARS-CoV-2. So I think the structural proteins of SARS-CoV-2 have been the recent druggable targets, and these are something which all of us are aware of. But my targets are not these structural proteins. So apart from these structural proteins, we have non-structural proteins, and there are approximately 16 non-structural proteins. So among these, NSP5, which codes for main protease, and NSP3, which codes for papain-like protease, are my targets of interest, because these two proteases first autocleave from their polyprotein chain, and then they only after they get freed, they start to release the other non-structural proteins and stimulate the viral replication and the cytokine expression and all. Another thing which was recently discovered is that the papain-like protease strips out our host, like our macrophages and all the other cells in our body of ubiquitin and interferon stimulating gene 5, which is essential for activating the innate immune response. So if we can inhibit these two proteins, then we can control the pathogenicity of SARS-CoV-2. So my next target would be the NFKB signaling pathway. So NFKB signaling is the reason why there's this over expression of cytokines at the later stages or the hyperinflammation phase. So what happens is that initially when the virus enters our body, it evades the immune response, but at the later stages it activates the entire immune response and causes hyperinflammation by stimulating too much of NFKB signaling. So if we can inhibit it, then again the cytokine response and all the multi-organ failure which are witnessed in the later stages can be controlled. So our third target would be NLRP3 inflammasome. So as mentioned earlier, the like NFKB, this NLRP3 is also responsible for the cytokine production and hyperinflammation. So the NFKB will activate the NLRP3, but apart from it, when there's excess presence of damaging signals like E protein and all, the NLRP3 will also get activated, and its activation will reproduce active caspase 1 enzyme. So this enzyme will again stimulate the production of interleukins, that is the pro-inflammatory cytokines. Another thing is it actually stimulates the nearby macrophages also to stimulate cytokines by forming gastrodermal pores on the particular macrophage, which is host cell which it is currently occupied in. So through the formation of gastrodermal pores, 1 minute, ma'am, I think it's changed. Yeah. So through the formation of gastrodermal pores, it releases all these pro-inflammatory cytokines, which goes about and attacks the nearby macrophages. So what are my herbs of interest? Androphagus paniculata, Ginkgo biloba, and Vitis vinifera. So first up is Androphagus paniculata. So this will act at the hyperinflammation phase, and it will inhibit the activation of NFKB signaling pathway. So this particular herb has the active constituent andrographolide, which is a diterpene lactone. So basically, this is the structure of andrographolide. So as you can see out here, there's this exocyclic double bond. So previously, like some years back, when this studies were done on this particular herb, and it was found that this exocyclic bond is responsible for binding to the cysteine residue of the P65 subunit of NFKB transcription factor. So when it binds to this particular factor, it will reduce its movement into the nucleus and also its transcription into pro-inflammatory cytokines. So currently, dried extracts of the leaves are very useful for effective and acting as antiviral. And it was found that increasing the dose will increase the increase the immunomodulatory effect. So this was a docking study which was done some long time back by some particular scientist. So as I mentioned earlier, this is our andrographolide. It will bind to the cysteine residue of the NFKB P50 subunit through covalent interaction, and through some other interaction, non-covalent interactions also, it will inhibit the binding. It will inhibit the action of NFKB signaling. So if this signaling is inhibited, all the respiratory syndromes which are witnessed in COVID-19 patients like COPD or even the ARDS can be controlled. So our next herb is ginkgo biloba. So ginkgo biloba again has a lactone as an active constituent, which is zincolide A. So zincolide A is actually useful to control the two important phases of COVID-19, which it acts at the early response phase and in the hyperinflammation phase. So at the early response phase, because the papain-like protease will strip off our proteins of the ubiquitin and interferon stimulated gene, if this particular activity is inhibited, our own immunity will start working against the virus, and it will destroy the virus. So what this particular herb zincolide A does is that it will inhibit papain-like protease, so this particular activity gets restored. And in the hyperinflammation phase, this will again inhibit the movement of NFKB into the nucleus, and it also inhibit the degradation of NFKB from its inhibitory protein IKB, because when it is present as a complex, it will not go inside the nucleus. But when it is released, it will go inside the nucleus and it will start the production of pro-inflammatory cytokines. So it will so it will make sure that this complex is stable and that there's no release of free NFKB. So recently, two scientists have done docking studies on this, and they have found out that ginkolide A interacts with the papain-like protease through hydrogen bonding and van der Waals interactions, and then it can be used to inhibit papain-like protease. Another one was that by Neda Shagahi, who found out that ginkolide reacts with the main protease, and it is found to bind to 3 highly conserved amino acids. So basically, these two show that ginkolide can be effectively used to inhibit these two proteases of COVID-19. So finally, our third herb is resveratrol. Resveratrol is found from Vitis vinifera, which is actually grapes. So the dry skin of grapes you will get a lot of too many concentration of resveratrol is found in the skin of grapes. So basically, what resveratrol does is it will stimulate the sirtuin-1, which is an acetylase, and stimulation of this deacetylase. Sorry. So this sirtuin-1 will inhibit the NFKB signaling and the NLRP3. So if as a result of this inhibition, again our pro-inflammatory cytokine formation is reduced, and we can control the inflammation response. But this is visible at lower doses itself of 5 micromolar. And right now they have found out that about 25 micromolar of resveratrol itself can increase mitochondrial autophagy. So it will increase mitochondrial autophagy by activating the P38 mitogen activated protein kinase, which in turn will reduce mitochondrial damage, because E protein is a damaging signal. That is the envelope protein of COVID-19. It will damage our mitochondria, and because of this damage, NLRP3 gets activated. So what our resveratrol does is it will ensure that this particular activation or mitochondrial damage is suppressed and ensure mitochondrial autophagy. So the activation again is reduced. Finally, why I am saying that these three herbs can be used as a combination to control COVID-19 or treat COVID-19 is because andrographolide is actually called as Nilavembu in Tamil. So I think it was used to treat dengue. I mean, everyone is aware that it was used to treat dengue and was used as an antiviral, which means it is actually very potential and effective. So the similar things is that similarly, ginkgo biloba has also been used by many individuals for its anti-inflammatory and immunomodulatory effects, and the same goes for resveratrol. So basically, all these we already have, as I mentioned out here, 400 mgs are taken of andrographolide taken every day, and even ginkgo biloba is taken as 40 mg or 60 mg tablets. So we have established doses of these per herbs, which can be used as supplements in our daily life. So if we can try to find out a combination to formulate a dose or dosage and take this every day, then maybe we can, because of the synergistic effect of these three, we can tackle COVID-19 and its responses in human beings. Finally, I would like to say that these three herbs are potential to inhibit the various stages of SARS-CoV-2 disease progression. But these are currently used as supplements across the globe. So even at 600 mg per day, they do not produce toxicity. And even if they do, it is only mild like nausea, vomiting, and stuff, which go away with time. So, but the combined use needs to be studied further. And if someone takes up and studies the use of these three herbs, then yes, we could control not only the viral replication, but also the immune response, which is produced at the later stages. So thank you.
Thank you. Thank you very much. So I have two questions. Okay. In fact, three questions. So what are the common qualities of the three herbs you mentioned in providing a potential solution to this COVID-19?
Like, yeah,
So I think it's a bit of, I think people need a bit of a summary kind of.
okay.
Yeah.
Ma'am, basically all these three can be used to control the hyperimmune, that is a pro-inflammatory cytokine response, a cytokine storm, which we witness at the later stages of COVID-19. So that is the commonality between these three. I mean, they do act at the same stage, like they act at NFKB signaling at the NLRP3 inflammasome and some other like the protease thing, but at the end of the day, they do the same thing, control the immune response. Hello?
Okay. And does suppression help in combating against this COVID-19? So normally people try to use immune boost, right? Immune boosting
Yes, ma'am.
compound.
I get it, ma'am. So actually, as I think in the first day, one of the lecturers told that in COVID-19 there are three pathogenic phases. The first is the viral response phase, and the second phase I don't remember the name, but the third one is the hyperinflammation phase. So initially, our immune response gets evaded by this particular virus, and so there is no immune response, and that is the place where the immune boosters come into account. But when the 10th day goes, when you cross the 10th day, your immune response gets reduced, and at that time we need something to, sorry, your immune response becomes too much as an overexpress, and you get into the cytokine storm phase. So what happens at that time? If you suppress the your immune system, you can control the organ failure and all. So these drugs, instead of giving, even if you take it normally, there are no side effects at all. But if you give it to the ones who are in the hyperinflammation phase or on ventilator, then you can control their immune response and the side effects. But this is only a theoretical approach because I don't know if it'll, because based on the current existing literature, it seems to be effective.
But the one in the ventilator requires a lot of immunity, right? Immune boosting ability, right?
Yes, ma'am, but actually the one on the ventilator is also, being affected by the organ failure. His lungs get affected, which is why he's damaged, which is why he's on ventilator. So the lungs get affected because of the cytokine response. So basically, I thought we could suppress the immune, then the immune response, then it will, naturally revert and all.
Okay, and once it revives back, and then you want to support it with immune boosting compounds as well. So that is what you mean, yeah.
Yeah.
And combining all these three compounds can affect each other's bioavailability or would improve it.
That is what I'm saying. Actually, I do not know if we can, right now, Andrographis paniculata is given in combination with 9 other herbs, and there's no modulation of its bioavailability. But ginkgo biloba and vitis vinifera, they're only given as supplements as, individual components. So basically, if research is done to find out whether we can give these three together, then that would be good, because from the literature search, there's no information on this aspect at all.
But is there any composition or a ratio of these three herbs that you want to give it to the patient?
No, I don't have any composition or ratio, but yes, based on existing papers and all, it is found that you can give around 400 to 600 mg of these three herbs extract per day, and there is no side effects at all to that particular patient. Not to COVID-19, but to normal people and to the diseased people, like the general cases.
But why did you, but how you picked these three in the first place, and how can they be like a combination? It's quite difficult to imagine in a way, But of course, they all have medicinal properties. No, I mean, no doubt at all. But how did you pick to arrive at a combination?
Yes, ma'am. So basically, recently our CDSU and all stated that instead of going with a single drug to treat COVID-19, we need to go for combinations. So I was thinking, why don't we start for looking for herbal or phytopharmaceuticals and in combination? So at that time, I wanted to choose for drugs which are effective against the main protease and the immune signaling. So when I started going through literatures, I found out that andrographis paniculata has been extensively used for cancer studies, especially for its immunomodulatory effects. So since the signaling pathways are same with COVID-19, I thought yes, we could even use it for COVID-19. For ginkgo biloba and Vitis vinifera, it is because ginkgo biloba recently there are molecular docking studies, so I chose that one. And for Vitis vinifera, it is because one particular university abroad is currently doing clinical trials on this particular compound, and they are saying that this might be effective in treating COVID-19. So that instead of going, individually using these compounds, why not go with a combination? So that was my basis of
Is-
picking up.
But resveratrol has like wide benefits when it comes to like wide benefits on anti-aging,
Yes, ma'am.
So
Anti-aging, antioxidant, and too many. It has too many positive effects actually.
exactly. So, okay, yeah. And there's one last question to finish.
Yes,
There will be a synergistic activity of various metabolites in the host, right? So, what is the need to use three plants together for immunoboosting? I think to correct it, it's like it's going to be an immunosuppressor, right?
yes, ma'am. This is an immunosuppressant. Immunoboosting effect it doesn't do. It is actually an immunosuppressant, and you're right, ma'am.
Yeah, and what will be the synergistic activity? Like there will be a synergistic activity of these metabolites in the host, but what is the need to actually use these three plants all together?
Altogether is because if we use them together, the immune suppression will be higher, and the chances of getting cured from COVID-19 might be faster. So that was my ideology actually.
Mhm. So like it's like all these three compounds contribute to contribute like an additive effect.
Yes, ma'am.
Yeah. Okay. So I hope this answers your question. Oh, okay, there's another one. I'm sorry, but if you suppress more, is there any other infection that can cause
Yes,
even
ma'am.
even
Yeah.
a adverse effect?
Yes, ma'am. So that was what I was saying. This is purely a theoretical approach. If some scientist again takes up this combination and does the research work, then from clinical trials we can find out the adverse effect. Right now, this is just a possibility. So there's no clear idea about what are the adverse effects or if there are any or not.
Okay, so I think it's important to standardize the dosage and
Yes.
standardize the formulation,
Yes,
etc.,
ma'am.
and then to bring it to a commercial product.
Yes, ma'am.
Okay, thank you, and thank you for the presentation. Yeah.
Yeah, ma'am.
And who's the next presenter? Shri Harsha. So do we have Shri Harsha here?
Yeah, I'm here.
Yeah, please.
Yeah, I can
Yes.
share my
Yeah, I could
video.
see you. Yeah.
Can you see it?
Yes.
Okay. Yeah. 1 minute, please. Yeah.
Yeah.
Yes. Good afternoon, everyone. I'm Sri Harsha Vaddamudi, an alumni of VIGNAN'S University, and I'm currently doing my master's in ASC at FAU, Germany. So today I would like to talk about the symptoms and social responsibility one should have during the hard times of COVID-19. Well, I choose this topic because at my university, when I was voluntarily working with the student groups in the mask production, I could understand the importance of social responsibility to the virus to actually stop it to a certain limit. So I could clearly say that from past few months, COVID-19 has been keeping the world in suspense. So places that were once teeming with hustle and bustle of daily lives have become ghost towns with massive restrictions put on our lives. So it's an unparalleled global response to a disease. But without jumping directly into my topic, I would shortly take some time to speak about what actually is COVID-19. So it's a infectious disease which spreads from a small city to the world, now dealing with the pandemic of the virus. It infects the cells lining your throat, airways, and lungs, and turns them into coronavirus factories that go on to infect more cells. So speaking how it spreads, we can see that the basic reproduction number actually refers to the spreading potential of the virus. So each infected person can at least transmit to one another person. It spreads primarily through the droplets of saliva or from the discharge or from the used tissues. If come in contact with some other person who is not infected, it can spread it like that as well. So from an infected person to another, those two in turn affect two more and so on. And how fast does it spread? Well, for humans like us, something which grows linearly is easy to understand, but viruses don't grow that way. They grow exponentially. Exponentially, I would say that imagine a chessboard, and if you see the picture on the right side, it's a map. So you would wonder, why would I put a map with rice grains there? So if you imagine a chessboard and then starting with A1, if you add a rice grain to every day, on a linear growth for 64 days, we might have 64 grains only. But on an exponential growth, after 64 days, there would be like incredible number of rice grains. So that's how the virus spreads. Here, I would like to quote some study done by author Adam Kuchersky, who analyzes the dizzy outbreak mentioned about the dots theory. The D in the theory stands for the duration, which defines the duration of infectivity. The longer a person is ill, the longer they can infect the people. And if you talk about the opportunity, it means how much chance do we have to spread the virus from one person to another. This clearly maps our social behavior. And the transmission probability is how likely the virus actually transmits from one person to another. They say that actually happens on every third occasion. So now we know the virus is transmitted. We can talk about the susceptibility. This is how much it is possible for a person to get well. So the vaccination does not exist at the moment, clearly, so we can't really talk about it. Moving from this, talking about the incubation period, this is when the virus is establishing itself. So the incubation period is the time between infection and first symptoms appearing. So this is widely on an average of 5 days, but it can be sometimes from between 7 to 14 days. And now coming to the main part of the symptoms, as I mentioned already about symptoms, it's time to talk about it. So the fever is, as you can see from the picture, is mostly all what people will experience. 8 out of 10 people got these symptoms of fever and cough, and the body aches, sore throat, and headache are possible but not guaranteed. Fever is actually a sign of your immune system responding to the infection. It recognizes that the virus and tells us that the hostile invader has entered your body. And cough is initially a dry one, probably due to the irritation of cells. And also some people have coughed up sputum, thick mucus, which they say contains the dead lung cells killed by the virus. And this is the best we need to understand at the initial stage. However, studies emerging say that it can also cause cold symptoms as well. So if the disease progresses, then that means our immune system is overreacting to the virus. And chemical changes causes inflammation, damaging our body parts, which also might lead to severe disease like pneumonia and etc., because there is a lot of breathing difficulties in your lungs. And on a critical level, there are around 6% of cases where people are critically ill, where the body starts to fail and sadly leads to more chance of death. And how contagious? What is the contagious period? On a person showing the symptoms we spoke about, there are chances of them to be contagious for 7 to 12 days if the disease is mild. We know that the virus is transmitted by droplets, so the deeper they come from in the lungs, the longer the virus remains active. As now mentioning about how the virus is transmitted, we should talk about our own responsibility and think about others as well. When you notice mild symptoms, self-isolate at home for at least 7 days to reduce chances of being infected. As you can see, you should probably wear a mask, cover your face wherever you're outside, and then practice social distancing, maintaining distance, because when someone coughs or sneezes, the small liquid droplets may contain virus, and that's how you get infected, and you may be infected with the virus as well. So if you talk about the do's, we need to stay home whenever it's possible. Avoid going to the crowded places because you're more likely to come into close contact with some who are infected. And work from home if you can, because that is much nicer, I guess. It's difficult with the virtual world now, but I guess that's much safer. You need to get used to it, at least for some time. And if you're unwell, isolate yourself. Safe home and self-isolation is better, because even if you have minor symptoms, it might be a problem for someone else who is not really ready to be infected or who's very well and who's scared about all this virus. You cannot just go and spread this virus to another person. And you can have someone to bring the supplies for you. If you're back home alone, call a friend maybe and then ask them to pass on the supplies, just keep it in front of your door, pick them from there. That's how it works. That should work. I mean, that's how people have been solving their problems. And avoid public transport. Even if you use the public transport, there you can use certain measures such as wearing a mask from getting infected or infecting others with your minor symptoms also. But mostly suggested would be to avoiding them as much as possible and keep in contact with each other, because there are issues of mental health, people suffering from mental health. And I guess being in contact with your friends and family and saying hi, how are you, doesn't take much of your time. Like 5 or 10 minutes a day with one person is actually good for your health as well. And washing your hands for about 20 seconds with soap will actually is very much needed, because hand rubbing will kill the virus that may be on your hands. And sometimes you can use a tissue for coughing and sneezing when you're in public or when you're at your home. If you do not have a tissue, you can use your sleeves. And not to do is surely not touching your face and eyes, because our hands touches many surfaces and can pick viruses that can surely be transferred and infect you. And close contact with people who are unwell can be avoided for the time being. I know we are there for each other. We are there for friends and family. If they're not well, it is mainly suggested to stay away from them. But the main question, I am sure, would be like, does this all help? I mean, well, when our warriors, our doctors are doing so much to fight against the virus, and many other great researchers are putting all the time in finding the vaccine, I guess we can surely try doing these basics things, and not every time and every day we get to spend time with our family members. So, or find some time to spend for yourself. So I'm sure you don't want to regret later when the virus bids a goodbye. Talking about the end game, the final question for which no one has still found an answer is, when will we actually get on back with our lives? When will it end? According to me, what everyone needs is an exit strategy. That means a way of lifting the restrictions and getting back to normal. But remember, the virus is not going to disappear. The restrictions is what is holding the virus back to an extent. No country has a clear-cut exit strategy yet. It's a societal change and it is a lot of big challenge. I'm sure it'll take time. Other than that, what we can do is turn the tide. That means send the coronavirus packing back home. It surely would take a long time for the tide to go out, but mentioning again the DOT strategy, that means isolating the sick, avoiding social contact, washing hands might flatten the curve, and that's what we surely aim for. The third alternative would be changing and adapting to the new normal, because there is no clear endpoint as of now. And adapting to the new normal would surely keep the transmission rates low. on a clearly long term, vaccine is the one way to fall out of this, and hoping for that to happen as quickly as possible. Let's make sure that we do our duties right. Stay home, stay safe. Thank you. That's it from my side.
Okay, thank you. Thank you, Harsha. So now I think before we move on to questions, participants, we have turned on the responses button. So you may actually send your feedbacks using the link. And any questions? Harsha, I have a question.
Yeah, sure.
In the recent reports, or even people, there are some media talking about like social distancing at the moment is not really working, and despite that, there is increasing cases. So, do you have a comment on that?
Yeah, so I think social distancing worked for a while, surely worked for a while, because take an example of the country I'm staying in Germany, we did not meet people for like 3 months. I mean, I sometimes meet people now, and I've seen people generally moving, and I don't think it's back to normal as always, but the social distancing has reduced in a way, just keeping it confined to 4 or 5 people rather than having a huge crowd. And obviously, we are following the basic measures yet still. We still wear a mask when we are outside. When we're meeting our friends, we surely say hi and bye, but we are hugging or shaking hands is not what we do, like unlike the normal times. So social distancing did work, but I guess we need to do it in the right way. That would be probably the right thing to do. And yeah, it has.
Yeah, and there is like beyond that, there is science that has to play an important role to mitigate this COVID-19 actually. So, any other questions, participants? And I think one of them commented that your slide quality is really good, and thank you for the wonderful presentation. Oh, yeah, there's another question. 2 more questions. Yes. So, what do you think? How long we will be in the practice? Pranav, I guess you meant about sensing. Okay, yeah.
Okay. So I'm not sure about how long we will need to practice, but I can clearly say that you can practice it on a long term until and unless you are sure that the virus won't affect you. And it's nothing wrong. I mean, it's about yourself. So think about yourself and your family, and maybe we can just predict the vaccine to be coming up in the next few months, and it will be all great. I mean, yeah, that should be fine. Practicing is nothing wrong. I mean, I'm almost used to it now. So I carry my masks if I go out because I've been doing that for 3 months now. So
We have been trained to do that in some or the other way.
yeah.
I think, but all these practices in a way are kind of debating as well. You know, there should be another session or a conference really talking about, with regard to the social responsibility and all. So yeah, there's more to talk about this, and that's what even the media debating on a day-to-day basis. And there's another question to finish about the face mask. Is there a way to wear it?
Yes, there is surely a way. I would say that if you're using your homemade masks, like from the cloth masks, make sure that you do not use them after once, or the suggested would be if you use it once, then probably wash it again, like soak it in the sun and then wear it again. Don't use them again and again. That is not good. And if you're using the surgical masks, there is a procedure of disposing them. It would be great if you dispose it in that way. And here we use some at our university. We researched on certain mask production where you can use them by we can reuse them by after 3 times of usage by putting it in an oven or heating it at a 70 degrees centigrade, and then we can again use it. So that the material is made like that from the polymers department. So we can do that way. So yeah, the way of wearing it would surely be not to touch the area where your mouth, nose, and face will be in contact with your hands. The internal part of the mask should not be touched with your hands because you're not saving the virus to come in contact if you're touching the internal part. Yeah, that's it. Thank you.
Okay, that's it. And thank you. Thank you for the presentation.
Thank you.
And then I think we have three more people to go. I think we missed one of them in between. Is Nooper Parim available? Okay. So if not, Sabah Fatma or Ruby Anjum. So anybody available? Yes, yeah. Dr. Sabah?
Yes,
Can
ma'am.
you, yeah, can you be the
Can
next
you hear me,
presenter?
ma'am?
Yeah, I can hear you. You may share your slide now. Yeah, you may share your, I think, yeah, you are the poster.
Yes, ma'am.
Am I right? Yeah, okay.
Is it visible, ma'am?
Not yet. I request you to use the share content icon, which is next to the video mode. Yes. Yeah. Yeah. Yeah, please switch on the slideshow.
Now visible, ma'am?
Yes.
Okay. Should I start now?
Yes, yes.
Good afternoon, everyone. Myself, Sabah Fatma. I'm the MD scholar in the Department of Tahaffuzi wa Samaji Tip, that is Preventive and Social Medicine.
Sabah, your voice is feeble and there is some background noise. Can you be a bit louder?
Okay, okay.
Okay, thank you.
It is better now, ma'am? Hello?
Yes.
Okay. Good afternoon to everyone. Myself, Sabah Fatma. I am the MD scholar in the Department of Tahaffuzi wa Samaji Tip, that is Preventive and Social Medicine, in the School of Unani Medical Education and Research, Jamia Hamdard, New Delhi. And the topic of my poster presentation is non-pharmacological approach to combat the impact of COVID-19 on mental health. As we all know, COVID-19 is a global pandemic that is supposed to infect millions and kill up to thousands of people, with a troubling increase in cases every day. No doubt it is a deadly virus. However, what majority not yet considered are the secondary mental health effects that will likely occur, the ongoing efforts to reduce the impact of COVID-19. At present, billions of people worldwide have been told to isolate themselves to prevent the spread of COVID-19. No doubt, this self-isolation strategy is the correct and necessary method to reducing human-to-human contact could help to lessen the impact of the disease. This social isolation will likely lead to an increase in anxiety, depression, insomnia, and other mental disorders, a link that has been established in previous studies in humans. On the other hand, due to lockdown, there is a lot of uncertainty developed in society in regards of education, employment, and economy, which is also responsible for ill mental health. Unani system of medicine is an ancient system of medicine which deals the patient on holistic parameters jointly together for preventive and curative aspects. In this poster, the efficacy of non-pharmacological approaches to combat mental illness through Ilaj bil Giza, that is dietotherapy, Ilaj bil Tadbir, that is regimental therapy like Dalak, massage therapy, Nutul, and other modalities will be discussed. The objective of this poster is to review of the non-pharmacological approach to combat mental disorders which may arise in the pandemic with the help of Unani system of medicine. As we all know, commonly prescribed sedative or hypnotic medicine should not be taken for longer than 3 weeks. Their adverse effects, addiction, and dependency limit its abuse. And in comparison to sedative medicine, we may provide an effective method of control of insomnia and depression without any adverse effects, addiction, and dependency. In Unani system of medicine, Mizaj, that is temperament of an individual, act as an imperative risk factor for various mental disorders such as stress, anxiety, depression, or melancholia. There are 4 types of temperament which are described in Unani system of medicine, such as sanguine, that is Mizaje Damvi, choleric, Mizaje Safravi, phlegmatic, Mizaje Balgami, and melancholic, that is Mizaje Sodavi. Although mental disorders seen in all 4 varieties of temperament, but according to the ancient Unani scholars, among the 4 temperaments, the people with melancholic temperament, that is Mizaje Sodavi, predisposes an individual to develop the depressive symptomatology most. Now come to the management part. First one is dietotherapy, that is Ilaj bil Ghiza. Ancient Unani scholars prescribed diet to the patient of anxiety, insomnia, and depression on the basis of humoral theory so that the correction of the abnormal temperament will be done. Here are a few principles generally suggested by renowned Unani scholars like yellow bile and black bile producing item, that is molide soda vasafra, must be avoided like stale, salty and astringent food. Light and delicious food items are recommended. Emollient diet, which we called as Murattib Agzia, which provide moistness such as barley water, cucumber, pumpkin, milk, greeny leafy vegetables like spinach, lettuce, etc. are suggested. It is advised to use bilious concoctive fruits such as orange, lemon, damson, and other citrus fruits. It is highly recommended that the diet of an individual, especially anxious person, include about 20 to 30 grams of fiber a day. Most of us probably take only 10 to 11 grams a day, only half as much as we need. Inadequate intake of fiber in diet contributes to constipation also. Chapatis must be preferred to over rice. Gourd or cucumber are recommended amongst the vegetables to patients suffering from chronic stress. Both are documented diuretics which did the excretion of pathological melancholy. Kaddu is also beneficial in delirium. Buttermilk and curd should be a part of the prescribed diet. Among the fruits, watermelon, pomegranate, sugarcane, and grapes are helpful. Citrullus lanatus, that is watermelon, and sugarcane are known for their diuretic properties. Punica granatum, that is pomegranate, is a potent antioxidant. Vitis vinifera grapes is effective in degenerative nervous diseases. It is also a reported nootropic diet with known adaptogenic and antioxidant effects. Now regimental therapies, Ilaj bil Tadbir. First of Ilaj bil Tadbir, first of all, Mutul, that is irrigation. A watery preparation.
Hello, Dr. Sabha. Can you enlarge it a bit so that people can see your poster more clearly? Yeah, I think that's better. Yeah. Yes.
Nutul therapy, that is irrigation. A watery preparation like decoction, infusion, oil, or solution, either hot or cold in state, is poured from a particular height over the affected part of the body to cure disease called nutul. In case of mental disorder, it is poured onto the forehead. Nutulate murattiba, prepared with banafsha, that is Varla odorata, Nilofar, Pulesurk, Tukmikadu, Leptuca sativa, Kashnizi subs, Coriandum sativum, Post, Abe Shibbat subs, Anithium sova, and Tukme Khashkas, Papaver somniferum, and Hordeum vulgare are very effective. The other therapy is Dalak, that is massage therapy. Massage of head with Rogane Kaddu, Rogane Kahu, and Rogane Khashkas, each in equal amount at night, induces sleep. Apart from this, massage of forehead and sole with Rogan Labube Saba is also very effective. Hammam, that is the bath. Mu'tadil hammam is useful in mental disorders, particularly after digestion. Avicenna recommended sweet and lukewarm water for insomniac. Murattib hammam is also very effective for removal of dryness of brain. Riyazat, that is exercise. It is also helpful in inducing sleep, but exercise should be light, not vigorous in the nature. Lakhlaka. Lakhlaka is inhalation. It is aromatic based preparation of watery or solid drugs which are kept in a wide mouth container and allowed to inhale through nose. Lakhlaka of Ave Barge Kahu subs, that is Lactuca sativa, and Tukhme Khashkhas, that is Papaver somniferum, and Rogaine Nilofer is beneficial in insomnia. And other type of regimenal therapy is shamoom. Shamoom is the drug which is sniffed and its volatile constituents reaches to nose is called shamoom. Afune, that is Papaver somniferum, Atropa belladonna, Lactuca sativa, barley flour are commonly used in insomnia as shamoom. Tila. Tila is liniment, which is diluted, oily or watery preparation used topically on a particular part of the body is called tila. Kurse musallas with Abe Kashmir subs coriandrum sativum is applied over forehead. Kahu means Lactuca sativa, belladonna or handbane, and Papaver somniferum, each in equal quantity, boiled and apply as tila on forehead. Tila of Kurse Anzurut is highly effective method of tabliğ dimag, that is cooling of the temperament of brain. Zimad. Zimad is a kind of paste. It is semi-solid preparation applied externally. Applying paste of Ushna stone flower, Cyprus scariosus, leaves of hemp, that is cannabis sativa, and goat milk over soles. Sandal Safed, that is Centellum album, which with Abe Kashmir subs, Coriandrum sativum, or Abe coconut, Papaver somniferum, removes excessive heat and induce sleep.
Dr. Sabha, I think we have one more minute to finish. Can you finish it a bit sooner? Because we have questions as well.
Okay, ma'am, I'm finishing it. As
Yeah,
you know,
then some background noise. Can you take care of that and be a bit more quicker? Yeah. Thank you very much.
Okay, ma'am. It is completed, ma'am.
Okay. Yeah. Are you done? Can you hear me? Can you hear me? Hello? Hello, can you hear me, Sabha? Dr. Sabha? Hello? Okay, I think she's disconnected or so. I'm not sure. Sabha, can you hear me? Sabha, are you here? Yeah, I think Sabha is connected, yes, but I couldn't hear from her. She's also an unmute. Okay, I think she left. Maybe we should move on to the next presenter. So do we have Poonam Agarwal or anyone from her team? Please say yes if you are here. I think Okay. Okay. Emil has a question. I would like to answer that. She's asking, I have a question about the certificate. Does it include the name of co-author? Since you presented, Emil, so you will be getting the presenter certificate. I hope this is clear. And anyways, all the presenters' abstract will be added on to the conference proceedings. So, that will have the whole set of your team anyway. Since you presented the topic, you are the presenter, so you will receive the presentation certificate. Yeah. Okay, so now we have Poonam Agarwal. Can you hear me, Poonam? Poonam, can you hear me? Poonam, can you hear me?
Hello.
Hello, Poonam, can you hear me?
Hear me.
Yeah. Kindly share your screen.
Yes, ma'am. Don't share it.
Okay.
Share option or cannot be seen.
So you have the option after, like next to the video mode, okay?
Connecting.
Okay, can you share your screen, please? It could be your phone. Yes. Yeah. I think there is so much background. Hello, everyone.
Yes, ma'am. Yes.
There's so much background noise. Kindly mute your audio, please, participants. It was quite disturbing. Okay. So, yes, you may change it to landscape mode if that works. Yeah. Yeah, you may start, yes. Can you hear me, Poonam? Poonam, can you hear me? I couldn't hear. Okay. Hello, Poonam. I couldn't hear you. Can you start from the beginning?
1 second. Yes. Good afternoon.
Good afternoon, and we could hear you. Yeah, you may start, yes.
Hear me? Hello.
Hello, yes. Yeah, you may start, yes. Can you hear us?
Okay. Good afternoon, ma'am.
Yeah.
Myself Poonam Agrawal, Assistant Profession in Raigad College of Pharmacy. Today topic is one second. Experimental treatment with the broad spectrum antiviral drugs. Sorry. Experimental treatment and its broad spectrum of antiviral drugs are used for the treatment of the influenza and COVID-19. Hear me? Hello?
Yes, yes, we could hear you. Yeah. Yeah, you may continue. Yes.
Introduction. Unable to the first systemic antiviral therapy and many antiviral agents have acyclovir subsequently been developed. Acyclovir target happens simplex virus and they are the varicella zoster infections and their individual antiviral against the targets one viral infection caused by the one virus, and they are so similar family of the antiviral against. The favipiravir shows better efficacy in regarding to influenza infection, having the osteomalavir Tamil flu as a brand name, its efficacy in the treatment of the pathogenic avian influenza, and they are the oseltamivir resistance virus has been confirmed in animals. It is being developed and manufactured by the Toyama Chemical. Fujifilm Groups has been approved by the medical use in Japan 2014. In 2016, the China one company will be approbation by the API. It becomes generic drugs in 2019 for India. These are the chemical structure of the favipiravir drugs and its broad spectrum of RNA virus activity. A compound that first shows anti-influenza activity was often in the screen and of antiviral drugs treating influenza, and favipiravir was optimized in terms of activity and stability in animals, suggested that the chemical structure of the active compound and its derivatives includes favipiravir resembled a nucleoside analog, and the presumed mechanism of action was to inhibit RNA synthesis. It involves mainly three chemical compounds, that is the 5-amino imidazole, 4-carboxylamide, 1-beta D-ribofuranoside, which are biosynthesized from amino acids. All antiviral drugs like ribozoal, favipiravir, and ritonavir have similar chemical structure, suggesting that favipiravir functions as a purine homologue and inhibits viral RNA synthesis. Favipiravir is mainly incorporated in the several pathway for the purine nucleotide through the purine phosphorybonyl transferase, and it's further phosphorylated to favipiravir diphosphates is a substrate for viral RNA-dependent RNA polymerase. The phosphorylase of favipiravir are the hypoxanthine guanine phosphoribogel transferase was confirmed in the subsequent study. Favipiravir shows anti-influenza virus activity towards all subtype seasonal influenza virus strains. These are the chemical structure of the favipiravir. Having generic name is the favipiravir and brand name is the Abingane. Mechanism action of favipiravir. Mechanism action of favipiravir. Antiviral activity of favipiravir was attenuated in the presence of a purine nucleosides or purine bases, including the competitions of the favipiravir with purine nucleosides rather than the pyrimide nucleosides. Median Darby canine kidney is a scientist. The cell are well used as a in vitro assay of influenza virus. These cells were treated with the favipiravir and cellular metabolites were analyzed by HPLC. Favipiravir ribofuranosyl 5-dex triphosphates, favipiravir ribofuranose, and favipiravir ribofuranosyl 5-dex monophosphates not detected. These results suggested that the activation of favipiravir takes place when it is incorporated into the cell. Favipiravir ribofuranosyl 5-dex triphosphate were chemically synthesized, which tested in the inhabitants of the RNA polymerase activity of influenza virus is assessed by the incorporation of 30 pGTP is the channel of the receptors. Favipiravir RTP inhibits the viral RNA polymerase activates in the concentration ranging from the nanomolecule to micromolecule. None of favipiravir and favipiravir RMP affected influenza RNA-dependent RNA polymerase for the 100 micromole per liter. These results indicated that the favipiravir except its antiviral activity as a prodrug. So its prodrugs of activity is the chemical activance is favipiravir ribofuranosyl triphosphates. The viral genome and RNA dependent RNA polymerase complex are transported to the nucleus where the transcriptions of viral RNA synthesis begin. The synthesis of RNA does not contain the cape structures required for mRNA, and the cape structures from the host mRNA is excited and transferred to the viral RNA. In the cape-dependent endonuclease of viral RNA-dependent RNA polymerase complex, resulting in the formation of the viral mRNA capes. After viral mRNA is produced, the viral protein is translated. The viral proteins and RDRP RNA complex form viral particles that subsequently bound to the cell membrane and thus inhibit the viral RNA replication and inhibits the growth. Research suggested that the favipiravir include lethal RNA transfers of mutations producing non-viable viral. These are the favipiravir structure will absorb or incorporated to the enter into the cell, and during phosphorylation process the favipiravir are enter into the cell. And RNA polymerase, RNA dependent RNA polymerase will the RNA cell in body, and during the replication process they will be assembled and releasing the effect. Conclusion: favipiravir was discovered by phenotypic scanning against influenza virus at research laboratory of the Toyama Chemical Co. Ltd. We undertook further study to investigate the mechanism of action and antiviral effect against virus types of viruses. Favipiravir is phosphorylated in the cell to be an active form. Favipiravir RTP, which is recognized as a purine nucleotide by RDRP and inhibits the RNA-dependent RNA polymerase enzyme activity. Favipiravir is the exhibit no effects on DNA-dependent RNA or DNA polymerase. Favipiravir is currently approved the influenza infection in Japan and it's advanced chemically dependent in the US evidence. To date from chemical studies, so there the favipiravir is well tolerated in human. Wider antiviral spectrum of favipiravir was to purpose clinical research for the devastating virus infections such as the Ebola and SFTS. We strongly believe that the favipiravir with its unique profile will be promising therapeutic agents for unremediated infection by the RNA virus in the near future. This is my reference. Thank you.
Thank you, Dr. Poonam. And okay, so any questions, audience? Audience, any questions? Now, do you have a question? So, participants, please mute yourself, mute your audio. Okay. How different is influenza infection to COVID?
Influenza infections having similar types, but influenza always is the fever and their breathing difficulties. COVID is similar. So slightly but different. They are the influenza are notly long affected for the breathing or lungs, but COVIDs are more long times they will affect it.
Okay. Okay, thank you, Dr. Poonam. Thank you. Thank you for the good presentation.
Thank you so much, ma'am.
Thank you. And I think we have one more as well. Let me see. So, okay. Do we have Nilofer Shaikh or Renu Vyas or anybody from this team? If you are there, I think this is the last presentation, poster presentation of this e-conference. Anybody? So I think they are not available. Yes, Mr. Papri Basak, I understand that we have to limit the time of oral and poster presentations, but we have given 10 minutes and 5 minutes for oral and poster presentations respectively, but sometimes it's difficult to it's important to appreciate and encourage students as well, students and in general the presenters. So sometimes it does go beyond time. We apologize for that. Uh, yes, and I think if that is all, we can go with the valedictory session. So do we have the faculties of uh, VIGNAN'S Foundation for Science, Technology and Research here? Do we have Abraham Venkateshwaralu sir?
Hello, Divya madam.
Hello sir.
Hello, Divya madam.
Yes sir, we could hear you.
Yeah, madam. I think all participants have completed, no, madam.
Yes. Sir, it's echoing time. My voice is echoing there.
Madam.
Yes, sir. And okay.
Hello, madam.
Yes, sir. Yes, sir. Can you hear me? Can you hear me? So participants, please wait for a few minutes. Uh, we will be starting the valedictory session soon. Yes, sir. Can you hear us? Yes. Uh, your voice is not clear. It's breaking, sir. So we may announce the best. We will announce the best poster and the best oral presentation award in 5 minutes time. Kindly bear
Hello,
with
madam.
us. Hello, sir.
Hello, Divya madam.
Yes, sir. Yes, sir. We could hear you. We could hear you. I think you are in mode. We are. I think you are in mute mode.
Divya madam.
Hello, sir. Can you hear me? Yeah. Yeah. Can you hear me?
Yeah, now I can hear, madam. Thank you.
So I just mentioned that the best oral presentation and the poster award announced in 5 minutes.
Yes,
Uh,
madam.
yes, sir. So please, participants, please bear with us for a few more minutes. So participants, you will be, so we will play a video of the of few words that has been conveyed by the chairman. So here it is. So I mean the chairman of the VIGNAN'S Foundation has given a video record in YouTube. So we will play it for you all.
I am Dr. L. Rattaya, Chairman of Vignan Deemed University, situated in Guntur. I am happy to learn that our biotechnology department, headed by Krupanidhi and other professor Venkateswarlu, organized a 3-day international conference on COVID-19. And the trends, diagnostics and therapeutics, this is the topic of the conference. It was organized very successfully from the last 3 days. And in this conference, about 7 to 8 foreign professors are participated. Another 4 or 5 industrialists also participated in this conference. And there are around more than 1,400 participants registered for this conference and participated actively. And there was an interactive session during this conference. I came to know that it went on very successfully. I am very happy during this crisis also, during this ambiguous period also, our department, Biotechnology Department of Vignan University, successfully conducted this conference involving so many renowned professors and industrialists. But as all of us aware that this COVID-19 is a war on countries the world over. I mean, the whole humankind is suffering from this. It is more than a war. We do not know the potential of this virus. We do not know how it enters into a particular area, and we do not know what is the remedial measures for this. And so far, no drug is discovered, though it is some of the companies are claiming it is not proved beyond doubt. The whole humankind is playing in great uncertainty. This is a very peculiar situation. We do not know when this virus becomes passive and it would be killed. The human race will get resistance to contain this virus. We do not know. We are playing with a lot of uncertainty. Playing with uncertainty is a great deal. The whole humankind is suffering from this. During this time, the scientists and the doctors and the industrialists sit together and come out with some solutions. And I hope and I wish the real solution will come out in the next few months to save the humanity. And the whole thing is, this is one side of it. So discovery of a drug and implementation of it and all these things is one side. The other side is, I request all the participants and also the public in general, so the most apt thing is we have to develop the immunity to resist this virus. I mean, to contain this virus. So to develop immunity, we have to have a good food, a small exercise, exposed to sun and free air, and also sometimes very specific food, which gives the glycine and other elements, greatly help the human beings. So we have to try both sides. So one side is the discovery of a drug, something from doctors or the scientists to counter this. The other side is to develop the immunity so that we can resist to this virus. So in this, it's one way it is crisis and another way it is a very interesting situation. In this situation, the conference was conducted very successfully. I really thank all the delegates who have participated in this conference, and really from the bottom of my heart, I thank all the professors, especially the international professors who have participated very actively in this conference and made the deliberations useful for the delegates. And I hope and I wish all of us sit together, one side the scientists, the other side the industrialists, the third side the academicians, sit together and come out with a solution in the next few months so that it would be a great help for the humanity. So far, the countries have tried with nuclear weapons and all the other things. Now it is the biological weapon. Whether it is created or a natural phenomenon, we do not know. But this biological weapon is more dangerous than any nuclear weapon, any atom bomb, any kind of thing. So we have to be more careful. We must be ready to face this situation. And scientists and academicians and other people will be part of finding a solution for this. Once again, I thank you all. Thank you for all the delegates and everybody participated in this conference. I hope and I wish this kind of conferences should be held very frequently till we get the right solution and a reasonable solution to save the humanity. Compared to many other countries in the world, India has registered very low percentage of COVID cases. So number of cases is close to more than 1 lakh, but the percentage is very less. It is because of the Indian traditions and Indian culture. So taking the food itself in India is mostly antiseptic and also creates a lot of immunity. So that might be one of the reasons. And the culture is also normally Indians, they keep distance and pay respects through namaskarams. So because of all these things and also the lockdown process, India has relatively registered less number of cases, less percentage of cases. I hope and I wish that Indians will continue their traditions, especially the traditional food, to maintain their immunity levels, and hope the other country people, because there are many participants, I request them to study the Indian foods and habits, and if they are good, we can introduce this kind of culture in other countries also to attack and to resist to the COVID phenomena. So I am happy to learn this conference was being held with the cooperation of many organizations. One organization is Kansas State University from America, Andhra Pradesh Academy of Sciences. The third website provider is LLB, and other university is Al-Quds University of Palestine. So these people have given a lot of support and made this successful. I thank organizations on behalf of the university. As the chairman of the Vignan University, I thank them. Let us have a common platform in the future also. Let us grow together in the future also. I thank these universities and also the organizations very much for making this conference a greatly successful. Thank you.
Yes, now you can say, sir. Now you can be online. Yes. He will be giving concluding remarks and poster and oral presentations. Dr. Virulu, sir.
Hello, madam.
Yes, sir. Yes.
Can you hear me, madam, now? Hello, Divya, madam.
Yes, sir. We could hear you.
Yeah, madam. Now I can start, madam.
Hello, sir. We could hear you.
Yeah, madam. Can I start, madam? Closing.
Yes.
Yeah, madam. So I am Professor T.C. Venkateswarlu. So Deputy Head, Department of Biotechnology. So VIGNAN'S Foundation for Science, Technology and Research, GUNTUR. So now there's a 3-day international e-conference. So we have successfully completed. So this e-conference is hosted so in association with the LLB-SCHOOL, and Andhra Pradesh Academy of Science and Kansas State University, USA, and Al-Quds University of Palestine. So the e-conference is successfully hosted by LLB-SCHOOL. So from our side, we deeply express my sincere thanks to Divya Madam, Mahesh sir and Pranav sir and their team. So the sessions has successfully went on all 3 days. So in 3-day international e-conference, a total of 18 speakers, invited speakers, so they have said their views. And apart from this, so more than 45 participants, so from different corners of the country, so they have actively involved for delivering their talks, were oral and poster presentations. So the authors, the invited speakers, they have shared the many ideas, so they have shared their knowledge. So this must helps very much for a young budding scientists and students and other scientific community. So which will also helps in this pandemic period, which may helps for development of the vaccine or other suitable drug. And I express my thanks to also a professor today, end the talk by a Professor Venkateswarlu Kanumarlapudi. So this professor has voluntarily given his talk. So he has given an elaborated lecture on developing a novel antiviral treatments for COVID-19. And also a industry experts by name the Girinath Pillai sir. So he is the director and founder of Jasra Innovations. So he also given a computational approaches. He explained related to the drug design and dynamic simulations, so which will help a lot for developing and studying drugs at in silico level. And one more speaker I must especially thank by name Panduranga Rao sir. So he is working as Associate Vice President at BioVit Private Limited, Bangalore. So sir has explained the vaccine platforms, and sir also updated the different current status of the vaccine developments, and he also explained how many companies from India involved for development of vaccine. Then sir also expected the vaccine may be launched soon, and recently the Bharat Biotech, they also got approval for trials. And and also a doctor from USA by name Dr. Roger. So he also given a elaborated information. He also given a very brief insights on COVID-19. So it is a very generalized talk. This helps for a lot of common people and students. Sir has given a special emphasis on various symptoms and taking measures for a COVID-19. And other speaker from Australia by name Rajalakshmi Madam, she also supported a lot, and Madam has delivered his talk on artificial intelligence and machine learning approaches in drug discovery, especially against the COVID. And Ho Liang sir, so he's also a professor, associate professor from Kansas State University. So he also given a wonderful talk on AI-driven drug discovery. And another doctor from Siddhartha Medical College by name Dr. Raman Kumari Madam has given a talk on peripheral blood picture. His madam has said the practical evidences from the pathology lab, Siddhartha Medical College. And in the today's talk, Doctor started with Perugu Shyam sir. He has given a talk on small molecules driven drug discovery. He also enlightened on a natural products. He enlightened about the turmeric, and he also mentioned the usage of Ayurvedic based products for treating the COVID-19. And Amit Ajita, she also given the talk, wonderful talk on stem cell. And another speaker by name Sandeep, so he's working as a director clinical development Genesis Diagnostics. So he has given a talk on development of a molecular based diagnostics tools for detection of the COVID. So he is actually a busy fellow. Inspired them. He has given a talk. So they are also working on CRISPR based diagnostic tools for detection of the COVID. And Andrew Mibiti sir has given the impact of nutrients on balanced immunity. And I already mentioned the Professor Venkateswarlu Kanamurlapudi. He has given elaborated talk, today's end talk by Professor Venkateswarlu sir. So with this, I am concluding. I also express once again my deep thanks for our Honorable HOD sir, Professor S. Krupanidhi. So we have successfully executed this e-conference by the support of Professor S. Krupanidhi, and also a energetic, active organizing secretary by name Dr. Abraham Pile from our department. So he also helps a lot for accelerating this e-conference. And at the end now, my deep heartfelt thanks to Divya Madam, LLB-SCHOOL, the co-founder of the LLB-SCHOOL. So the Madam has hosted all sessions. So with the help of Mahesh sir and Pranav sir. So we thank you, Madam. We thank you once again, Madam. In future also we'll continue hosting some more FDP programs, Madam. With this I'm concluding. Thank you. Thank you, Madam.
Thank you, sir. I think, yeah, in few minutes we will announce the best oral and poster presentations.
Madam.
Please be patient. Participants, please be patient. Just for a few more minutes, we have some clarifications. We will come back to you. Participants, you want to say something about our LLB school? Pranav, can you hear me? Pranav?
Hello.
Hello, Pranav. Okay, we can play some videos and do you have anything?
We can share our channel actually.
right? We can show option. One second.
Again. I have unmuted you. I sincerely thank Pranav and Pranav for also equally handling all the sessions, for hosting the sessions, in fact. And he presentations. So I sincerely thank Pranav for supporting me, and also I thank Mahesh, the founder of the LLB school, uh, who also equally supported in conducting this conference. So if participants, if you want to, yeah, Mahesh, if you can unmute yourself, you can also give your words. You can speak a few words. So Mahesh was equally responsible. Mahesh was responsible in developing this LLB scientifically channel, and he is the brain behind this LLB groups that we have at the moment, and we facilitate researchers all around the globe in answering to queries, and also in conducting webinars. So if you have any research queries, you can always post. There are always somebody who's there to help you through our Telegram platforms. And anything you want to know, you may put it in the chat box, and I could answer in the meantime. We have uploaded a lot of videos on different topics until now. So we request you all to subscribe to our LLB Scientifically channel. And here you see a lot of videos we have uploaded on different topics related to biotechnology and bioinformatics. So, and if you want to learn some programming, for example, if you want to learn Python, R, we have people who can support you with that. We have experts, and these experts are university alumni and who are currently doing postdocs or who have done their PhDs and are working in various labs across the world. So these experts can definitely guide you through your doubts. Yeah, I think Python is applied in biotechnology as well. So, I mean, I either use R or Python to do all my statistics to validate my data, my quantitative data. So that's one example, but Python or R or Perl, they have variety of applications. So if you want to learn, you can always look into our videos, and we will also be organizing different levels of Python courses in the upcoming months. So kindly kindly subscribe to our YouTube channel, as well as please join our Telegram groups and Telegram channel to know more updates on it. So at this moment, I should really thank the organizing committee, Dr. Abraham, and Professor and Professor Kripanathy sir. You may also see one of his talks in the talks given to LLB school in the uploaded videos. So Dr. Kripanathy sir is such a knowledgeable person, and he was very supportive and very encouraging throughout. And the Foundation and yeah, I sincerely thank VIGNAN'S Foundation in the future workshops or conferences to come. We will definitely be supportive. We will definitely be supportive. And again, like, thank you, participants, and congratulations and all the best for your research, and then for many more success to come. And we should say one thing, that we organize a lot of journalism webinars. So definitely time for you to share your knowledge. For LLB school. So here it's written like what we are actually doing, and who we are, and what's our main objective. And the future, it depends, because we are actually we have recently started this organization, so we have many more miles to go. So our ultimate aim is to bridge the vision of biotechnologists and bioinformaticians to conduct healthy science and help the novice to ask and discuss any biotech or bioinformatics related queries. So in addition, we will also post the latest updates about science, anything like biotech or bioinformatics related topics. And we will also update with some jobs or internship opportunities, competitive exams, etc. And we will give access to some paid or unpaid. So we do have many more plans, and we will include it one by one. So our next plan is to develop a website, and that is already in process. So people can definitely get all the updates through the website. At the moment, we are actually communicating science through Facebook, WhatsApp, LinkedIn, and the Telegram platform mainly. And we are also in the process of developing a website. So, and soon you will get all the updates, and all the things that we are doing in Telegram will be translated to the website, and you can watch and you can see everything. So thank you very much, and so I think the updated, so we have actually this kind of a old invitation. When we actually started this LLB school, we did this. But so once you receive proceedings, you will actually get the full picture of what, LLB school and what we are doing. So you will receive it soon, and that will be released by VIGNAN'S Foundation. You can also have a look about what they are actually doing, what VIGNAN'S Foundation is all about, and what they are doing as well, in addition to what actually LLB school is doing. So I think we got the results. Please hang on. Hello everyone, can you hear me? Yeah, and thank you for being very patient. And now I request Dr. Professor Venkateshwaralu sir to announce the results for the.
Yeah. Good evening, Diva Madam.
Yes, sir. Please announce the results.
Yeah, Madam. We have received a good many abstracts from different corners of the country and world. So we received total of 112 abstracts, especially related to the COVID. Then out of that, we have selected 55 for oral and poster presentations. And out of selected 55, more than 45 participants have presented, have delivered oral presentations, and also delivered their poster presentation. So among all, our committee and with support of LLB-SCHOOL, so we have evaluated. So finally, we have selected 3 oral presentations, and we have selected 3 best poster presentations, and certificate of appreciation, we have selected 3 students. So now first I will announce the certificate best oral award. The first prize goes to Miss M. Pratibha, Department of Medical Biotechnology. Oh, yeah. Now I will announce poster first. The first prize is the Miss M. Pratibha, Department of Medical Biotechnology, IIT Madras, and the second poster prize is Dr. Abhishek Singh Gupta, Department of Biotechnology from Amity University, and third poster prize Dr. S. Jagdish Kumar from VFSTR, and best first oral presentation to Dr. Vinod Kumar from School of Chemical and Biotechnology, Sesra University, and second oral prize Dr. Rohini Krishna from VFSTR, and third prize yes Kalaivani Priyadarshini from Aquatic Animal Health and Environment Division, ICAR, Chennai. Another participant, another supporter, Department of Biotechnology from Lady Doak College, Madurai. And apart from these 3 certificate of appreciations, so one fellow by name Dr. Estila from University of Madrid, Spain, and Mr. Sriharsha from Friedrich Alexander University, Germany, and third appreciation certificate G. Aishwarya, VFSTR, and also 2 special my request from organizing committee. One faculty and one scholar have helped in organizing this e-conference. So for them also one appreciation certificate by name Mr. Miss Nagalakshmi, Assistant Professor, Department of Biotechnology, VFSTR. Then one more scholar by name Sivakumar from VFSTR. Thank you, madam.
Thank you, sir. Thank you, sir, and hope you're all convinced with the results. And thank you very much for attending this conference, and thank you for the support given to us. We will continue to associate with VIGNAN'S in the future, and thank you very much. Thank you all the participants. So the certificates, I mean, with regard to e-certificates of participation, you will be receiving them in a few days' time upon feedback form submission. So the feedback forms can be filled only until 12 a.m. midnight. So, okay, yeah. Please fill the feedback forms until before 12 a.m. on or before 12 a.m. tonight. So, and then we will send all the participation certificates in a few days' time. About the poster presentation and the paper, I mean presentation certificates, we will be sending over to the individual mails by next week. And the best poster award and the best oral presentation award certificates will also be done the same way. So you will be receiving them to your individual mail IDs by next week. Thank you very much, and I hope it is clear now. So the certificate for all the presenters, we will be issuing the certificate presentation only, not participated. And for other registrants or those who participated the conference, in the conference will receive as participated. Those who presented will receive it as presenter. I hope it is clear. So I mean, those who participated as well as presented in this conference will actually receive 2 certificates, provided if you had filled the feedback form. I hope it is clear for all the viewers, all the participants. If you have any other questions, please immediately type it in the chat box. I would be able to answer. I hope it is clear. Okay, thank you very much, and thank you for all. We heard a lot of, we heard a variety of presentations, and it was very interesting. Although time consuming, although it was a long day for the last 3 days, still we enjoyed listening to all the presentations, and we appreciate your commitment as well. Thank you very much.
Hello, Divya madam.
Hello, sir.
Madam, Abraham wants to thank you, madam. Please, madam.
Okay, sir.
Hello.
Hello.
Yes. Thanks for the opportunity for giving me an opportunity for connecting this international e-conference, madam. I thank my university, Vignan University, VIGNAN'S Foundation for Science, Technology and Research, deemed to be university. And I thank my Professor Krupanidhi as well as my Professor Venkateswarlu for giving me this opportunity. And I personally thank you, Divya ma'am, for giving us great support in conducting this great conference. And I also thank all the participants from India and outside the India. Once again, I thank you very much, Divya madam, for making this event so successful.
In fact, you all gave us this opportunity. We should thank you a lot. I think we have no words to fill in. So thank you very much.
I am looking more forward to work with you, ma'am. Thank you so much.
Thank you, Abraham, and thank you all the. You may leave the meeting now. Yeah.